Again, these bivalves, they belong to the genus Calyptohyena. Calyptohyena is
Another one.
a composite of two words. Calypto, it's the nymph in ancient Greece, Calypto, and hyena most likely is coming from the Greek verb hyeno, which means give birth.
So for people here online, we just started the stream again, and I'm going to say goodbye for now and leaving you with Dr. Lawrence Seiler, that she's the principal investigator on this exciting Atacama Trench ancients expedition, and I think you will be enjoying his narration while we keep sampling this methane seep that we just found. So, muchas gracias a todos por escucharme durante este par de horas, y ahora os dejo en la compañía de la doctora Lawrence Seiler, que es la co-investigadora principal de esta expedición de Atacama Trench, en la que acabamos de encontrar emanación de metano que estamos muestreando ahora mismo. Muchas gracias a todos y hasta luego.
I cannot pass this opportunity and comment that our colleague Anthony also has a very nice voice that he could be using as an ASMR narrator. If the audience thinks so, please let me know.
No,
Both him and Andrew.
It's
I
very
mean,
true. Yes, and Drew too. You have a very nice speaker.
you guys could be moonlighting as ASMR narrators.
Drew, you need one more cord. Do you want to try to take it from here, or do you want to take it from the other side? Because we can extend our dive time another 4 hours.
I am fine with trying to get it from the site. I was under the impression that the site was probably not viable.
It might be difficult to get you another intact core. We've been having trouble getting.
I feel guilty of taking more course here.
Yeah,
For me.
I don't want. I feel a little strange.
Yes.
I'd rather than move
Oversampling
it to
it.
the site.
Let's move to the other one.
Yes.
I'm happy to.
Lori, may we take a rock sample from the rocks around? I don't mind which ones. Just so before we leave the place, just
Okay.
make it be a low sample. We don't need to break the rock first.
Okay, can you just point out to me where you would like it?
When we are moving the
Antonio, okay?
Sorry, everybody kind of dropped out of the conversation for a moment there. This is Dr. Lawrence Seiler from Stockton University. We are currently off the coast of the Atacama Desert in Chile, and we are sampling what we believe to be, well, it has not yet been confirmed, the deepest methane seep ever found off the coast of Chile. We spent most of the day and a good chunk of 2 days ago searching using bathymetry data, backscatter data, and subbottom profiling. And we finally stumbled upon this one, and it's not very big, but it's still ours, and it's beautiful. We're trying very hard not to completely disturb this area while we sample. There is another site that we found nearby, and so as soon as we're done taking samples here, we're going to hop over there, take a few quick cores, and then I believe we will spend the rest of the night until early morning tomorrow looking for more active seeps. We've been collaborating with numerous people and talking to the ROV pilots and other folks on board the Falcor 2 who have experience in looking for seeps. They think that this is a very, very promising area. And so we're going to do our absolute best to see if we can find more active seepage along this site all through the night. So we'll see how long I last. Right now I feel pretty awake. I'm pretty jazzed about it. So we're just going to do the best we can.
I
think 8. They said they could do another 4 hours. Alrighty.
So this is specimen number 5.
Specimen number 5, Biobox 2B. And then we're going to try to grab just one more. And then I think we'll be on our way.
What do you want here? What do you want here?
We just want one more, please. For 6 total? Thanks. Sorry.
Para examinar la variabilidad en esta población, por ejemplo. Para competencia.
Those little squat lobsters are so funny. I like watching them swim backwards. It's like they're going butt first into everything. I love it. These clams are super beautiful. Can't wait to characterize them. They're so weird looking. Drew, are you logging now? Who's logging right now?
We go.
Okay. Are you sure you don't need a break, Mika?
No, I can stay until 2. No
Okay,
problem.
you've been at this for hours.
Yeah, I will end up being here for 8 hours, but it's okay. But I will leave at 2.
Oh my God. All right.
It's an ultimatum. I will leave at 2.
All right. Miguel's ride or die.
But I'm fine for now, so it's not.
Okay. It just, you're allowed to take breaks too.
Yeah. I had a glass of water like 1 hour ago, so.
Excellent. As long as you're staying hydrated.
The modern graduate students are amazing. They can run on water. So that is...
Unbelievable, right?
Yes.
Real facts though, no, not even joking. Our graduate students have been so incredible. They work so hard and they have so much initiative and they're organized and they've just done amazing work for this entire cruise. All of our sampling efforts have run incredibly smoothly because they came up with a whole plan for getting samples off the ROV and off the CTD and they've just been so systematic and everything has just run like a well-oiled machine thanks to their efforts. So shout out to all of the students who are on this expedition. You're incredible.
Here, here.
Thank you.
That's the last clam we're going to take from here. So that should cover all of our needs for characterization. And Antonio, you said you needed a rock sample?
If possible, yes.
Okay. And Drew, you're going to take your core from the other site?
Yep.
Okay.
those two also.
Yes.
Okay. So we did a little bit of both. We tried to, yeah. Okay. Yeah. Yeah, just try not to damage the site too badly as we're sampling here. We're trying to be responsible biologists. This is, after all, a very interesting and unique ecosystem, and we want to study it and understand it better, but we don't want to destroy it in the process. So there's another similar site nearby, so we're going to take a couple of samples from that one as well, try to space things out, make sure that everybody's sampling needs are met, and then we'll have some time to explore the rest of this ridge.
Tiene caballitos encima. ¿Ninguno? Porque podemos preguntar para un school, entonces la otra es site número 2. Ahora vamos para site número 2. Sí, voy a preguntar para el anthropogenic también. What's my site? This is white. Just in case cancer. Eso tú crees, no encontramos esta pepino de mar otros días,
Oh, yeah, yeah.
Did we drop the probe? Oh. Yeah, Antonio wants a rock. Antonio, can you show us where you would like a rock from, please?
Yes, from the surroundings. So
You can use the pointer if you want.
Tiene 4 tentáculos en la dorsal.
Those three are
Okay. All right, we're just gonna grab some samples of rock for one of our geologists. Just need to get our probe out of the way here first so we don't squish it. A lot of you are up late. Oh my goodness. Someone said it's 5 a.m. where they are. That's hardcore. Respect. That's incredible. Staying up late for the love of science. That's what I'm planning to do tonight. So, that's outstanding. All right, we're just repositioning the probe here. And then we're going to set down and take some rock samples. I apologize for not having more to say right now, but we're kind of running logistics and also trying to get everybody their samples. And I've been up since about 6 a.m. So, starting to run out of steam a little bit, but it's okay. I'm hoping for a second wind any minute now. I'm normally not a morning person. I am absolutely a night person, but life on a ship doesn't necessarily respect your usual sleep-wake cycles. You work when the work needs to be done, and that's that.
Sì, entiendo. Sí, sí, puede usar como el
I'll
biofilm.
certainly do my best
Okay,
to
sí.
send some wind to the folks in the chat. Some of you have been with us all day. I'm impressed. This has been a long dive. And we're not done yet, so.
Sí, entiendo. Okay. Okay.
Oh. Did you just drop the probe again? Oh, okay. I'm having fun looking at this and doing this work and having all of you here with me, so that's wonderful. Oh my goodness. A 24-hour dive hanging for the whole time? Oh my gosh. I don't know if I could do that. That's really impressive. We are super grateful for all of the folks who are watching us and joining in on this exploration with us. It's been a truly incredible day. A lot of emotional ups and downs, but a truly incredible day nonetheless. The energy in this room when we found this first seep was impossible to describe. It was really amazing. I'm not sure if what we're looking at right now is bubbles. I think a lot of it is just sediment being kicked off the bottom. Ordinarily, yes, methane seeps. Well, I don't want to say ordinarily. Often methane seeps will produce lots of bubbles. But the ones in Chile, according to the literature, don't tend to bubble, and this one is no exception. We've seen like a lot of little tiny bubbles around the clams and in the mud at some of these sites. It's possible that they would be bubbling more if they were in shallower water. Right now, we are at over 2,800 meters, and so there's a lot of pressure down here. We're talking over 280 atmospheres. And so that might be keeping the gases in solution and keeping them from bubbling out. We may also be looking at a seep on the decline. This one's kind of small, and we're seeing some dead clams here, tubeworms. And so we suspect that this might be an older methane seep. Methane seeps do have a lifetime that they reach the end of eventually, and they can be very short-lived, depending on how much gas is trapped underneath the sea floor. And eventually that methane does run out. And so we may be looking at a methane seep where the methane has started to run out. So after we're finished sampling these two sites that we've found, the plan is to move along to further up the ridge. We have a few more sites that we would like to check out. We've extended the time of the dive, so we have a little bit more time to explore this area, because according to our collaborators and the folks that we've been working with at SOI, this looks like a very promising area to find more seep activity, and we've only just started to explore the upper part of this region. We found this seep. We started looking around this little hill, and we found a little bit of seep activity. So we're hoping that if we continue up this ridge, up this anticline, we will find even more. So the night is young, as they say. If you're a night owl, you're in luck. Or if it's daytime where you are, even better. Good question. So it can be methane generated by geologic activity, or it can be methane generated by decomposition. So there's actually two different types of methane seeps. The ones that we're hoping to find here, and what we think we found here, are sometimes called true seeps, and these are formed along subduction zones where one plate is sliding underneath of another plate, and the plate on top starts to buckle and form ridges and bumps. And underneath these ridges and bumps, methane gas collects and then seeps to the surface. There are also seeps that are caused by deadfall. Sometimes these are called false seeps, and this is where a lot of decomposition is occurring. It's either to high productivity or something like a whale fall. It gets buried, and then methane gas produced by all that decomposition seeps up to the top. And I believe that these types of seeps are shorter lived than the ones that are caused by geologic activity. Both are interesting, and we would be happy if we found either one. But we think what we're looking at right now are geologic seeps, or true methane seeps, because we're in a really good setting for them. Did we get Antonio's rock? Or are we about to get that? Okay. We're going to get Antonio's rock. And then we're going to go to the other site that we found and collect a few more samples. And then we've got a lot more exploration to do. Do you have the tools to isotope the carbon to determine which it is? Not currently with us. The geologists are taking some samples of the rock and the sediments, so they can perform some more work on those. But yes, that is a very good point. You can check the isotopic ratios of carbon to determine whether it has a biotic or abiotic origin. Carbon-13 is a stable, heavy isotope of carbon, but it's not as common in the environment. And living things tend to preferentially take up the lighter isotope, the carbon-12, which is more common. And so the ratio between carbon-12 and carbon-13 in substances will sometimes act as a clue to tell you whether something was made by an organism or by a geologic process, because geologic processes don't selectively take up one isotope or the other. You've kicked up a ton of sediment around here. The substrate down here is extremely fluffy. That's due in part to the fact that we are underneath a area of very high productivity, because there is a large amount of seasonal upwelling here. It produces a lot of productivity in the epipelagic, the top layer of the ocean, that eventually sinks down to the bottom as detritus. And we also think that the clams are further loosening up the substrate here. So we tried to take a couple of cores in this spot, and for the first time on this cruise, actually, we had a few cores fail on site. We pulled them out and they just slipped right out of the push core. So we're going to go to the other site to try to take some more cores, because we think we've exhausted this site for usable cores, and we also don't want to completely damage this natural ecosystem that we've just found. Seems a bit rude to stumble in on somebody's house and just start taking everything and turning everything upside down. And that's the tricky part of science in general, and I think biology in particular, is that whenever you study a system, you want to disturb it as little as possible, not only to ensure that you're getting usable data, but also out of respect for the organisms that we study. Yeah, Aparna, are you in back? Yeah, we can take another bottle for you if you want. At the next site?
I don't need too much. I just need 500.
Okay, we can take one of the small ones for you. Yes. All right, so we're just going to grab a rock sample here for one of our geologists, Antonio, who was just narrating. If you've been here for a bit, you got to hear the dulcet tones of Antonio. We were just talking about how he has a really nice voice. I think a lot of our dive watch folks have really calm voices. Calming ASMR.
Alright, that's fine. No, we understand it, alright.
Is that a sea anemone right there? On the left?
Yes.
Oh, that's a pretty one. Yes, when we use the word bottle, we're referring to a Niskin bottle. So, Niskin bottles are bottles that have openings at the bottom and the top, and they can be triggered to close to trap seawater inside. So we can use them to sample at discrete depths in the water column. So we have a few Niskin bottles on the ROV, so we can take water from particular locations and bring it back up to the surface while we're on dives. Yeah, I'm trying to talk in a calming voice, especially because we're all in here working and we're doing very delicate work, and we don't want to disturb the pilots while they're trying to concentrate. This is very tricky work. So normally I'm a bit louder than this, but I'm putting on my calm voice. That way you can all relax and look at the beautiful, relaxing ocean view, and everything can be mellow. Nice big sea slug there. Woof. I've seen a lot of, not big sea slug, sea cucumber, I should say. It's not a sea slug, it's a sea cucumber.
Well, that's how we're gonna fund it looks like the next cruise with, people with the calming voices, make inside money. Right. We accept donations.
That's right. We'll start a Patreon.
And you want a Niskin bottle here? Niskin?
Oh, yes. Oh, no. You wanted a Niskin from the next site, right, Aparna?
Yes.
Okay. We'll do that at the next site
Right.
before we take any other samples and kick up a whole bunch of schmutz. Yeah, I think we should start a Patreon of just like calming oceanographic ASMR. Do you have trouble sleeping? We're going to go to the second seep that we found. I believe we put down a marker for it. It's pretty close by.
Seep 2 is right down below.
There it is, yeah, seep 2.
So it's south. Yep.
Thank you very much. Yeah, all the sub controls are in real time, so it's like flying a plane. Only you're not in the plane, I guess. Like the world's most scientific video game. Oof, look at all that floof. It's made it very difficult to see. The visibility has been not great around here at this point. There it is. Right? Is that it?
Shoot, one of those,
Yep. Okay. So we need a bottle for Aparna. One of the I think bottle 6, the other small one.
Yeah.
Okay.
There's no one 6s.
Great.
So are you gonna want to collect these clams or?
No more clams. We're just getting push cores.
Okay, so Lauren, just to recap.
Yes.
Do you want to up in the water column or on the seabed for the?
Aparna, do you want to be on the seabed when you take the water?
What is a seabed?
You mean like, do you want to be sitting on the seafloor when we take the bottle, or up in the water column?
No, it's fine up in the water column. It's fine for me. Yes.
Okay, whichever you prefer
Yes,
is fine.
I just want to prepare media using the water.
Okay. Either one is fine. So if it's easier to land first and then take it, your call.
So Lauren, to recap.
Yes.
Here we need juice core,
Yes.
Nick's core.
Yes.
The geologists have asked for another core, if possible.
Okay.
They said that it could be in a place without seeps, but it was them assuming that we were not going to go to another seep, so they have asked me to correct that.
Okay.
That it would be here.
Okay.
And that would be all, unless you want another core here, which
I'm not going to take another one here. I'm going to save some space just in case we find anything else.
okay.
And if we absolutely don't, we can always come back to the spot and I can take another one.
Okay, so the thing is, Katie's going to need someone to compare it with. So if we end up coming back, it's not an issue. I have already solved that. If we end up coming back, then we'll do it with that. And if not, I will just take some sample from Nick and try to help her with. So
Okay.
not an issue.
6 to 5 is long.
I haven't been able to leave this room, so. Okay. Did that fire? Okay, cool. All right, Aparna, we have your water. All right, Nick, how many cores did you need from this site? 2? Okay. Mm-hmm.
Number 6 is ours.
I don't know. Did you find it already? Okay, then I will.
Okay.
Can you take a core on a biofilm?
Yes.
I don't care what my core is taking, so I can help here.
Yeah.
Not a core normal, whatever.
Okay.
So this is the purposes.
Understood. So, does yours need to be on the seep or away from it?
It could be half a meter away from it, 1
Okay.
meter away. Yeah.
Okay, cool. All right. So, we need a core that captures some of the biofilm around this. And Geology asked for another core. This is not a very big site, so all right. Well, let's start with the core that captures the biofilm before we start stirring up a whole bunch of stuff. Because this one's smaller than the other one.
posición de cuerpo o, por ejemplo, si hay un raya, si hay
I also see some biofilm over here, Aparna, if you want to try to get some of that. Some of that biofilm.
I think I don't have course mode because I have already taken two. So if I can share with Lee probably.
Well, yeah, I'm saying we could take one of Nick's cores over here.
Yes.
Okay, then that way, I just don't want to take too much from around here and damage the site. Well, let's take, because Nick still needs one of his, and he does want one from the seep. So let's take a core for Nick here. Yeah, I think we want to try to aim for that if we can.
So, whose score is this one going to be?
This one is for Nick.
Okay.
Does that work for you, Katie? That spot?
For Nick or for me?
Or not Nick. Sorry, Drew.
Okay.
Is there biofilm?
It looks like there is. It's like a darker color, though, than the white fluffy stuff. Or we could take it back here where there seems to be more of that white feathery material.
He's living or are they dead?
They're alive, these clams. They're alive. I mean, they're not just shells, so I assume that they're alive.
Are you guys pushing a core here too or no?
We can if you need us to.
I would prefer to because we don't sequence in our lab.
Okay.
I will also appreciate it.
That's fine. We can do that.
There will be at least 4 spare cores after this.
Okay.
After all of the courses.
Yeah, that should be fine. Right? Just want to make sure everybody's getting what they need. All right. So, Katie, you want to take from the area where we have clams?
Yeah, it claims biofilm.
Okay. All right.
Yes.
So I think that this dark material on top might be biofilm. It's not the white stuff. But I think that was pretty difficult to capture. Anyway, I don't know how many people actually got any of that material. I think Aparna might have gotten one.
It was not that common around
Yeah.
the methane seeps.
Yeah. So I think we can try for that area that you indicated earlier, and that will be Drew and Katie's. Or Katie, do you want to pick a spot or?
I will try not to be too ambitious and not get very
Is
close
it
to
gonna
the
be too
clams.
soft over here?
Yeah, I think if you get too close, it's probably it might not hold up. But I think up in that corner, you might be okay.
Okay, I just want it as close to the claims as possible just because of what we're looking for.
Yeah, of course. Yeah, we'll do our best. I mean, we don't know what's gonna happen with it until we try, so we'll give it a shot.
By the way, Ron, I think the
Mhm.
geology guys will be more interested in an extinct seep. I don't know if we
Oh.
will go through any of. We will go through some of those again.
If we come across another one as we're going, we could stop and take a core.
I will check with them if they rather have a possible
Okay.
extinct one or an extinct one 100%.
Yeah.
How about
Hello to our friend from Lithuania. Did I get it right?
I think right there?
Hello, Lithuania. Good morning.
Mm-hmm. Yes.
Right there?
Yeah, that works. Yeah. I say go for it. Or maybe actually trying to nudge over this here. I think that's a little bit more. Yeah. Because you can see all the material, like where the clams are, looks like extra soft. We had a couple cores already just like fail on us on the seabed, which hasn't happened to us yet so far. So You've been super lucky with cores up until this point.
Look.
One of the new questions that arose with the discovery of this methane seep is the presence of this genus so far up north. The previous records were down in Concepcion, and after some short discussions I had with our local invertebrate expert, Dr. Guillermo Guzman, now we're interested in comparing the genetic divergence between the population of this species from Concepcion versus those from here. Now, the sampling size is limited, but at least we're going to get a preliminary view of if the populations have diverged a little bit or not. So not only we have the projects people have proposed before the expedition, but imagine that every project will give rise to, I don't even know how many other projects will arise from this preliminary data that people will be getting.
Yeah, it's very exciting. Yeah, we made it. All right, let's take one more in this area for microbiology.
Yeah, and the one from Nick as well, I think. Or not?
Nick, where would you like?
General.
Okay.
Okay. So.
Okay.
So not for.
Oh,
Yes,
Nick
for,
wants to capture this film back here so that he can give that to Aparna. Okay, so let's take one more here.
One for microbiology, then one for Nick slightly further away, and then I'm waiting for confirmation on the geology people.
Okay. Does that work for everybody?
That works here for us, and then
Fabulous.
another corp anywhere.
Great.
Okay, so one more in the same where we are right now.
Yeah, one more here, and then we'll go over there and we'll take two more for Nick.
If we could maybe for the microbiology core not use number 9, which might be a bit cloudy,
Oh,
and then we can use that for Nick, because doesn't really care about bacterial
right. Okay.
contamination.
Yeah, that makes sense. Okay, so let's use 10.
10?
10?
Yeah, that should work.
Okay.
Yeah. Push core 10.
In the same.
Yeah, in the same area or nearby. Okay, Miguel,
Yeah.
where do you think we should take this one? Do you want to try
Let me
the...
go there.
Yeah, come on over here with the pointer. Help us out here. I will let you,
Stick of power.
yeah.
Get around here. Should be in stables. Maybe here makes sense.
Yeah, I think that would work.
Yeah, sure.
Let's go for it.
Not so close to the clouds? Okay.
Not too close about maybe.
Yeah, we don't.
About as far away as
Yeah.
maybe slightly more, but not much.
Yeah, we're trying not to disturb the clams, and also
Yeah, I'd rather
the
not.
sediment near them seems to be really unstable, so it's a twofold operation here. Yeah, we've definitely had some cores fail because of all this fluffiness, and actually collaborators warned us about that before we got here. But I think they were also sampling closer to the coastline, where apparently the sediment is even fluffier, which makes sense because in an upwelling area, the productivity is going to be concentrated near the coast, and as you go further out, it becomes less. So as we've gotten closer to the coast, the oxygen minimum zone, the OMZ, has gotten deeper and over a greater range, probably because the productivity is greater in these shallower areas. So we're not sampling close to the coast. We are only sampling outside of 12 nautical miles from the coast on this expedition. So we've been doing a lot of deeper dives, and so far we haven't had any issues taking cores until today. So these methane seeps are tricky little buggers. Oh, looks good. Everybody cross your fingers.
That's great.
Sweet. Nicely done. All right. So we can go over to that other area, and cores 9 and 11 can be for Nick. And then I think we are done sampling for a while. Oh, no, wait, geology.
I don't have any news from geology. If I don't have any news by the time we have both of Nick's samples, then I think we can just try to get to aquarium and inactive or less active seep in the next 4 hours. if that
Okay.
works.
I mean, we've certainly seen a lot of evidence of inactive seeps and clamshells and things, so we can probably stop and grab them something.
They just told me to get one around here as well, so we can finish it up.
Sure. Okay, so they want one from around here?
Yeah, maybe on the other side of the seep, making like a triangle.
Okay. All right, let's grab one from here then. That can be number 9 because they don't care about contamination, right?
Yeah.
If there's
that's
some
good.
fluff in there. Okay. So push core 9 on the other side of this little clam bed here.
So where?
Maybe like
On the
over
outside?
there? Yeah.
Like a triangle pattern?
Yes. Thank you. So some questions in the chat. Given the abundance of sediment here, which I assume is primarily organic in nature, why do we still see limited life here? So there's a ton of sediment here. A lot of it is marine snow and other organic matter that is sank from the productive top layer of the ocean. However, by the time it gets down here, it's been chewed up pretty good by planktonic bacteria that can colonize these particles as they fall and chew up all the good bits. So what you end up with is what's called recalcitrant organic carbon, which is it's still organic carbon, but it's difficult to break down, or, as one person just said, not particularly good for deriving energy from. So certainly there's probably plenty of microbial life in this detritus that's capable of breaking it down and using it for energy. But microbes don't need as much energy as multicellular life, so they can get by on a lot less. So there's lots of life down here. It's just not all life that we're capable of seeing. And we've also noticed that at least around this part of the ocean, as we've been exploring, that most of the life seems to be concentrated around the rocks. We've seen, for example, mollusks, brachiopods, brittle stars, echinoderms like crinoids, all attached to rocky substrate. So they like to have something to anchor onto. And in this fluffier sediment, what we've seen primarily are lots and lots of sea cucumbers and the occasional fish. And some acorn worms. We've seen lots of those. Brittle stars, yeah.
Alright, so it looks like there's more dark closer to the planets. Do you want to worry about that or just getting the general?
I don't think the geologists care about that.
Okay, so I'll go a little bit in the middle of this too.
Maybe we
Maybe.
can try for it to be about as far away as the last one.
Yeah, like,
Yeah, I'll go right about here.
yeah, that works.
So they can get a little bit of black thing just in case they're
Yeah.
interested.
Alright.
We've also seen lots of really cool polychaete worms down here, like all kinds of. There's an acorn worm down there, down at the bottom of the screen right now. So yeah, there's actually quite a lot of life down here. In the bottom of the ocean, it's
And also let me defend the invisible life that is living in the interstitial spaces of the sediments. This is actually so the I understand what the viewer is saying. However, this place is teeming of life just because it's a little less than 1.5 millimeters. You cannot see it without the aid of a microscope. It doesn't mean that this sediment is literally crawling live. We just don't see it.
exactly, yep. Nick is a defender of the tiny.
That's right.
Yeah, you gotta stick up for the tiny.
Absolutely. Speaking of the tiny, for example, if one of the things that we do is as scientists, of course, we have a big component is the field work, another huge component is the lab, another component is the analysis and the writing up of a manuscript. But then, one of our obligations is not only to disseminate the results and enthusiasm as we do here with the public, but also we are almost expected to attend conferences where we present our results in front of our colleagues. And I'm saying that because I was just looking at where am I going to present possible results from this expedition. And for the stuff that I'm doing here, a very suitable conference is the International Myofauna Conference, which I have been following on and off since I was a graduate student. And in 2025, it's going to be in Calcutta, India. Yes.
Oh.
So I'm going.
Oh wow, Aparna just got very excited.
Myofauna is in Calcutta 2025. Yes. I was just checking because the last one was in New Zealand. Unfortunately, they made it online because of COVID. And I was going to go to New Zealand, but as soon as I heard it's online, thank you very much.
Wow, you myofauna guys get around. New Zealand, India. That's,
Yes, well,
wow. That's
we have to
incredible.
make up for work with little things.
Yeah, you gotta entice them. That's fantastic. I like that.
So, yeah.
How exciting.
And what month down the
I guess summertime usually. Summertime, and usually the month and the date gets pinned as
All
we
right.
get closer.
Okay. Did we get everything we need from this spot?
Yes, we need to go get Nick's this what are on the mats. Right?
Okay. Yeah. Can we
Yeah,
go
we're down with this place.
over there then? Thank you.
As soon as I get more information about the game, I'll let you know. Yes.
Correct. So this is open, so I don't know if they just failed on that one. Do you want me to try again on that one sometime, or do you care about it? You just work on me.
Oh, All right. Yeah, I gotta get real bent out of shape about speciesism myself because I'm a microbiologist, and so when people are like, "Oh, there's nothing alive here," it's like, oh, there's probably lots of things that are alive, but you just don't see them. Or a question that I like to ask my students is, does all life need oxygen? And usually they say yes, and I say no. Actually, there's a lot of living things on this planet that will die in the presence of oxygen. They're microscopic, though. You can't see them.
For a huge chunk of the history of our planet, oxygen is poison for everything.
Exactly. Yeah. First billion years of life on this planet, there was no oxygen in the atmosphere. Everything was anaerobe. All right, so we're going to try to get some of this white biofilm type material here.
Right here?
Yeah?
Right exactly there, yes.
Okay, so right here, can that be number 11?
Okay.
Does that work for you?
It definitely works for me, and I think I'm not going to talk from 4 apart. Is that okay with you too? Okay, yes.
Okay, 11?
So this one will be shared between Nick and a partner. It will
Yes.
be technically Nick's, but shared between the two, right?
Yes.
Okay.
Yeah, everything good.
There's some dead clams here, or clam shells here, so looks like this might have been an active part of a seep at some point, and there's still biofilm here, but no more clams. As Nick put it, it begins and ends with bacteria. I really liked that. I liked that expression.
I gotta give kudos to my two professors that introduced me to myofauna, Dr. Tom Shirley, emeritus professor at the University of Alaska Fairbanks and emeritus professor at Texas A&M. Retired now for a while, but still very active. And also Dr. Bruce Cool, emeritus professor at the University of South Carolina. I had an amazing time working with these two people. I only remember positive things, so even now we still talk and of course, and we still actually work together.
That's wonderful.
So the white right there in the middle.
Yeah. Maybe like right here.
There.
Yeah.
I also want to include as well the two other people that had a big influence on me academically, my postdoc advisors, even though it was on a very different field in theoretical population genetics. I worked with Dr. Bill Banner at the Field Museum, Natural History in Chicago.
Oh, that's a great place.
He's right now a senior professor at the University of Queensland. And also I worked with Dr. Richard Hudson at the University of Chicago for 2 years in theoretical population genetics. He has retired now for several years. Fantastic people.
This one I'm a little worried about.
Nice. Uh-oh. Linda's back. All right. Okay, and Nick, you needed one more, right?
Yes, please.
Okay, one more core here, and then we can get back on the track to the next waypoint.
Where would you like the next one?
Nick, do you have a preference?
No, it could be very close to those dead clams, for example.
On the left?
Yeah, to the left, yes, sir.
Okay.
All right, this is core number 12?
Yeah, we'll do
12. Once bacteria fall down with marine snow, digesting it, how do they get back up? Or are they just stuck there at the bottom? They are stuck at the bottom. I mean, they could be brought up by an upwelling event or a move with currents. Bacteria, well, some bacteria are planktonic, meaning they just kind of float, and some bacteria can swim. But bacteria swimming are never going to be able to reach the top of the ocean before they would die. They're just too tiny. So most of the bacteria in the water column, I believe, are planktonic. They just kind of drift. The swimming bacteria actually like to be attached to a surface, and they swim to find surfaces to attach to. So once bacteria sink to the bottom, they may end up in the sediment. They can be carried by water currents so that they don't ever actually fall down all the way to the bottom, or they can be buried in the sediment. Yeah, these cores are going really well. We've had very few setbacks with the cores.
Anywhere over here?
Yeah, I think,
Oh, right.
yeah. All right.
Yeah, so this undisturbed is fine.
Yeah, any old place you like. How common are upwelling events? Upwelling events occur, they're determined by currents, so whenever water is transported away from the surface, and it can then be replaced by water from the bottom. The water doesn't really get murky during those events exactly. Well, what happens is that the water on the bottom of the ocean tends to be richer in nutrients. And so when it's transported to the surface, it causes an increase in productivity, which, yes, would make the water more murky because it's so full of phytoplankton and bacteria that are eating the phytoplankton. For example, water that's very nutrient rich is going to appear more murky, more green. It's going to be harder to see to the bottom, whereas water that's very nutrient poor tends to be more clear because it has less phytoplankton growing in it. So, if you've ever been a place where you're like, oh, the water's so clear here, it's probably not very nutrient rich. Otherwise, it would be loaded with phytoplankton, which would make the water more murky and greenish. And eventually, all of that productivity gets eaten up by other things like fish, bacteria, and can end up drawing a lot of oxygen out of the water in the process, which creates these oxygen minimum zones. Upwelling events can also cause what's called a harmful algal bloom, like a red tide, or the oxygen can become so low in areas that it becomes too low and it can kill fish. So upwelling events are good to a point, but they can also cause things that are damaging to an ecosystem, at least in the short term. Things always seem to balance out over the long term. Okay, I think that's everything, right? Are we done? You got everything?
Yes.
Sweet. Okay. So can we start heading towards our next point? Let's see. Yes. Yes.
Come again, Chris?
The samples don't have a strong smell when they come up on board. However, you forget them for 12 hours at room temperature. That's, yes, you will want to deal with them very fast.
Yeah, Aparna and I were just talking about this. We do not know yet what are the microbial communities living in these things, but it's true that they're quite black, usually it can be due to there's that element of sulfur and the sort of sulfur-producing bacteria, which may be associated in communities with anaerobic methanotrophs, which are likely to be the case. I have no idea, but I have not studied these things before. If that's the case and we smell sulfur when processing the sediments, I think we may need to process them quite quickly.
Yes. There are sulfate-reducing and sulfur-reducing bacteria in these mats that are associated with the methanotrophs, and they will produce sulfides, which have a characteristic rotten egg smell. So if you've ever smelled something that has that like stinky rotten egg kind of smell, like I grew up at the Jersey Shore and spent a lot of time going around on the bay and boats and things, and the salt marsh smells like rotten eggs. It smells like rotten eggs because salt marsh sediment is full of sulfate-reducing bacteria that produce stinky sulfides. And in my lab now, I'm culturing a sulfate-reducing organism, and every time we clean out the test tubes or take out a sample, the room starts smelling like rotten eggs. So there's actually a myth, a common misunderstanding. People think that methane stinks because methane is a lot of times referred to as natural gas. So if you have natural gas in your home, it actually has a compound added to it that smells bad. So if there's a gas leak in your house, you'll be able to smell it. Methane actually naturally does not have any odor. It is odorless. So if you smell something stinky and rotten egg-ish, it's not methane, it's sulfides.
All right, Rich. Good morning. Fantastic. Thank you very much.
Good morning, Alan. You can go nice and slow for that.
Yeah, unlike bacteria, we can't tolerate sulfides very well. Yes, methyl mercaptan has that stinky rotten egg smell. If you've ever used it in the lab, which I have, it is potent stuff. It smells real bad, and they add it to natural gas so that if there's a gas leak in your house, you'll be able to smell it, because otherwise it would be odorless. So far, most of the cores that we've taken have not had any particular smell. They've smelled like seawater, despite all of this detritus on the bottom. But if we find sulfate reducers at these seeps, which I suspect we will, those will probably be pretty stinky. So, we're going to be here for, I think, the next 7 hours or so. We'll see if we start getting the ocean madness by the end of all this. I've been up for like 19 hours now, so woo!
So I think I will go to sleep and I'll leave just with you.
Yeah, you've been logging for like 9 hours, haven't
Yeah,
you?
you can go faster.
Yeah, 7.
7. Oh, okay.
It's not even a full.
Well, I appreciate you, but go to bed. No naps. No sleep till seeps.
Hey Lauren, I forgot to ask you if you wanted a coffee. I'm so sorry. I just got one.
I don't know. What is that? Can we zoom in on that real quick? What is that rock? This over here? Can we take a moment to get some interns? Yeah, you know, usually you do this sort of thing in shifts, but that is definitely anthropogenic in nature. What in the heck? Jess is data logging for me once again. We're the dynamic duo, partners in crime. And we just found some trash. It looks like it was like metal and it's like rusted or something.
Yeah, it looks like iron.
Yeah, it's like flaking apart. It's unfortunate, but even down here at 2,800 meters, we're finding trash, y'all. All right, you keep going. I just needed to log that.
I just updated the bridge. Told them we're going to extend the dive time for a little bit.
Yep.
Probably depending on what else we find. Is it?
Yeah. Well, we have several Niskin bottles and 5 push cores left. And I think room in some bio boxes. I don't know what the situation is with the bio boxes.
So to start from Chattanooga, Tennessee.
Ooh. I just love rocky outcrops. They're always so interesting looking. Maybe it's because I grew up in an area with like no topography, no rock at all. I grew up in South Jersey at the shore, and everything is very flat, and the Pine Barrens are very flat, and seeing rocks and mountains just makes my heart sing. But I also really love the Pine Barrens. I can't lie. I still live and work in the Pine Barrens, and it's a beautiful place. It's truly a unique ecosystem, and it has this incredible smell that's like this mixture of pine and oak and iron and wood smoke and salt, and there's no other place like it in the world, and I absolutely love it. It's the home of my heart, and that's also where my husband and my 2 babies are right now. I have a 5-year-old and a 7-month-old at home.
Oh, I didn't see the pictures today.
Oh, you know, I gotta show you. I got some pictures today. Josephina's my 7-month-old, and she's making this face that, actually she's 8 months old today, I think now. Good grief. She was making this incredible face where she was just like kind of sucking in one lip and sticking the other one out, and it was adorable. Everything she does is adorable. Ooh, that's a pretty.
Tube anemone.
Super pretty.
Tube anemone.
A tube anemone. Anemone. It's 1 a.m.
Nick, what are we gonna do without you? We don't know.
Oh man, I don't
We
know.
don't know all the species.
What are we waiting?
You know, even if we hadn't found any seeps, this is just a really pretty part of the ocean. I'm glad we found seeps. I'm super glad, but this is also just really pretty to look at. Can't lie. So we're just flying here over the bottom of the ocean.
Is that a,
That's a starfish, yes.
Yeah, a really nice sea star down there. So much purple stuff. I didn't expect to see so much purple on the bottom of the ocean, and I'm here for it. It's my favorite color. It's been very exciting for me.
The little biology now, I still have a little energy. So why do we see corals? Why do we see soft corals, sea anemones so deep? How is this possible? The shallow water counterparts heavily rely on photosynthesis, not because they are able to photosynthesize themselves, but because they rely on their symbiotic symbiotineacia, which are photosynthetic algae. So up to 50 to 60% of the metabolism of the corals or the sea anemones or the soft corals are coming from the symbiotic algae, also known as zooxanthellae, depending on your background, or dinoflagellates. Okay? And they are usually found around the gastrodermis of the cnidarians. So 60% of the shallow water, many of the shallow water cnidarians is coming, 6% of the metabolism of the energy is coming from these symbiotic algae. So what's going on with this, with deep water counterparts? I mean, obviously there is no sun here. Anybody from the audience can guess how do they get their energy? We have, I know we have many educated or well-versed audience, members of the audience.
Yeah, you guys are impressive.
How do the deep sea cnidaria get their energy from?
Well, other question, who is still awake?
Let's start with that.
Anyone? Bueller? It's a great question.
So I'll give it just a couple seconds.
Yeah,
and
give,
then-
give,
yeah, all
give
right.
people a few
Let's-
moments to think about it. Look at those anemones.
Those big single polyps are sea anemones.
Oh wait, is that down there? Is that a sponge?
Yeah, that's a hexactinellid sponge.
Oh.
Yes.
Okay.
Organic.
Are they the
like-
ones that have like shrimp associated with them?
Many of them have associated shrimp, and sometimes the association is species specific.
Oh, wow.
Yes, very interesting. And
Is-
also we have the famous case of the very long hexactinellid vase sponge. With what happens with it is it's like a net, and then a young couple of shrimps, a young couple of shrimps of the genus Spongicola gets in the growing hexactinellid. They can go in and out for a while, but the shrimps grow fast, and they reach a point where they cannot escape anymore from the hexactinellid. So they are trapped there for life,
Wow.
a pair. So this is a symbolic gift in some cultures of
Just zoom
wearing.
in on it for a second.
You can take it, different ways. Anyway, I'm not gonna, either you're in love for life, you're trapped for it. I'm not gonna make. But it's a very symbolic gift for being together forever.
Aww. I just wanted to zoom in on this. What is that, Nick?
That's a sea anemone.
Oh, it's big.
Yeah. There's no
Oh, yeah. Yep. Sea anemone.
Yeah, as soon as you see the mouth, that's the mouth that gives rise to the siphonoglyph. Yeah, that's.
Yeah. Beautiful.
So.
We have some answers in the chat
Some
to your
answers.
question. Yeah.
There may be a little. I'm not so sure about chemical synthesis, but some things, some other things may be contributing a little bit to the metabolism. But corals are Cnidaria are predators, so they use the tentacles to catch zooplankton. That's how they can get their energy. You can keep shallow water corals in darkness, and you can feed them zooplankton, and they will be fine.
Wow.
So they use these long tentacles that you see here to catch epibenthic zooplankton, or maybe they can process detritus. So that's how they get their sources. Okay, so they are. Let me throw you some scientific words here. So they're heterotrophs. Okay, so they can, especially. So okay, let me get back to the autotrophs and the heterotrophs. So when a shallow water coral gets the majority of its energy from photosynthesis, you can say it's an autotroph.
Right.
But in the night, when they get energy through eating zooplankton, then it's becoming a heterotroph. Okay, because they can do both.
Wow.
Just to that's right. So that's how they can get the energy below a depth where there's no more light.
That's really fascinating. So they're mixotrophs then, kind of.
They're the shallow water ones, you can call them mixotrophs.
Yeah.
The deep ones are predators.
Yeah, right.
Yeah.
And I think I remember learning in grad school that corals have zooxanthellae that are symbiotic with them.
That is correct. Yes. I was talking about the Symbiodiniacea. That's the new name. And actually, there are photosynthetic dinoflagellates that you can find them very close to the gastrodermis of the Cnidaria, which are responsible for about 50-60% of the coral metabolism. That's right. Yeah. There are several names, as you said, zooxanthellae, or photosynthetic dinoflagellates, or Symbiodiniacea. Yeah, it's a very active area of research right now to figure out when the dinoflagellates leave the coral in case of warmer temperatures, with the so-called coral bleaching, who is in control of the system? Do the corals expel the photosynthetic dinoflagellates, or the dinoflagellates live on their own?
Right.
So very hot area of research right now. But anyway, that's the answer for how do the deep sea cnidarians, how do they get their energy?
By
eating zooplankton.
Yeah. Very cool. I think, from a basic standpoint in biology, we tend to think of there's stuff like plants that makes its own food, and then there's stuff that has to eat. But there are a lot of things that kind of exist in between or can do either one.
Exactly.
Depending on their needs. So biology is it's really fascinating that way. We like to categorize things, but sometimes there's a lot of gray areas, and it gets messy in the best possible way. Lots of cool rock outcrops here. It's very, very pretty to look at. And these huge purple sea anemones are sending me. They are gorgeous. Absolutely stunning. I want to wear one in my hair. There's a little sea fan down there.
see some comments about octocorals in the chat there. One way to tell an octocoral from let's say regular coral, hexacoral apart is the number of tentacles. Octocorals, you can tell by the name, it has 8 tentacles, versus the polyps of the corals, you say the hard corals have 6 tentacles.
Oh,
Or multiples, yeah.
I did not know that. Well, we are going to be here for the next 6.5 hours, potentially. Could be less. If we find another methane seep and we fill up the ROV with samples, then we may come off of the bottom sooner. Originally, we planned for this dive to go until 4, but we needed to take some time. We found a seep. We found 2 little ones, and we needed to give everybody time to take their samples before we could continue and look for more. So we decided, since this area looks so promising, to extend the dive so that we can look for some more methane seeps. And so that's what we're up to right now. We are off the coast of the Atacama Desert in Chile. We are at 2,806 meters. And we are searching for methane seeps along this ridge. We're looking here in particular because over the last couple of days we've been extensively mapping the area every night. So we've been measuring the bathymetry, we've been using backscatter and subsurface profiling in order to try to find indicators of methane seeps along the bottom. Of course, the best indicator of a methane seep is the presence of bubbles, and backscatter can sometimes detect those, but they're much easier to find if you're looking at a shallow seep, or if the seep is really large and just producing a ton of bubbles. And what we've found so far is that seeps located off of the coast of Chile tend to not bubble. So that makes them a little bit harder to detect from the surface. So the best thing that you can really do is find some areas that look likely to hold seeps based on the topography, the bathymetry, and backscatter data, and then just get down to the bottom and start looking. And that's what we did.
And increase your bottom time. This is why we have, I think, we extended the dive to 24 hours now, or 25, I think.
Yeah.
We originally planned to stay down for 21, but we would like to search the area some more.
Yeah, we started around 7 a.m. this morning, and we didn't find anything until around 6 p.m. So 11 hours of just looking around. What is that up ahead?
The purple one, that's a sea cucumber.
No, this clear thing.
I don't know, let me see.
Can we zoom in on this?
Yeah, we're going to right now.
It's a cephalopod. Well, no.
No, it's a fish.
My eyes.
It's looking right at us.
Oh man, we're getting the stare down. Hey, buddy.
It's a rat tail.
Oh. Whew.
Beautiful.
Yeah. Looks like the Godfather.
Yeah, that family has 200 species.
Wow, pretty spots.
That looked like he had some.
Spots, huh?
Yeah.
Jules fish.
Oh, your little boy loves the deep ocean. My little girl loves the deep ocean thanks to the octonauts. When I started showing her videos from past live streams from SOI, and I told her how deep we were going to be sending the ROV, she said, that's really deep. That's the dark ocean. I was so proud. My heart was swelling in my chest. I was getting all misty. I was like, yeah. I was so proud.
Yeah, we're currently at 2,800 meters depth, but I think on the last day we'll have a very deep dive.
Yes, the plan is to go.
The current plan.
The current plan, yeah, unless that changes, is to go as deep as ROV SuBastian can go, which is about 4,500 meters. So the deepest we've been so far, I think, was yesterday, which was 3,800, roughly.
That sounds about right.
I think that was our deepest so far. That was more of a biological specimen collection dive, though we were not looking for seeps on that dive.
What is really interesting today, we found the so far deepest found methane seep in Chile.
Yes, that's what
And
our
the
collaborator told
northernmost.
us. Yes, and the northernmost. Yeah. So most of the methane seeps, all of the methane seeps so far in Chile have been found further south, particularly around the triple margin of the Nazca plate, the South American plate, and the Antarctic plate. But we've found them further north and very deep. So
We're really excited. We're gonna go up and down this little slope in the hope of finding more methane seeps. We're coming up. So this is a fault zone. We're coming up to where 3 fault lines meet. So let's see what we'll find there. We'll just be zigzagging the area to look for any visual indications. Perhaps the sonar will tell us something. If there's any bubbling, we will see that.
yes, we are running the sonar, which will detect bubbles coming from below the surface if there are any. This is very weird looking. We've seen a couple of these.
Ascidians.
Ascidian?
Yes.
Oh, my little boy tells his toys about the different ocean zones. I love that. I love that so much.
We collected one the first day. I haven't seen it. It goes to the museum.
Yes, we are collecting specimens for a museum in Chile so that they have specimens of organisms from their own waters. And so we're working in partnership with them and collecting as many specimens as we can so that they can show them to the public in Chile and the Chilean people can see organisms that live in their own waters.
Oh, by the way, I missed the discussion on the new species that you guys found yesterday, right?
The snail?
The snail? I had to walk out of the room. I don't know what the...
Guillermo knows the family well
Yeah.
of the snails, and he, as soon as he saw it, he said this has to be a new species. So he seems to be pretty certain about it. Yeah, I don't know the characters that he's using, but I mean he's very knowledgeable. Well, we do expect to find many new species, really, or many new distributions. Especially, of course, the smaller the organism is, the bigger the chance that you're going to have a new species, and also that we know that this area has not been sampled, or even if it has been sampled a little bit, the chance to find new species is great.
So far, we've had some pretty successful dives in that respect.
If we had the taxonomic power and the taxonomic expertise, when I mean power, I mean availability of people with a high level expertise, and if we could work all the samples, I'm sure there would be dozens of new species.
Wow.
The bottleneck is the expertise and time.
Yeah. Well, that's true of pretty much all of science, I suppose. Yeah. Expertise and time.
Oh yeah, I'm really curious to see how long the extraction and the analysis of all the samples is going to take.
Yeah, we've taken a lot of cores. We've filtered a lot of water. We were just looking at a very large white sea anemone. Beautiful. I love the anemones. They look like big flowers. I guess that's why they're called that, because an anemone is also a flower. Roomba-looking anemone. Yeah, it did kind of look like a Roomba. Pom-pom Roomba. Seeing a lot of these squat lobsters. I'm learning so many species. And as a microbiologist, I definitely do not know enough about marine animals. But I've learned so much in the last few days just from my colleagues. It's been a great experience.
Oh, is that a sponge?
Absolutely.
Yeah, that's
Exactly.
a sponge there. Yeah. Is that pink guy? Is that crab with the anemone on its back, or is that a new one?
Yeah. Oh, that's a good eye. We can zoom in a little bit there if you want.
Can we zoom in on the little pink fella?
With a very small. Oh no, to the yes, like that. Guillermo said that this is possibly a new species, and I don't know. Did you have the chance to collect one for him?
I don't think we've been able to grab one yet.
Oh, didn't we? I thought that was the first day.
I
From what I remember.
I thought so too. I don't
Did we
remember
try and then, geez, I don't know. Maybe we should grab one just in case. Is the sucker still clogged with that squat lobster? I don't know how we're gonna be able to pick this guy up. The pinchers are, he's quick.
There's no way I'll grab one.
Oh, I think I don't have an updated list on what is in the suction sampler. Is there?
It's on our side. We're still on one.
I don't think we've
Oh,
been able to
still-
add anything to it because it's clogged with a squat lobster and we
Ooh.
haven't really used it since then, I
Okay.
think.
All right. Oh.
Yeah, that guy's really on the move. I feel like if we had the suction sampler, we could catch him, but I don't know about trying to grab him with the.
Sure. Let's.
Would it go through?
I don't think so. That's why
Either.
I think he would fit if it wasn't clogged.
I don't know. I think they tend to like stick out their legs.
Oh, and try not to
Yeah.
get pulled in.
Mhm.
Yeah, I mean that's what I would do.
Yeah.
So.
It's always the same species of anemone that seems to be when it's.
It's
So
wild.
should be the
It's
case.
bad.
Yes.
You guys want to give it a shot or not?
We already spent time on him. Let's give it a shot. Huh? No?
Okay.
Yeah, if you want to, Nick, I'm
The,
I'm willing to
just,
try.
Let's try one time.
okay.
All right.
It'd be nice to grab one for Guillermo.
Okay, I do have. I'm sorry. I do have the note on the
On the lobster?
squatty.
Yeah.
Oh, there's Guillermo. Hello. Good to see you guys.
Alexis.
More biological expertise in the room.
Always good to have.
Guillermo, te ves estar feliz ahora, ja, 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 porque estamos para un chemical con asiático. ¿Qué es? ¿Tú sabes esta persona? Fabián. Alumni, ¿sabes? ¿Qué vas a decir ahora? Ahora, no, ahora estamos para el chemical con asiático. El problema es que el otro cangarrito
I think we are going to try to grab this guy. It's always good to have more than one specimen for identification. But we won't spend too much time on it, so. We'll be here all night, folks.
Literally. Yeah, I just updated the daily schedule, so the current schedule is 8 a.m. off bottom.
Mm-hmm.
So that means sample recovery or ROV recovery with samples at 10:30.
Mm-hmm.
And that reduces the bottom time of the next dive by 4 hours.
Right, but we have a shorter distance to cover,
Yes.
so.
The association of crabs with sea anemones is well studied, and it's an amazing association. How many people have heard of the pom-pom crabs?
Oh yeah,
Yes,
the
we love
pom-pom
them.
crabs.
This is kind of one of the celebratory cases of this association, where they can actually take them, they
Yeah.
can move them on their carapace, they can use them as defense mechanisms. So it's
Yeah, they really punch with them. I've seen a couple.
Yeah, it's funny.
it's amazing.
Really quite incredible. Who wouldn't want a cheerleader crab?
There's also a dancing shrimp.
Oh yeah. Oh, are they the ones? They're on TikTok. They like have the shrimps dancing and they play like rave music. They're like
Yeah.
doing this.
They wiggle around.
Yeah, love it. There's a dancing spider too. That like raises its
Oh.
back legs and does this.
Beyond this,
Yeah, what are they called?
I forget.
Words. Peacock.
Yeah, peacock spiders. Yeah.
Peacock jumping spiders, I think they're called.
Biology is amazing.
There are so many different species with different dances. Really, really
Yeah.
interesting.
Definitely getting stuff through it. You can see it going through. So.
Lord of the dance. Gotta hoover that crab. Oh my god. Science is very serious. Except when it's kind of silly. That's why it's fun. Behavior and color are so much easier for taxonomy than microscale stuff. Ain't that the truth? Wow. Yeah, we got him. All right. All right.
Which sample box is that?
Which collection jar did that go into? 2 if he makes it. Okay. Yeah, as a microbiologist, y'all who study macroorganisms, you're spoiled.
Keep going.
Do you know what the sample ID of that one would be?
I don't.
Oh, okay. Maybe we'll start this with 1 0, and then 1 1 1 2.
Sure. Yeah, every so often we decide to totally rearranged taxonomy because we've figured out that the previous system was not the best. And that happens in microbiology pretty
the time.
constantly. Yeah, all the time.
Because then there's genetic evidence, then there's protein evidence.
Yep.
Depending on what you look at,
Mm-hmm.
there's different results.
Then we find new clades and,
And rearrange everything else around it.
Yep.
Yeah. Constantly in flux.
Yeah.
One sign of maturity in the field is when the phylogenetic tree or the relationship between species is becoming stable. So, for example, two of the best examples we have is the mammalian tree, or all the relationship between mammals and birds also. So that's obviously they have been well studied by many labs, and they're huge resources compared to other fields, of course, to work with mammals and birds. Not that every question has been answered, I'm not saying that, but the overall relationships of the species has been rather well worked out.
So you said that there's a squatty in the hose as well?
Is it still stuck there?
I didn't come through with that thing.
Oh.
Wait, so they're both
They're
in there?
both stuck in there?
Ambos cangrejos están en el tubo.
Well,
Ambos, no en caja, en el tubo.
well, that'll be interesting when it comes back up, I guess. Yeah, in microbiology, we also have horizontal gene transfer. So microbes can just trade their genes around.
Oh, and sometimes that's done by viruses too.
Yep.
So giant viruses will take up some of the genes and infect another microbe and then transfer the genes, which are not always harmful.
Mm-hmm.
But that's how everything gets a little mixed up.
Yeah. There are pathogens that derive their ability to cause disease from genes that they get from a virus, which is just mind-bendy. This is why I don't study pathogens. That and they just kind of depress me, and I'm too much of a germaphobe to work with anything above a BSL-1. I'd rather study things that do nice things.
Methane munching microbes.
Yeah, methane munching is nice.
My last audiobook. So right, I mean, it's okay if I name a book, huh? Right? Or is it against?
No, I think that's okay.
All right. Yeah, I don't know. So my last audiobook was the Tangled Tree by David Quammen. So this is very relevant to all the microbial people, I think. And for me, it was fascinating because one of my hats is evolutionary biology. So in this book, he's going over the history of using 16S to describe the microbial diversity, and he's talking about some very big names in the field responsible for establishing the 16S gene as the unit of measurement of relationships between bacteria. And he's traveling us from the very first idea of finding a universal gene to do this type of analysis in the late 50s, early 60s, I believe, late 60s, something like that, up until just a few years back, because some of the big names in the field passed away. So huge book, and of course, many chapters are dedicated to horizontal gene transfer, which we just mentioned. We just the passing, because genes are not just passed vertically from generation to generation, but also they're passed vertically through the means of viruses, plasmids, autophagy of genetic floating genetic materials. So there are actually many different ways. But I truly enjoy this book, and I recommend it to all students that want to get into that field, because they get a historical perspective on the people that established the field. When I mean the field, I mean using genetic tools to describe the diversity.
Yeah. Until we understood DNA, we had to describe the interrelatedness of things by observation and just sort of put things together that looked similarly or behaved similarly. And now we have the benefit of DNA, and it's really just changed the game of biology completely. Look at that.
Dr. Guzman is telling us that this is a blind lobster.
A blind lobster.
We see no eye spots.
Wow, yeah.
It doesn't mean that it may not be able to detect light, but no obvious.
That's beautiful.
Oh, so then.
Polychelidae. It belongs to the family Polychelidae.
Ah, wow.
So they might have light receptors but no eyes?
That is possible, yeah.
Okay.
Yeah.
Looks like a ghost.
Even though I see no reaction to the strong light.
Well, down here it's pretty dark.
And cold.
And cold.
Less than 2 degrees Celsius.
Yeah.
Oh, by Nick, we had a question earlier that I think maybe you can answer. The crinoids that we see, can they change the orientation of their arms?
Their tentacle. Yeah. Yes, they can, because they can retrieve. They have the muscles to retrieve the tentacles. Absolutely. Now remember, there are two ways to do this, especially the crinoids that have cirri that are not attached to the substrate, that just anchor. They can do whatever they want.
Yeah, like feather stars,
yes,
right?
Yes, like those. They can do. They can swim away. They can switch around. Yeah.
But the stocky type ones, they can still turn around their heads.
They have control of
not
them.
heads, but their arms.
Absolutely, because that's how they. Well, of course, the audience don't see me, but they can retrieve the tentacles to the mouth, so they can get all the detritus that they catch.
Interesting. Do they orient themselves into the current so that they can?
They have to
pick up
be. They
particles?
have to be able to orient themselves against the current. Yes, they have to.
Because we noticed earlier that all the crinoids were facing the same direction, like sunflowers turning to the sun.
Yeah.
It's really cool.
That makes absolute sense.
Nick, do you mind taking over for just a few minutes for me?
Sure.
Thank you.
Are you getting a coffee?
No, I have to take a biological break. I'll be back in a bit.
Lauren has been on a long shift since 7 a.m. this morning. That was when we started our dive. Oh, that's when we were on bottom. So this is going to be a 25-hour dive. We recently found methane seeps, and now we're scanning the area for larger seepages and hoping to find something. So we're mostly looking for visual indications, because from seeps further down south, we know that there aren't a lot of bubbles usually, but we also keep an eye on the sonar. So if there were any bubbles, we would see that.
Hello everybody, this is Nick Skizas. I'm giving a break to the PI Lauren. I'm one of the invertebrate biologists in this expedition. I'm a professor at the University of Puerto Rico, Mayagüez, and I have been studying invertebrates since 1991 as a beginning graduate student. My lab is specializing in questions regarding biodiversity using genomic tools, and most of the work has been done with crustaceans and corals. Since the labs are based very close to coral reefs, we have immediate access to very good coral reefs. Seagrasses, mangroves were based in southwest Puerto Rico, where you have access to the field any day of the year with water temperatures varying from 25 to 30 during the hot months. So it's lycra only diving, unless it's a 3-tank dive day where at the end you're going to be so tired you're going to be a little cold. So you may need like a 2mm suit.
Are you doing saturation dives, or is it usually shallow?
No, university rules are pretty tight with insurance is very tough. So it's However, we had a very nice. We had a NOAA grant, NOAA crest, deep crest, which was dedicated to the study of mesophoric reefs. These are reefs found between 30 meters and 100 meters in the Caribbean. And we had a dedicated 6-people crew, including students and professors, that they were doing trimix rebreather diving. So people in the audience that know about this type of diving, it's a huge dedication. It takes a lot of time, a lot of training, and it's definitely much more complicated than throwing a tank on your back and getting into the water. So we have been studying corals until about 90 meters in the Caribbean, where photosynthesis is still happening. So yeah, I've done actually training in rebreather diving, but only with the diluent was air, which is several steps, 2-3 steps before you're qualified to use trimix, which includes helium. It's definitely complicated, and you really have to. There has to be a reason that you would want to do this type of diving. There has to be a good reason to go beyond the regular nitrox diving, for example. It's just too much time, too many things.
Too much planning.
Too much planning. That's right. I think for young people it's great to start with, and then
I'm a science diver actually, so I did a lot of science diving in Germany. But we didn't do saturation dives either. It was mostly either nitrox, so enriched air.
Yeah.
But then shallower, of course. But yeah, never other gas mixes. And I mean, the Baltic Sea also isn't very deep, so it was mostly at 20 meters.
Yeah. So we do dives regular air down to 150 feet, which is about 50, very close to 50 meters. Of course, the bottom time is just a few minutes there for if we want to do specific collections. So we routinely dive anywhere from 20 meters to, let's say, 45 meters. But at the most, just air, or we do different mixes in nitrox, whatever it is. I prefer this type of diving so much better. The thing is, we're not limited with time. It's not like we have a 1-week field season. If a dive does not go well today, well, I can go there tomorrow. So it's I like that. I like this.
or you have an ROV and you can dive anytime.
That's right. Well, I like to be in the water.
Mhm. Your personal little ROV.
That's right. Yes, I definitely like to be where I am, where I have access to the water any time of any day of the year. So
That's awesome.
yeah.
Yeah, I haven't done a lot of diving lately, unfortunately. I don't have any diving equipment. It got stuck in transit somewhere and I lost it. So I had a dry suit that I sent over from Germany when I moved to the US, and that never arrived. So,
Wow. Well,
yeah.
that's and I know dry suits are very expensive.
Yes, yeah, that was a bummer. It was a tri-laminate. That was a nice one.
I had a DUI
Yeah.
DUI dry suit. Yes, from San Diego. Excellent. In the late mid '90s, when I was diving in Alaska for 3 years, and it was
Wow.
excellent. And I sold it. And it kept its value. Very good material. But I've done my fair share of cold diving, not ice diving, just cold diving.
How cold was it?
We're diving winter time in Juneau, so which is pretty south Alaska. So that's, I don't know, the top layer of the water was frozen.
Oh.
It was very cold. It was very cold. Yeah.
Yeah.
But if you're a beginner, a young graduate student, those things don't matter to you. You just go and do it. But I've been diving for a long time. I love it.
Oh, we have a question, Mr. Narrator. Nick, is AI helping you in some way, and how? And if not, what do you think how it can help these initiatives in the future?
This is a very good question,
Yeah.
and it's and believe me, academics and philosophers and many sections of the society are thinking about what you just asked, and I believe there is a room. My initial reaction to this question is it could be useful under certain circumstances. I can give you some very specific examples where I would use it and where I would like to use it. I'll start with a very specific example. So, at the stage of my career, I write proposals, I write manuscripts, but I don't have a lot of time to learn, for example, use a very useful language in detail, R, which is amazing for statistical analysis and graphing.
Yeah.
Right now, with AI help, I can tell. Let's say I'm going to use those verbs loosely here. Okay, I can tell to AI, I would like to do this type of analysis in R. How do I do this? And it will give me a very good start. So this is an example where I think it would be extremely useful for me with no negative consequences, because I really don't gain much at this stage of my career to spend hours and hours actually you need days and days to start feeling comfortable with that particular language. So that's one personal example that I have.
Yeah, I second that. I did learn R before the option to use AI, and it's a lot of work. It's like learning a new language. So if you have help in that, then it can help you really save time. And I use it for if I have to write anything in Python or Bash or whatever, HTML.
Absolutely.
You can easily convert.
I'll give you another application, another example that I think is extremely useful. Let's say, for example, people whose English is not their first language. So you write a manuscript, you write a proposal, and you would like your English to look very good on paper. So this can be done with the right application of AI. I think these two examples, are extremely useful applications. A negative application of this would be a student a day before the report is due is asking ChatGPT, can you write a 10-page report on this thing? I think that's not a good application.
Because you don't learn anything, right?
That's right. That's right. Are these examples good enough so you understand where I'm coming from? I'm asking the audience.
Yeah. Somebody mentioned just finished my first big Python project using generative code, which is a very useful application.
Yeah.
I mean, the limitations currently are you're still coming up with your own ideas. You can just use it to help you analyze data sets, synthesize or summarize some data that you have. You could even suggest publications that they might read. AI scans publications and summarizes the information for you, because sometimes there's hundreds or thousands of publications you would all love to read, but there's only so much time we can spend. So, yeah.
So there are definitely many useful applications. Absolutely. I mean, it's going to stay controversial for a while until it's, I believe, let's say, widely accepted. And but I think AI is going to. I mean, obviously there are many people have been thinking about this much deeper, many more than me. But obviously, the extended application of AI will have a major change in the society.
To use it wisely.
That's right. That's right. And I believe jobs will be lost forever. New jobs will be created. So people have to be ready for this.
Yeah, I know there's a lot of applications also in automated tracking for animals. So they're actually integrating that into ROVs as well. So if you want to keep the position relative to an animal, especially very tiny ones, that's really difficult. So there's automated software that does it. I think there's a researcher at Ambari, an engineer working on this. We used, I think that was 10 years ago now when I was working on that project. We used neural networks for tracking individual chromatophores on the back of cuttlefish, to see how they changed over time, to see when they expanded and retracted, and that was very successful. We could track thousands of individual chromatophores over days and weeks of the animal, and get really beautiful correlations with pattern we'd show the animals. So that was kind of the start of the earlier versions of AI.
Chromatophores are the cells in cephalopods
Thank you.
that are responsible for these unbelievable changes in coloration in the octopi that you see in YouTube, or the mating dances of squid or calamari. I mean, there are so many YouTube videos out there, and I think most of you have seen them.
There's one particularly good one. I think it's called Kings of Camouflage. That's on YouTube. That's really worth watching. It's all kinds of different species of cuttlefish, octopus. They can do amazing things.
Unbelievable. To finish this thing with this discussion with AI, I think it's going to be. I personally think it's going to have as big an impact as the entrance of internet in our life, or even maybe the effect of industrial revolution in our history. So I really think we're looking at that level of an effect. That looks like a piece of trash.
Oh yeah? I'll try not to forget logging.
This looks like plastic. seaworm here.
Yeah, unfortunately, we see a lot of trash all over the ocean floor. So we found a couple of methane seeps earlier today. We saw them we saw them first kind of in the distance by a couple of clams, and we found microbial mats, and now we're scanning the area for more. We're zigzagging along a fault line, actually along several fault lines that are meeting up about north of us, and in the hope of finding more methane seeps.
Jess, what about developing or adapting, because I think those things exist, adapting a machine learning program to go over the countless hours of these videos.
Yeah.
The number of species are very few,
Yes.
really, very
Yes.
few.
Yes, they have actually been working on that, and there's a researcher at Ambari. She works at the automated species identification. So they have big databases. I know a couple of people at Geoma actually also that they were, I think there was zooplankton. Oh, I can't talk and log at the same time. Missed it.
No, I think the discussion is very interesting. Okay, well, that was a predatory ascidian, okay, for the audience. But, yeah.
Sorry, I made a late remark. Yeah, they had a zooplankton scanner, and they had a training data set for students, and you could click through and assign whether it was a copepod or some other type of zooplankton, and that would train the I think it was a neural net that they used. It would train the classifier so that in future they can automatically identify the different classes.
To the left there is a hermicrab, just on the left corner of the screen. Let's
It looked like one of the species that we collected actually.
let's
Do you
keep,
think?
let's keep an eye for. We lost it, huh? It's we're still looking for more methane seeps, but if I get a little faster warning,
Yeah.
we can. Alexis, por favor, un poquito más rápido. Podemos. Okay?
Yeah, because we have to stop and then backtrack a little bit,
Yeah.
maybe. Sometimes the ship needs to update its position based on where we are in relation. So Falcor we're tethered to Falcor, of course. So we always have to work in synchrony.
Yeah.
So we're looking for microbial mats. You can usually tell that by discoloration on the ocean floor.
To the left, yeah. Okay, we'll stop on this and we'll continue.
Oh yeah, that's one of the ones we collected.
That's
I
right.
think. Yeah.
Yes. Together with us is Dr. Guillermo Guzman, a specialist in the local fauna, with many papers and species descriptions, especially in crustaceans. So here we're looking at what is the genus, Guillermo? The genus? Could you tell us just? En español, no.
Okay. Todos estos cangrejos ermitaños de profundidad pertenecen al grupo de los Parapaguridae, con el que colectamos anteriormente es del género Parapagurus, pero este es de otro género de la familia porque la anquela está redondeada. Eso es probablemente un Sympagurus o un Onchopagurus.
We're looking at the genus Sympagurus or Onchopagurus. You see this, it has been collected before, but only very few specimens. In order for you to do a good study, biological study, you need several specimens.
I think we collected one on the first day, if I remember correctly. I was there. Yeah, let's keep going.
Yeah, we can go.
So we extended our dive time until I believe it is 8 a.m. now. So that gives us a little over 5.5 more hours to search for methane seeps in the area. So we'll hope we'll find something. We already found a couple of smaller ones. And yeah, let's see. We'll keep an eye on the sonar, so if there's any bubbling at all going on, we'll see that. But the last seeps that we found, we could identify more by the species that were living there. So it was a specific type of clam that has symbionts, so it lives of the methane itself. And we found microbial mats. They were mostly white in color. We took some samples for our colleagues aboard.
The genus is of the clams, of the white clams, is Calyptogena. We have a question. Guillermo, the question is, which Chilean institution is participating and where the samples are going?
La mayor parte de estas muestras van a ir a dar a la Universidad de Turopat, al Museo del Mar de la Universidad de Turopat, para su análisis y de ahí coordinar con otras instituciones la identificación del material.
Can we zoom in on the rock formation on the left? I was just wondering if those are carbonate depositions. Please. Okay, thank you. I think we can keep going. It might be carbonates.
Well, I wish the geologists were awake.
Yeah.
Yeah.
Yeah, it's all biologists in the room.
Yeah. Well, we're pretty happy with just collecting
We
them.
have I'm sorry, we have a geologist and we didn't introduce him.
Oh.
Sorry. Our multimedia man. Do you know if these are carbonate outcroppings, maybe?
No idea, but they look like.
They look like it, yeah. It could be, yeah.
Well, I salute our geologists. Okay. Well, what about a giant Ecuran proboscis at 2 o'clock in the morning? Just happily collecting detritus, and now it's been retrieved, as you see it, by some powerful muscles that they have in called limniscus. Oh my God, I don't remember right now. But that was the feeding apparatus, let's put it another way, of some Ecuran worms. So they stick the proboscis out by means of almost like a hydraulic way, and they leave it out there. It gets full of detritus, and they retrieve it back to their mouth.
Oh, I forgot. Did you translate that? That was the museum in Antofagasta?
Yeah. No,
Oh, no.
in Santiago.
In Santiago. Oh, I'm sorry.
El museo está en Santiago.
Está en Iquique, más al norte de acá.
Okay.
Same area where you're at, just down a little bit, right there. Yeah, just bring it up that way. Up a little bit.
Oh, Iquique.
There you go.
Oh, in
Okay,
Iquique.
Iquique,
Oh,
yes.
I'm sorry.
You were point for.
Thank you.
That's not on my map. I don't know. So we'll have to pay attention to that. No way, no one. It was on whatever bathymetry you gave me. Was it bordered or anything?
Yeah, thank you for writing out the name of the museum.
I don't know if that helps or not. I don't know what it looks like otherwise.
Bien diferente la formación acá.
Mhm. The formation here is very different compared to what we have seen so far.
Hey, Charlie. Yeah, can you just keep us honest regarding that 12 nautical mile zone? Make sure we're not going to pass into that with our zigzag pattern, please.
Zigzag pattern. Maybe I can try to get some of the geotip.
So you're only getting the co-pilot screen, you're not getting the pilot screen.
That's right, yeah.
Okay. I'll see if I can get the EMTs to possibly help us load that up on the co-pilot so you have a copy.
We have, I think even the co-pilot will go, so we've got our own instance up there. Yeah, possibly if you've got geotip with the
That I don't have. I don't have it with the boundary. I just didn't know if you guys were able to tell where we were at. Otherwise.
I can see the ship. We close my boundary on like this.
Uh-huh.
But I can't see where the ROV is.
Gotcha.
So I can let you know if we're approaching it.
Okay.
If it's good.
That would be helpful. I'm staying pretty close to that 50-meter ring, so if it looks like we're close that way, just give me a shout and we'll back off.
Sounds good. Excellent. I'll do that.
Thanks, Ollie. How are you doing today?
Very good, yeah.
Good, thanks. So I'll try and give us a heads up if we get close.
Okay. Thank you. Just coordinate with the bridge because we're getting super close to an area that we don't have a permit to go into. But this fault line that we want to visit is the fault line that we want to visit is right along the track line.
Nautical mile away from the edge of the 12 mile, so 30 miles out.
Oh, okay, we should see close to that then.
Another mile.
there's some interesting rock formations. I wish the geologists were here.
So, I mean, judging by this, I'd say we're pretty much gonna stay away from there. We're not going any further east really than 2.10. Yes. Which is only like, I don't know, 500 meters away. Yeah. It's okay.
I can't even, in the sea logging, define a dominant or unique feature. I'll just say it's an other feature.
Oh, there's a question direct to the Mr. Narrator. That would be me for now. And if there's going to be an investigation in Peru, or there will be an investigation. I have an answer, but I'm not. I do know that the next expedition of the Smith Ocean Institution is going to be in Peruvian waters. So yes, there will be an expedition and another investigation, but I'm not involved at all in any way with that work. Other scientists will be flying in and they will be working with Peruvian colleagues. But I cannot even give you, for example, the title of the project. I'm not, but I do know that the next expedition is in Peruvian waters, and I'm sure Peruvian professors, researchers, are doing excellent work. But I'm not. My base is the Caribbean. Okay, so that's an area away from my concentration, the region of interest.
I think so I just looked it up. The expedition is called Journey to the Nazca Ridge. So they will explore the Nazca Ridge Marine Reserve. I had a question, Nick. If you're saying your research is based in the Caribbean, what is your objective here? I guess, are you comparing it to the species you find in the Caribbean?
Well, my geographic region is the Caribbean.
Mm-hmm.
I have done working. I mean, for example, I have projects in Mediterranean. I have a project in Taiwan. Yeah, I am. But also, I'm a specialist in copepods. And with my good colleague Paulo Gorgosino from Brazil, we are trying to build phylogenies of certain groups. And I expect that the collection of myofauna, including copepods from the sediments I'm collecting, will give us access to species that we don't have access to. So that's one thing. Second, I'm here because of my application of genomic tools to measure biodiversity, and two of the taxa that I will be targeting, brachiopods and chitons. I am sure I can provide some good ideas if this, about the position of these lineages that we're collecting. Third is my previous knowledge with, or experience, I would say, with expeditions. I don't say that I have a huge experience, but I have done expeditions in, Alaska, U.S. Caribbean, Bermuda, Taiwan. So that's
wow.
Yeah. So I have done some work.
So you have a lot of seagoing experience then.
Yeah, I consider myself completely marine in terms of experience. Yeah. Small boats to oceanographic ships. Yes.
Oh, that looks like. Could that be a mat? Microbial mat, down on the center.
That looks like a microbial mat.
There's something going on there. Yeah. I'm sorry to interrupt you.
No.
Yeah. We also write in one of the fault lines. There's definitely some shells.
There's some shells.
Yours.
Ah, well, the big shell to the left there.
And I keep forgetting to log.
Yeah, that has to be
Yes.
Calyptogema.
Wow.
Calyptogema. A gigantic sea cucumber in the background to the left.
Rubbish, las
Yeah.
conchas chicas son de la otra especie.
Yeah. It's not a branchiopod, is it? Don't do that to me. No, no. But,
Or is it?
uh,
Or is that on?
what
is that?
that looks like a branchiopod from here. Just we will try to collect the suction.
If that's a brachiopods, then I would strongly suggest.
I wish it was like this, so I could see the bivalves.
You could see the-
But it's like this now,
Yeah,
vertical.
it's really hard to say.
Yes. I would suggest to use the suction and jam it between the two crabs.
Sorry.
There's a log jam in there, that's for sure.
So I'm referring to
Grab the hose and then I'll.
a couple of hemicrabs stuck on suction tube. Yeah, I have the feeling it may not be, but we'll have to give it a try because it's.
Yeah, I don't know. We'll try though.
I'm 50-50 right now. But
Yeah.
it's-
I know it's difficult to see. Yeah, but this is definitely a seepage area. You can see the microbial mat, the discoloration on the floor. There's one clam, and there, at some point, there were probably more. You can see the clam shells.
Yeah, absolutely.
What's the temperature?
1.79 degrees.
Oh. It's not higher than ambient. But it's also, there's not a lot of seepage going on. The seeps we found earlier were a little larger. But we're on a good path. And it's right along this fault line.
Yeah, unfortunately, the best camera you're gonna be able to see it in is this one.
I'm wondering if we can just push the sea cucumber.
Cucumber with the, like, the shell just there.
Yeah, so Nick is gonna, he hopes this is a brachiopod. We will know when we see it. I think from this position it was hard to tell. Oh no, oh!
I turned it off.
Okay.
All right.
So sorry,
No.
sea cucumber. We're so
No, sea
sorry. We didn't mean to. And anyway, the sea cucumber is just fine, of course. It's not a- Well, if it looks like a brachiopod, it is a brachiopod.
Oh yeah?
I wish still a little chance that it's not. But
Yeah, but even if it's not, I mean, it lives close to a methane seep. That in itself is very interesting.
No.
Oh, and there's the sea cucumber.
Brachiopod. Professor Guillermo Guzman also thinks it's a brachiopod, so let's see. And this is a different species.
I did read some publications on brachiopods that are associated with methane seeps, but I seem to remember that they were found further south of Concepcion. And I haven't found any evidence for any up north, but nobody's ever found a seep this far north in Chile, so.
Bring it back.
We will know more tomorrow.
I don't know if it went in the hose or not. I don't know if it went down it. If it did, we can put it in a bile box and pick it all
I
up.
see.
We can see what we got. Yeah, that's not gonna go down in there. So
I see.
what we're gonna do is we're gonna put it in a bile box.
Oh.
Yeah, it's definitely.
It's a brachipod?
It's not a bivalve. The only
Yeah.
other thing that could be is a brachiopod.
Oh, wow.
Yes, and there's a second specimen, but there's still work to be done with the first one. So also, I cannot. I need to mention the role of my professor, Tom Shirley, who is the one that introduced me to Armando, the principal investigator, and so he suggested that I could play a positive role in the expedition. So thank you, Tom.
Put it in one of those bile box lumps that everyone is in, mark it whichever one is not. I tried to see. I still have a little bit of suction on it, but
Oh.
now it's off. Just try and shake it out in there again. It'll go. It's going. There it goes.
Fue un coral.
It's in there. Good job, Claude.
Wow. Was that Biobox 1B?
1B. Okay. Happy with that?
Yes,
Okay.
very happy. Yes. Thank you.
Was that the brachiopod end in one of the crabs, or?
No, it's just the brachiopod.
Wow.
Yes. It was very close to the left leg. Well, yeah, we're fine. Yeah. And
No, I think we should keep
keep
looking.
going. Yep.
This is a rather small seepage, but it's something.
It's not the Spanish dancer for the person that is asking. It's a sea cucumber, but not the Spanish dancer. The Spanish dancer, also known as the headless chicken monster, is primarily pelagic. It's in the water column. Okay.
Don't forget to log. Yeah, it looks like there was more seepage at some point. You can see on the right there's some clam shells, some microbial mats.
We got a comment from a viewer that may be right, that what I call a sea cucumber, which is part of the phylum Echinodermata, may not be a sea cucumber. That is, probably either a sea anemone or maybe a Scyphozona. I didn't get the chance to look at it. But we are not afraid to revise what we say if there's, if obviously the evidence pointing the other way. That's what this is about. I guess my excuse is that I'm up 20 hours looking at the screens.
I think I'm at 22 now.
Yeah.
23. But this is science.
Yeah.
And we're really excited that we found some seeps today.
Yeah.
So this helps. We're still waiting for Drew's glow sticks. He said it's going to be a party and he's going to bring glow sticks. I think he'll be here. He said he's usually waking up at 3:30. That is an hour from now.
We'll continue, yes.
Okay, yeah.
Yes, I expect Andrew to be here in 3:30, yes.
Yeah, he's an early morning person.
Yeah.
Me, not so much.
No, me neither, yeah.
This dive will be a very long one too. We extend our bottom time. We will be down here, the total time we will be on bottom is 25 hours. That is, I can't do math anymore.
Yeah. If you see a brachiopod, let us know from the audience.
Oh, yeah.
You know, some people, I mean, my eyes are not rested anymore, and I could definitely use some advice.
That looks like carbonates.
Sponge. Yes, sponge. We stop for chitons and brachiopods, so let us know.
Didn't we sample enough chitons? Are there any other species than leptochiton?
Yeah,
'Cause
definitely, yeah.
Okay.
More species, yeah. Yeah, all these are, yeah, it's a sponge.
Yeah, we've reached one of the outer boundaries. We're kind of mowing the lawn here, going over a field of where we think we have a good chance of finding more methane seeps. Could we go a little higher maybe to see microbial mats if they're, or just to get a larger area cover?
Bottom time is how many hours?
25 total, and we're coming up at 8 a.m., so that's 5 hours and 20 minutes.
So 25 hours total?
Yes.
The total bottom time will be 25 hours.
Yeah. Originally we were planning for 21 hours, but then we found methane seeps and thought we wanted to hang out here a little bit longer. And see if we can find any larger seepages. There's a lot of sediment. Oh, is that?
Fantastic, thank you.
That was probably us on the way here. Kicked it up, huh?
$8 a.m. will start going up, or 8 a.m. on the ship?
8 a.m. will start going up.
Okay, 8 a.m. Ascent, the start of ascent, and more plastic stuff.
That's the current plan. Oh. I'm so bad at logging. Quite logging and talking at the same time. And looking for microbial mats.
And then for the audience, just to get an idea of what will be the schedule for us is we may if we stay here until 8, then we'll have probably a chance to sleep for 2 to 3 hours, and
While the ROV is ascending.
yeah, and try to be up when the that's a sea cucumber, definitely. Try to be up when the ROV is ready for us to go get the samples.
And then Sebastian stays on Falco for a little while. We'll get all the samples, and then there's another planned dive for tomorrow.
Starting?
That's a great. I have the plan up here. I do not have it on the
All
top
right,
of my head.
all right. Yeah.
That will be the current plan is 1 p.m. local
1
time
p.m.
launch. That would be 3 p.m. on bottom,
Okay.
and then that gives us 4 hours for the other dive site. But if we can find any really large methane seeps here,
Okay.
I have a feeling that this plan might change.
Okay.
Oh wow!
Yeah, that is.
That is beautiful.
That is so gorgeous.
Oh, but that might have been what we saw on the backscatter too. It's a lot of rock. We need a geologist here. My rock descriptions are all rock.
We're all asleep.
Yeah.
It looks
Wow.
like basalt.
Looks like volcano. It's
It looks like a tough rock. That's how much geology I know. Like I have no.
Oh, thank you.
Unfortunately, my knowledge in geology is abysmal.
Okay, it's not. I only have the option lava flow or volcanic chimney.
It's neither of those things.
It's. I just say other feature.
I would say it looks like basalt.
I think it's.
Very
I think
dark,
it's
volcanic, ocean crust.
I think every time we try to describe a geological term, us here, the biologist, geologist having a nightmare.
Yeah.
You know?
Oh yeah.
Yeah.
I only know enough geology to get me in trouble.
Yeah.
Mm-hmm. Well,
See?
but our geologists were extremely supportive in coming up with the dive plans particularly for this dive. We went over the maps with them for countless hours, stared at the backscatter,
Cool.
all the acoustics that we could
Yep.
sample the night before. That's
Yeah,
great.
this.
Oh, and there's a
Wow, it's a beautiful sea anemone. Wow, that's a piece of rope. Okay. Well, that's the list of the. Yeah, Hermosa. That's right.
Really pretty sponge back there too.
Yeah. Okay. Wow, that's another.
oh wow.
That's a beautiful hexactinellid. What is the genus? Regula? The genus. Hexactinellid. Que bello, no. Beautiful. Yeah.
Lauren is back. I'll just give her a quick update. We found a very tiny seep.
Oh, yeah?
Yeah.
Oh, how
Just a-
exciting. That's good news. Did we mark it on the map?
No, but I have an observation from market.
Oh, fantastic.
It was Yeah, no clams.
More stuff, others.
We'll take a quick break.
Okay, espero para Andrew. Yo sí, Andrew va a llegar aquí pronto. Si Andrew voy para dormir, aunque sea. A las 3:30, Andrew va a llegar aquí. It's a sea anemone, yep. Biologists. Let's see, what of that basalt outcrop? We're still looking for the elusive methane seep, one with a lot of activity. I guess this is where you could use an AUV just to check the area, and then the real work is done with the ROV. Well, I mean, that's a whole different operation, though. But this seems to be an area where unattended surveys would be a plus. Have people used AUVs from? I'm sure they have. I mean, this is.
Yeah, we used, we had a couple of the Ambari AUVs on here beginning of last year when we did the Mid-Atlantic Ridge job. They went out and kind of found areas. We came back and actually found black smokers. Yeah, it was pretty effective.
Very effective, yeah. Okay.
Can you zoom in on some of these white little tubules on the rock?
Look at the left right here.
We saw some tube worms before.
We never got the chance to sample them.
No, we didn't. We couldn't mark them on the map, though.
These are calcium carbonate tubes, but let's see if the worm is inside.
Doesn't look like it.
No, from here, no, nothing.
might be one.
You're right. That's the same type of tentacles, kind of a.
Hang on, I'm gonna log this.
Yeah. That's gonna be impossible to. I mean, the best case scenario, if you just scrape it, you'll get, well, maybe with a suction, but I don't know.
I don't know. We just wanted to make a note of them if nothing
Okay.
else.
Okay, sounds great. Yep.
It would be great if we could sample them, but
The other side was better to sample, but
Yeah.
yeah.
I think maybe we could keep looking for a better
Sure.
potential place
Yep.
to sample.
Sounds great.
Cool. Thank you. I just wanted to see if there were any live ones in there. It looks like there are a few,
Yeah.
so that's kind of cool.
There's a question to the Mr. Narrator, and if we could introduce you to Professor Guillermo. Professor Guillermo has been working until now, which is almost 2:30. So he was very tired. He just left to went to sleep because tomorrow he's going to have a full day. I know Guillermo Guzman for just a couple days, and I can tell you that I respect his opinion very much. I immediately saw how much he knows. I know he has published several papers. His specialization is in crustaceans, but I think he has a kind of a knowledge of a natural historian. So I don't know much of his background. I wish he was here to tell you, but I tell you that he's very important in the expedition, and the vast majority of the big things we're collecting are going to go under his care. Okay, and eventually they will be placed in the museum in what was it, Iquique?
In Iquique?
Iquique, yes. But I don't know his CV, in detail.
No sleep, still seeps.
It's been patented to help us fund the next cruise. So it's,
I'll have to make everybody t-shirts.
yeah. Squat lobster.
They're so fun.
Loaded question about if we know anything about deep sea chemical ecology. And if pollution affects it.
That's kind of a bit.
This is not a discussion for 3 o'clock in the morning for
Yeah,
us.
man, wow.
Ask us about the beautiful colors. I mean, we can deal with that right now. So.
Oh, I mean, I guess it depends on what you mean by chemical ecology. The chemistry of the water, the chemistry of the rock, biochemistry. I mean, I'm a biogeochemist, so I'm thinking about all of those things together at once. And I'm an oceanographer by training, so I know a little bit about deep sea chemistry and chemical oceanography. As to whether pollution affects it, that's also kind of a broad question, because, like, what type of pollution? We're finding microplastics in actually quite a few microplastics here in the deep water, which is depressing but true. Could talk about ocean acidification, oil spills. I mean, to try to put it very simply, the ocean is an incredibly complex system, and it buffers itself and to a certain extent can adjust to change, but it's also a delicate system, and there's a lot about it that we don't know, and we're constantly learning how human activity affects the chemistry of the water. Organisms communicating with each other using chemicals. Oh, okay. So you mean more like, say, quorum sensing, for example, or chemical signaling, like with hormones and things like that. I know less about that. I do know a little bit about quorum sensing, which is chemical signaling between microorganisms. And it's basically a language that microbes use to communicate with each other, and it's an interspecies language. So almost a universal chemical language. Whether pollution affects their ability to communicate with each other, I do not know, and I don't know how much work has been done on that, but it is an interesting question. Yeah, that's a good question. I don't know.
It looks like just take a look. I have one example to give about chemistry and communications. I know that because of my it's probably nothing, but it has the right shape. Just a minute. Let me just observe an organism, and no, that's just this just oh. It looks like a sponge.
I think the thrusters are kicking up a whole bunch of debris.
Yeah, it's not a. I just saw some appendages at the end. It looked like a hexagonal sponge. We can go. So I was saying that I know of an example of very close to our labs. We have in the local reefs we have 6-7 species of ostracods. We have many more, but those 6-7 species of ostracods leave bioluminescent cues behind them. These bioluminescent cues are species specific, and they can be used for mate recognition. They can be used for prey avoidance. So I believe this is a perfect example of chemical communication. And the bioluminescence, now of course, I don't want to get into bioluminescence, but it's sometimes they have different colors and different patterns for different signals. So ostracods, if you do not know, are small crustaceans that just happen to be in small cells. These are very small organisms. You need a microscope to see them, okay? But it doesn't mean that they're not complex just because they're small.
Yeah, I think that's a misunderstanding that things that are larger are more complicated than things that are small. Certainly, We tend to think of microorganisms as being very, very simple. And on an individual level, one bacterial cell is a lot less complicated than an elephant. But when you think of microbial communities and the way that they function, if you think of them as living systems, they're quite complex and very difficult to study. So it really all depends on scale. And even a single organism can be a very complex living system. And there's lots of things about these living systems that we still don't understand.
I like to use in terms of complexity and size, I like to use the example of Loricifera, which is one of the smallest animals that exist. It's a different phylum, and within 150 microns, it has pretty much all the organs that we have, and the number of cells are so few, yet the basic organization is not different than ours. So just try to imagine our body compacted in just let's say a couple thousand cells. Okay, a very, very small size, but they're fully qualified as animals and
Wow.
as complex as anything else.
They're single-celled organisms, dinoflagellates that have a functioning eye. They can detect light and dark. Yeah. Ooh, what's that up ahead?
Oh yeah, the cells there. Oh yeah. So the other one that we found with Jess was, let's say, 75% of this, just to give you an idea of the size.
Mm.
So even smaller than this. Same kind of disarray of valves, but it did have one of those calyptogema kind of wedged into the sediment, not like this. This is completely.
Yeah, this one looks done.
Yeah.
You happy to move on?
Let me just log it as an observation.
Okay.
Okay. I'm good, thank you.
Okay.
more of those worms up ahead?
Oh, let's see those. They're in a very nice density there. Let's see.
Take a look. Can we zoom in on that rock with all the white tubes on it?
Yeah, or like, I don't know how we're gonna be able to collect those kind of a combination of scratching with the plastic end of the suction. I don't know.
Coming to a pause.
I don't know. I don't know if there's a good way to get these off of there. I mean, the suction would probably make the most amount of sense, but probably pretty well stuck on there. And the Brock is too big to take.
Yeah, it's too big to take.
No.
So maybe we could find a smaller rock with some live samples on it and just take the whole thing.
That's a good idea, yeah.
Yeah. Let's take a look around here and see if we can find a smaller specimen with some live worms on it, maybe. Because we still haven't collected any of those. It'd be nice to have some. Yeah, maybe over on the left there.
I'm surprised not to see coffee mugs. I gotta say something to the audience. It's not very unusual, even for professors or for let's say professional biologists to stay up all night and work. There are some instances that this has to be done. I mean, obviously, as graduate students, I can attest. I believe I speak for everybody. We have done our overnights.
Yeah, fair share of those.
But at this point, obviously, we don't need to do overnights. We have constant schedules, of course. But even that's a brachiopods.
Yeah.
Yep.
Oh yeah.
Yeah, that's a brachiopods.
Why don't we? Can we take that whole rock? It's got tubeworms on it too.
Not telling how big it is, but we can try.
A little bit on the big side. So, I mean, there are instances that we have to work overnight. Well, this is a great example right now. This is an unbelievable opportunity, and we don't get to do this very often at all. So it's worth staying overnight. There are instances where, well, even preparation for a huge trip, sometimes you may have to stay overnight to prepare if you haven't managed your time very well, or perhaps a huge deadline before a proposal. Even though that's not you need to be able to write proposals with your head straight. But these are some examples. Everything can be avoided with better management of time, but still, there are instances you just have to work.
It's got to be done.
Yeah. Grande.
big rock.
I remember my first overnight in the lab. Once my professor came down to the lab and saw me working there, he said, let's just go for breakfast. So it was nice. We talked about stuff, and I knew that he appreciated that I worked there and overnight. But he was telling me about time management and stuff like that.
You're just gonna have to wait till that clears. You can't see anything. It's just starting everything.
I was very lucky in grad school. My advisor would tell me not to come in on the weekend.
Oh.
I'd say, I could come in on the weekend to do it. He said, no, wait until Monday.
First day when I met my professor, his gift was a coffee cup. So that was an indication of what was expected. Yes. But that's the guy that I'm still working with right now. So 30 years after I finished with him. So
You must still like him.
yeah.
Is it too big? Or-
Yeah, probably the. I don't know.
Look at your set.
The brachiopods still there. He just.
Oh, is that the brachiopod?
Yep, that was a brachiopod.
Oh.
Just kind of underneath that. I saw it just rolling over.
Yeah. No, I don't think we're going to be able to get it.
Too big.
Yeah, we lost also the brachiopod. I know we have the species, but we got, so that means that they're around. So we got to keep an eye. I'm gonna keep an eye on them. Yeah. Yeah, I think.
See what you're seeing.
Yeah.
Okay. Maybe we'll find another one. Does that one have a live worm on it? I don't think those are. Nick, do those look alive to you? I don't.
I cannot see very well.
See any
No.
little. No, I don't think so.
I cannot see tentacles.
Nope.
Yeah.
Right here. See?
Oh, you see. Oh, my eyes are very tired. Now I see them, but
Oh.
my eyes are done. I think. Yep. No, I.
Oh, okay.
Yeah.
All right. Yeah. Where do we want to put that?
Well, you could go in the rock craters. Are they
You
full?
want to put it in the rock box?
So that, you can try and break that off. That's stuck to it. It's pretty solid rock.
Is it
Yeah.
part of a bigger rock?
Yeah, it's part of it.
Okay.
No. Okay.
We'll find.
We'll find something else.
Yeah. If the brachiopod was still attached to the rock, maybe we could do it. But no, it's okay.
It's all good. Hi, Drew. Drew's back. Did you get some sleep?
Oh, yeah.
Oh, good.
Pretty much the entire time.
Oh, nice.
Check if I 21. If I go to sleep right now, what time?
I think that the ROV will be back up on deck around 10:30, 11 o'clock if we get off bottom at 8.
So I'll get out for breakfast. Well, let me see.
There's a question about organisms evolving elsewhere and exploiting the seeps, or life arising and evolving around one. And same question for hydrothermal vents. I mean, yeah, that's a really good question. So there are certainly theories about life arising around hydrothermal vents, and the origin of life being associated with hydrothermal activity or
Yep.
geochemical activity. Another brachiopod?
No problem, thank you.
Okay. Do we want to use the sucker, or what do you want to do with that? Yeah. Okay.
Yeah.
What's the sample ID on this? I don't know.
Oh,
I don't know what system people have been using. I'll just say brachiopod.
I like to point things at the TV and say things. That's what I do.
Yeah. I haven't been doing sample logging, but just stepped away. So.
Andrew, people were keeping track of what you were doing.
Where's Drew? So yeah,
Yeah, it's another one too here too. I'm sorry, I just pointed to the second one. I just saw it.
which container is it going in?
2.
2? So yeah, life may have originated around hydrothermal vents, and there's certainly evidence that life at hydrothermal vents is very ancient.
Oh, it's free. There you go.
Ah,
Excellent.
all right.
As much as well. Yeah.
Another one over here, I think.
But that's actually a really big question in microbiology. Is there's a theory that was put forth by Bass Becking that everything is everywhere, but the environment selects. Meaning, all microbes exist in all environments. We only see the ones that are adapted to live in a particular environment, because microbes can so easily be transported everywhere.
You want to try and get that one.
So, yeah, it's possible that some microbes evolved later to be able to live in these extreme conditions, but a lot of
Yes, sir.
microbes
Yes.
under extreme conditions, like at hydrothermal vents and hot springs, are very old.
That's the first one in the jar. So.
It could be considered living fossils in their own right. They're deeply branching ancient lineages of organisms. Getting another one?
Yeah, same jar.
Same drawer?
Yeah.
Gotcha.
Same rack, same. Yep.
This one can be brachiopod too.
I don't see it. There it goes. Up the
Perfect.
tube it goes.
Okay. Excellent. Excellent.
I saw the first one go in the jar, so this one.
Perfect. Brachiopods, a very interesting phylum. A phylum that has a huge fossil record with 3,000 described genera, and obviously they were one of the dominant organisms in the early Cambrian. But they went through the Permian extinction and they almost completely got wiped out. To the point that we have now, remember the number 3,000 genera. Right now, we are down to 300 species. Okay, so this is obviously a phylum in decline. It's actually, if you think about it, about how ephemeral things are. Now, of course, that's a very different scale. Okay, but here we're talking about a phylum. We're not talking about single species. Okay, So,
Are we happy to move on?
yes. Now they're important in benthic ecology. They're filter feeders. They're characterized by the lophophore that they have inside, even though they look like bivalves, like the mollusks that we eat. They are very different. And they can be anywhere from very tiny to a size like the normal bivalves that you know. These are definitely on the small size. There are areas in the world that you don't need to go 3,000 or 4,000 meters to find them. For example, I have seen them diving in Alaska, and I know there are shallow water brachiopods in New Zealand. But we're here for a very specific reason, is because this ecosystem is likely undisturbed for millennia and representing ancient, representing some very old ecosystems, similar to those ecosystems studied in the Atacama Desert.
Hey Nick, I'm back.
All
I'm
right.
your data logger. I am Jessica Eberly from Germany. I work previously with
Continue. Thank you.
on research expeditions at Ambari and some at GMO and the German research ships. Done a little bit of expedition planning, and I've mostly been, I'm not here to do my own science, I've been mostly doing some science support for Lauren, and helped her with whatever she needed. Done a little bit of the dive planning in the last couple of days, with the help of our geologists and collaborators in the region.
To the left.
Yeah, was really happy that we found some methane seeps today. What's that down there?
Probably a piece of trash, plastic. Yeah. Just a few years back, there was a German expedition at the Puerto Rico Trench. Maybe, I think it was organized by Seckenberg institution up in Wilhelmshaven.
Yeah.
And I know the PI there, but Pedro, it's actually he's Spaniard. Yeah, Pedro Arbizu, a good buddy. I don't know, were you involved at all with?
No. I'm mostly doing microbiological projects.
Okay.
So I think Senckenberg's they use
Zoology.
sampling, zoology.
Yeah.
So no,
Okay.
I was not involved. I would have loved to be.
Yeah.
No, I love research expeditions. What is?
That's our waypoints.
Yes. So Drew was just asking if those were our waypoints. We have kind of a zigzag line over the area that we covered previously. So we looked at the map and the backscatter data, and these are kind of the outer boundaries of some anomalies that we wanted to check out. And this is also the area in which we found previous seepages. There's also at least 2 fault lines coming together. So we hope that we can find some more seeps and take some samples that will make a lot of people happy. So the idea is that we'll just kind of mow the lawn, run some zigzag lines over the area, and if we see anything of interest, like microbial mats, clam beds, or the mussel, the clams that we. Sorry, I'm really tired. Or sometimes you can see tubeworms. But usually the first indication that that we've seen were really the clams. They're they were alive, so they were active seeps, even though we don't see any bubbles. But we do keep an eye on the sonar as well.
The same group from the Senckenberg Institution, together with people from Oldenburg.
Oh, I know a couple people from Oldenburg.
Yep. I mean, heavy zoology stuff, especially with benthos.
Yeah.
They had done the Angola Basin. I don't remember which ship though. The Angola Basin, and what's the basin? Okay, so that's okay. Obviously, that's West Africa. There's another basin on kind of on the upper part of Africa. I mean, I don't remember right now the other basin. However, why I'm saying all these things. So they have, they were specifically they went there specifically for myofauna, and they extracted about 1,000 copepods from the samples.
Wow.
Yeah.
Did you look at all of them?
No. This is while I was still a graduate student. Yes, they look at all of them. They did. I mean, they had a lot of people back then. The group now is much smaller, but almost every specimen was a new species. So they had almost 1,000 new species.
Oh, that's a lot of work.
Yeah.
I did some copepod identification
Oh.
during my master's
Yes.
degree at Geoma in Germany, and that was.
Yeah, this will
was
break
hard
you.
work.
Yes.
Yeah.
Yeah.
Yeah, taxonomy is hard.
Yes. Yes, it is.
Especially for copepods. So
Yeah.
you have to count like their legs and the little. What is the word?
You have to count the CT on their swimming legs,
Yes.
and you have to count the CT on the mouth parts
Yeah.
and their antennae. Yes, and the patterns and yeah. Yeah, it was a powerhouse. That combination there with Oldenburg and Senckenberg, but I think the group is not as strong as anymore. But they're very good people.
Mm-hmm. Yeah, I studied with a couple of people from Oldenburg.
Yeah.
Oh. Did we find any significant seeps in my sleep time? It depends what you call significant. So we did find seeps with living methanotrophic clams. We found a couple of those, but we didn't find any larger seeps with bubbling or large clam beds yet. But we have about 4.5 hours more of bottom time. Yeah, it's still going. It's very hard to believe.
It may have been the Congo Basin. I think that's right now. I cannot remember. I'm very, very fuzzy in my head here. I don't know, but it sounds like yeah.
Do you want to take a break, maybe, Nick?
I was expecting somebody to log in. That's why I'm still here.
Oh, to log?
Yeah. I was expecting somebody to log in the chat.
Oh, okay.
Yes.
I see.
Yeah.
So you're waiting for a friend?
Yeah.
Oh, okay. No, I just don't want to keep you up all night.
No. I'm gonna go to bed. I need to sleep to be able to do something tomorrow.
Yeah. Yeah, tomorrow is all the sampling and
Yeah.
the species description. Hopefully
Yeah.
we'll find some new ones.
Today.
Oh, today. Sorry. Oh, I forgot.
Okay, let's see. I gotta put an end on this. So the time is 3:36. Okay, 3:45. I'm out. All right, here you go. All right.
Okay. Well, let's see.
It would be ideal to be able to talk to the friend and find a ship within the next. That would be.
Oh, I'm not.
Yes,
I'm
yeah.
not. We're not kicking you out of the room. It's just
Yeah.
we all need sleep.
Yeah.
But our room is empty, so
Well, it's gonna be a great sleeping. Yes.
my snoring will not keep me up.
No.
So our room is not empty, some nice ones.
Yeah. It's like, we haven't collected this one, but we're not gonna. Guillermo is not interested in starfish anymore unless it's something extremely peculiar.
Gotcha.
Yeah.
It's more rock crabs,
Yeah.
rock lobsters. Sorry.
Squat lobster. Squat lobster.
Spot lobster?
Yes. Which is not even a lobster. It's just a just a name. Yeah, but that's the name it's stuck.
Huh.
So in case the group finds a seep with some mussels again, Guillermo was talking about one commensal crab that is, I don't know if you remember, when we start moving those mussels, those crabs start sticking out. So he was telling me that these are most likely new species, so he was really interested in maybe try to capture something with a scoop, but anyway, that's not so easy, you know. But he was talking about those commensal crabs living in the bivalve.
Oh, interesting.
Yeah.
This could be carbonate again, or it's just compacted sediment. I'm not
To the left,
really sure.
the little whitey stuff.
Those little shelf-looking. Yeah, I don't know how to describe them.
Okay.
Rock formations, but not really. It looks like what we've seen on the. I think was it the first or the second dive side?
Second.
Oh.
A little bit like we saw in the second dive side. Second seep side, sorry.
It's really, really difficult to find methane seeps like this. It's like walking around with a flashlight in the dark and trying to find them in an area, in a huge area. This might be carbonate. Thank you.
Great.
Cool stuff, yeah, thank you.
Oh, chocolate. Thank you. So carbonate deposition could be an indication that there have been methane seeps in the past, but we're not sure if that's what we're actually looking at here. Let's hope a geologist wakes up soon and they can tell us more about that. Sea urchin. Oh, that looks like trash.
Goodbye to the audience, and we'll talk more in a few hours. Thank you for staying with us. And here comes Dr. Drew, as you say.
Dr. Drew. Night, Nick. Thank you, and I'll rest all the biological expertise in the room. morning, Drew.
Good morning, everyone. I'm Drew Palmer. I'm a professor at the Florida Institute of Technology, where I'm a biochemist and astrobiologist. Florida Tech is about 30 minutes south of Kennedy Space Center, for those of you who've never been there, which is highly likely. We're a small little school, but I work in areas of sustainable space agriculture and then Mars habitability and astrobiology. So on this particular expedition, I'm very interested in looking for organisms that survive here in the Atacama Trench, around methane seeps, and in other places in the search for weird life or life that survives in hostile environments, as ways of looking for biosignatures for life on Mars. FIT is a good school. It is a great school. That's right. Not just a good school. We're a great school.
Where in Florida is it?
So, if you pick Orlando as your destination, which I'm not sure why you would, but you go about an hour towards the East Coast and then down about 10 minutes, about 20 minutes. So we're about there. We're about a half an hour away from Kennedy Space Center. So we have a really beautiful coastline. We actually sit on one of the longest estuaries in the world.
Drew, how far away is that from St. Petersburg? That's where we'll be, I think, next week. You can come visit.
It's actually not far. I don't know the exact distance. It's a couple hour drive. Yeah.
Okay.
There's a question about research is being done. Today's research, we are examining. We're here going through the Atacama Trench looking for particular methane seep sites. That's what this 25-hour dive has been about.
Yeah.
And we are in the high teens, I believe, for the dive length at this point.
We're in the 20.
Oh, we're in the 20 mark. Okay.
Actually 9, yeah. No, you're right. In 15 minutes, we'll be at 20 hours.
It was a total guess. There was no mathing done.
Just excuse me for a minute to say hello to a friend. And then
Sure.
I can, take off. And I'm going to speak in Greek since we're offering many different languages. Γέρη, καλημέρα. Δεν ξέρω αν με ακούς καλά. Εδώ πέρα είμαστε με ένα group από επιστήμονες από διαφορετικά πανεπιστήμια, από την Αμερική, από το Πορτορίκο, από την Ισπανία και από τη Χιλή. Και κοιτάμε οικοσυστήματα που είναι στα πολύ βαθιά νερά, στα 4 χιλιόμετρα, και κοιτάμε την βιοποικιλία του βένθους και την βιοποικιλία των μικρόβιων. Είναι ένα φανταστικό μέρος, είμαστε σε ένα φανταστικό πλοίο με την τελευταία τεχνολογία και ελπίζω να βρούμε πάρα πολλά πράγματα για τα βαθιά οικοσυστήματα της Χιλής. Να έχεις καλή μέρα. That's it.
Thanks, Nick.
Doesn't Calimera mean good day?
Καλημέρα. Good day.
Good day, yeah. Okay. Yeah. No.
Good morning. Good morning.
Oh, good morning. That's right. It's late in the day, and yeah, we just learned the stream has been on for 20 hours and 27 minutes. Yeah, I can't do math anymore.
So we're not teenagers anymore.
Yeah, that's right. We'll come up at the current planners will leave bottom at 8 a.m. So that gives us another 4 hours to look for methane seeps in the area. We found some earlier last night. There were smaller seeps with a couple of clams, some microbial mats. And we're moving along fault lines, just scanning the area and seeing if we can find anything.
This has been an interesting challenge. So in some cases, methane seeps can be very visible, but in many cases, like the ones we found earlier, they really are areas of, there's high biological density, right? But then there's not a lot of, they're not easily seen, right, without getting down here and actually looking. So it's not like there's tons of bubbles coming up from a lot of these sites. Some of these sites that we found barely had any bubbles at all, but we could see, the clams and then the biomats, and so there was a lot of activity there, a lot of biology going on, but they weren't visible unless we were down here looking, and so we've got to just traverse this area. I think what was the analogy about the flashlight in the dark on a mountain?
It's a good analogy. We do have a sonar that would help us if we had some bubbling, but we're not sure that we would expect any, because the seeps that have been described so far in southern Chile, they didn't see any bubbling there.
Yeah. No, definitely we only saw really, really small bubbles at the seeps we found earlier in in this particular dive.
And the seeps we found are so far the deepest seeps in Chile.
Yeah.
And the northernmost.
These are the northernmost seeps also?
In Chile, yeah. I think there's a couple in Peru.
Excuse me.
Oh, I think there's another one off of Iquique, what Guillermo mentioned.
Isolate them and then send some of them, once they've got them identified, they send some of them to us for screening to see whether or not they have what we call plant growth promoting properties, which is exactly what it sounds like. They can do things that help plants grow better. And we've built a couple of microgravity simulators in our lab with engineers at Tech, and we try to see whether or not we can improve how plants might grow on the space station. And I'll give a shout out to one of my PhD students who's actually defending his dissertation exactly on that subject in about a month. I've been editing his work while I've been out here.
He didn't want to join the expedition.
Oh,
no time.
not the right time. Under normal circumstances, he would have been really excited to. I have two of my PhD students are graduating at the end of this summer, and they both would have wanted to come. But I do have one of my graduate students with me, Katie Hubrick, who literally graduated about a week and a half before she left for this trip with her undergraduate degree, and she was like, "Did my master's degree just start?" I'm like, yes it did. So she's been having an amazing time.
So these are samples for her thesis?
She actually already kind of has her thesis. She's she's hoping to start working on this to start getting work together for her dissertation. So Katie is planning about 3 or 4 years ahead.
Oh, wow.
Yeah. She's extremely well organized, and her undergraduate research, I think now almost reads like a thesis. She's been working with using oyster mushrooms, actually, with these Martian simulants that we talked about. So she's built a new waste management stream, basically, to improve. Sorry, I knocked something over with my hand and I caught it at the same time. So, both clumsy and dexterous. But she's been growing using basically inedible parts of plants that we grow in our simulants, and then recycling them to support the growth of oyster mushrooms. So. And she was kind of one of our Atacama enthusiasts, so she was super excited to get a chance to come to the desert, and a little more worried about what it was going to be like on the ship. But she's really loved it. I work in a marine biology associated department. I work in the ocean engineering and marine sciences, but environmental sciences is based out of there. But this is really the first time I've done any extensive marine work. I've previously done some estuary work, looking at human-sourced contaminants in organic sediments. So basically, what we call muck that accumulates at the bottom of some lagoons. It is actually the technically correct term. There's a scientific term for muck. And
What's the difference to mud? Muck is biological, or
muck is
organic?
organic. Yeah.
Okay.
So it's basically, it's got the soil sediment in it, but then it's suspended with a high density of organic matter. So a lot of times in Florida, you find it in like the estuary where we are is a low flow estuary. So they estimate that like things take like 100 years to diffuse out of the lagoon is like one of the estimates. And so what happens is you get, lawn cuttings that get go straight into storm sewers. We get agricultural runoff. When in Florida you get hurricanes, often will cause wastewater treatment plants to overflow, and then that goes straight into the lagoon. And so then all that organic sediment deposits at the bottom, and it creates a really anoxic or low lack of oxygen area. It's really strong hydrogen sulfide smells, and there's some interesting microbes in there, but we were mostly looking to see whether or not it was a reservoir for things like antibiotics and sunscreen and all these other chemicals. So my background is actually originally is chemistry, and so we were screening that for whether or not those sediments held onto those compounds. And they do, but unfortunately. But I guess while the chemicals are there, stuck inside the muck, one of the positive things is we did a series of experiments where we basically put the muck in buckets with water, and then we agitated them. We shook them up to see if the chemicals moved from the muck back into the water column, which was a big concern, and not extensively. So the muck settles back down, and because it's kind of greasy, for the less technical term, they held on to a lot of those compounds, and so they hold on to those more than they get suspended back in water.
Does it depend on the type of muck, I guess, or the different? Are there sediments involved as well, or is it all of them?
Yeah, so the muck's got a mixture of that. One of my colleagues, Austin Fox, does a lot, is really sort of one of our muck experts. Sorry, I was looking at something in the chat. He really spends a lot of time out there. He was actually the one who taught me that muck is actually the technical term, because I was like, really, what is it actually though? And he's like, that's really what it's called. And I'm like, oh. He spends a lot of time sampling. He does a lot of nutrient cycling in freshwater bodies like that in Florida. So he's looking at like nitrogen, phosphates, levels like that. So we've actually been working with him on some of our Mars work, because he helps us understand, as we transform this regolith into soil, what's that doing.
What kind of organisms was it again that you're using for the degradation? That was a type of fungus, right? I think I remember.
Yeah, so Katie's working with the oyster mushrooms. That's one of the ways that we're trying to use inedible biomass and convert it into edible mushrooms. But along the way, the mushrooms sort of transform the regolith, so they deposit other materials. They have a variety of chemicals that they release into it that cause the regolith to become more fluffy, more soil-like. Literally changes at the microscopic level, if you look at the particle sizes increase, their hydration increases, so their ability to interact with water. They hold on to different ions. So it's really pretty interesting transformation. We also have another student who's using the cyanobacteria, Anabaena cylindrica, which is a freshwater cyanobacteria, which is what we call a pioneering species. And the pioneering species are organisms that move first into a new ecosystem. And we're currently discussing our waypoints and zigzags.
And Drew, you were mentioning cyanobacteria. They found cyanobacteria in the Atacama Desert, the driest desert on Earth. I found that fascinating.
Yep,
that's kind of the basis of this project, right?
Yeah, and that's one of the reasons why we actually picked cyanobacteria that we're using was because its resilience. It's also interesting because cyanobacteria, like A. cylindrica, is capable of doing both carbon, what we call carbon fixation or photosynthesis, right, to bring carbon into a biological system, into the biome, but also
Are those tubeworms? Or the might have been at some point. Sorry to interrupt.
No, yeah. But they also, under low nitrogen conditions, like we would have on Mars, they can actually do what we call nitrogen fixation, which means they can take nitrogen gas and make it into fertilizer, essentially. So they can do both, which makes them a really good idea. And I have a former undergrad who is now also, just like Katie, starting as a master's student, who's going to be doing that. It's a very juvenile version of some of this that we've seen.
We find lots of evidence for methane seeps in the area. We found a couple of smaller seeps last night, and I think we ran into a couple of carbonate depositions today, some empty clam shells, and we continue to look for methane seeps.
So I will wrap up because I took that question of does FIT work with NASA, and I basically said, here's all what stuff I do with NASA, but there's lots of other people who do other stuff with NASA. We have in aerospace engineering, I know there's a number of people who work there. I know a couple of there's a robotics team or two that have done stuff looking at making Mars rovers, sort of new versions of Mars rovers, and a mining robot also. And there's a group that I helped advise on plant growth that was making a sort of an automated feeding system.
An automated feeding system for
plant growth system.
Oh, plant growth.
Yeah, sorry. No, they're going to bring the food right to your mouth.
Yes.
Yeah. The ultimate in laziness. But no, it had they basically were making sort of a conveyor belt mat system that would, they basically could use like almost for growing microgreens, and then they would just shave the microgreens off and give a low feeding wet drip nutrient system.
That's smart.
Yeah. And so they were trying to fit it into a pretty small space. There's a system that they use on the International Space Station called nanoracks, which is basically helps define kind of the size of the object that you're going to put up there. And they were basically, it was like 2 nanoracks sized, which is pretty tiny actually. So it had to control the environment, control lighting, and provide a way that harvested automatically. So all that the astronauts would have to do is basically pick up their shaved microgreens.
Very nutritious.
Yes. It's high density nutrients relative to time to produce and cost.
Oh, by the way, high level nutrition, this is one of the areas with the largest upwellings, so nutrient rich deep sea water from the deeper layers wells up in the coastal areas. This gives microbes a lot of food to grow, and they use up all the oxygen in the water, and this is what causes these big oxygen minimum zones here that we've seen.
Yeah, we've seen a couple of those.
Yeah.
Hey Jess, do you remember what the name of the green worm at the bottom of that was, of the screen?
That long
Nick.
one with the, I wrote it down somewhere yesterday, I think.
Nick had it right. He knew it. Yeah.
Actually did a Google picture search and found it. Let's see.
Was it like an echo something?
I'll do the picture search again.
Yeah.
My brain is not working anymore.
We've seen a few of them. Was it a spoonworm?
Oh, that sounds familiar, yeah.
Yeah, that does sound familiar. As soon as I saw that, it sounded familiar. Nick had it right on, so.
Yeah, green spoonworm.
Yep.
Bonellia. Yeah, we are traveling along a couple of ridgelines.
Looking for active methane seeps.
Your job is slow you down just a little bit.
Yeah, no worries.
Thanks, sir.
Oh, they wish your students good luck on their defense.
Yeah.
That's
No.
nice. Greetings from Chile.
Thanks. Yeah,
Greetings.
it's a busy summer. I've got 2 PhD students and 2 master's students who are all trying to defend by August. So I've been pretty much in my downtime here, which has not been a lot.
Oh, yeah.
I've been editing everybody's work. So
Wow.
it's,
You've been incredibly productive. I've been trying to put together a proposal, and I still have to work a lot for that, but there hasn't, like you said, there hasn't been a lot of downtime.
well, it's easier to sometimes work on that because I know I have to get them out. So
Yeah.
I do have a proposal I'm supposed to be working on. I have opened it once since I've been in Chile. So and went, nope, not tonight. So
Oh yeah. No, I do have to get this done. The deadline is next week.
who are you proposing to?
It's a project called the Peer Site Program. That's with the Florida Institute of Oceanography.
Those guys. No.
Whoa! The funny thing is, they bought the Western Flyer from Mbari, or got it gifted. I'm not entirely sure. And the Western Flyer is the boat on which I met my husband. So and he, and the circular thing is, he and I. So we went on a kind of traveling gap year, and then he was asked to build this RV for the Florida Institute of Oceanography, for FIO, or for the company who sells it to them. and he agreed. So he's back on the boat that we met on.
Well, that's nice.
On the Western Flyer, and sends pictures, old memories.
Yeah. I mean, FIO is a really good program, actually. They're USF, I think. They're affiliated with the University of South Florida.
Could be.
So I think it's USF, not UWF.
I am not sure. I don't know.
There's a lot of marines and oceanography stuff in Florida.
It makes a lot of sense. So, I mean, they have access.
Who'd have thought?
Yeah, who'd? They have access to this great body of water with a lot of a lot of research opportunities, but it's really shallow. So yeah, that's what I've been working on, and it actually happened during the preparation of this cruise. So I reached out to geologists that wrote a publication on the Cascadia margin, the Cascadia subduction zone, because I'm not a geologist, and I wanted to understand more about how to find methane seeps and where we could expect them and how we can find them. So those guys really, they helped me a lot. And then one of the authors, she asked me to write this proposal.
That's cool.
She's working at FIO as, I think, the science coordinator.
That's very cool.
Yeah.
That's okay. Yeah, we got an answer. It's USF. St. Pete.
Yeah, so I'm really excited about that. I'm gonna visit next week, and then I'll also go back on the Western Flyer. But I heard they have delays, so we might not be going out. Just zigzagging here across a couple of ridge lines, hoping to find more methane seeps. And we have about 4 hours more of bottom time.
So, Jess, you may not know the answer to this question, but since I also work with plants, so most of my plant work is really about how the microbes affect the plants. So we kind of joke that plants are just sort of a vehicle for us to do all the science we really like. I strongly think about the relationship, like circadian rhythms, which play a huge role in plants. So there's over 1,000 genes in plants that are regulated by day-night cycles, right? And can be trained on day-night cycles. And I'm just wondering if you know anything about circadian rhythms here in sort of the benthic environment.
That's a very interesting question because it's always dark.
Yeah.
I'm not sure, actually. I don't have an answer to that.
I mean, I know that most organisms, just generically seem to have a rhythm of some kind. So I just wonder if they base it around some other fluctuation in the environment.
We can ask the audience if they have an idea.
Raising more questions than we're answering.
There's actually a Nature paper, so I did a little bit of research just now. There's a Nature paper on the biological rhythm in deep sea hydrothermal mussel. Bathymetry.
Does the abstract say yay or nay?
Usually, if it's a Nature paper, I'd assume it says yay, but let me scan this. Let's see if my brain can still process that.
Either way, it'd be interesting, right? Because if they have one, then you have to figure out what's the environmental sink.
I do remember reading something about self-sinking at some point, but let's see. We reveal that tidal cycles predominate in the transcriptome physiology of mussels, fixed directly at hydrothermal vents. 1,600 meters depth.
Do tides have a... So again, this is showing my my terrestrial roots and lack of oceanography knowledge. Do tides have a big effect out here? I think of tides as being.
Tides do. So there's
So, yeah.
a school of thought that seeps are affected by tides, and we can see that because at certain times during the day we can see plumes during mapping that wouldn't be present at other times of the day. They seem to sync up during high tide situations. So I don't know if that's really been solidified in the idea, but it's definitely school of thought in a lot of scientists that way.
Huh. Right. So there is a relationship between tides and seeps potentially, and that would potentially affect biological productivity, which could then impact cycles. So, I mean, it seems pretty reasonable that most organisms would find some, what we call abiotic or non-living signature to kind of latch onto as a way of controlling a rhythm of some kind, because there needs to be on-off cycles in cells. You can't run everything at 24 hours a day.
Yeah, so we worked together with a researcher who did some studies on that. It's called tidal pumping, and the effects were assumed to only be present in shallower areas, but it was actually proven that they also play a large role in the deeper areas. So yeah, what Chris just said is very true. It's for methane seeps. It's also for nutrient distribution. Sorry to interrupt, but yeah, in estuaries it also plays a huge role. So the interstitial water, so the water between sediments, is really transported by tidal pumping.
So it's interesting. I'm right now. I'm very focused on the idea of these sort of environmental signatures and the roles they play. So again, thinking about one of my students, the one who's looking at microbes from the space station, one of his underlying ideas that he's investigating is the role that gravity plays in regulating interactions between organisms. So how do the bacteria respond to plants, both in and out of microgravity? And so, this is one of the things he's been trying to simulate, because, one of the gravity may be one of the only factors that has remained kind of a constant in the evolution of biology, because it changes less than 1% across the surface of the Earth. So it's pretty constant. It's been that way since the evolution of life, at least the biotic world that we know of right now. So.
So we're flying over more rocky terrain.
Looking around before we can see.
What is that?
Couple of crinoids. Yesterday, at one point, we passed this huge field of crinoids. Actually, it was really impressive. Yeah. We were wondering about the crinoids because we were actually it was Jess and I. We were looking at the crinoids, and they kind of remarked that they seemed like almost like sunflowers, and we were wondering what if they had the ability to kind of turn around in the water so they move with the current.
Wherever's the most advantageous position.
So whatever, turn the direction of whatever water's moving their way. That's cool.
Yeah, Nick also confirmed that. There's also a couple of crinoids that can swim.
Yeah.
So I think they're called feather stars. Not these though.
Yeah.
And yeah, crinoids are also considered living fossils, which is the title of our research expedition. It's living fossils of the Atacama Trench. But it's also living fossil microbes. We have a lot of microbiologists aboard.
Yeah. Astrobiology is heavily dominated by microbiologists, actually. Because when we're thinking about what life on other planets looks like, at least within our solar system, we're all pretty much convinced it's going to be on the micro scale if there are things out there that are surviving, because we would reasonably see evidence of more, multicellular, more complex, not more complex, multicellular, larger organisms would leave larger footprints. Big stuff, big signatures. Small stuff, small signatures, so to speak.
Do the signatures differ, and can you tell by the signatures who was there?
Well, so that's part of what we're trying to figure out, right? Is, one of the reasons for this expedition actually is looking at what are the limits of detection of various biosignatures. So this is built coming out of work from Armando, one of the PIs on this, was looking at microbes in the Atacama Desert and trying to figure out what the limits are of detection. They found ancient microbes, may just be the remaining DNA of them. Some of them might still be there and just difficult to culture. And that's one of the things that brings us to the trench, is to look for those signatures of those microbes as well. You were saying earlier about like cyanobacteria that might have been marine in origin that ended up in the Atacama Desert, which would have happened millions of years ago. So the signatures can vary, right? And so we're looking for signatures of different products of different metabolic pathways, DNA, other things like that. So I think the microbiology and the biosignatures kind of overlap a lot. And that idea of what signatures go to what organism is something we're actually trying to figure out. Is can we build a sort of a database where we could identify the types of organisms that we had based on the kinds of molecules that we find. And one of the things we're searching for here from my group are some interesting novel metabolites that might be indicators of that, especially as we think about ocean worlds like Titan and others, where there could be large bodies of water.
That's a microbial mat.
Yep. Yeah, please.
Yes, please.
Right, so got a small microbial mat here.
So that's a hint for
Yeah.
some sort of seepage.
Yeah, so it's a sign there might be something around. So the sort of cobweb that you see there, that's a microbial mat, and we've seen those almost exclusively at the sites that we've seen other seeps at. So we're just going to kind of take a look here real quick.
There's also some shell fragments.
Yeah.
Looks
yeah.
to be
Yeah,
shell
kind of
fragments around
all
it.
through here. So.
Let's see. Then we turn around and there's this.
Yeah.
Yeah, I think we can keep going. We've seen microbial mats like that, and we're pretty much right along the.
Yeah.
Oh, I should probably log that.
Yeah. See a few here, kind of scattered around. We got. This is a nice spot. Okay. Well. This could also be evidence of what we're referring to now as an extinct seep, right? And these could be kind of the remaining mats that have persisting on.
Very true. All right. Shall we keep looking?
Yes.
Okay.
Yes, please. Yeah, we'll keep looking. Okay.
Thank you.
Thank you very much. That's awesome.
Yep.
So in our 3 previous dive days, we had not seen any evidence of these microbial mats, and we've seen a fair number of them now over the last 20 hours.
Do me a favor. Can you speed up the torch?
We have a question. No sediment samples. So we kind of filled up on sediment samples quite a lot. We have about 5 empty push cores that we can use.
There's another mat.
There's another one.
Yes.
So we're in a good spot. So there's definitely something going on in the area.
Yeah, I mean, we definitely took some time and grabbed a bunch of cores about 4 hours ago now at this point, and then decided to carry on. Oh, look down there. So it looks like it's kind of scattered across the site, the field here. Oh,
yeah.
So this definitely looks like this area might have been a more active seep at one point. I'm gonna pin it down. There's lots of shells. How much gene sequencing is going to be done on the samples? A lot. So different groups are going to be doing some different things. There's going to be quite a lot of metagenomic analysis. Oh, yeah, right there.
Oh, more clams.
Oh, and down here too. So. Oh, look right there. Yeah.
Oh.
No. Oh, yeah, that. Okay. Yeah. Yeah. Jess, do you see it up here on this camera? Like right there?
Yeah.
That's
There's definitely some clams.
Is there anything you wanna focus in on?
I think we can keep going. Maybe look for some. Maybe let's look at the clams and see if they're alive on the. Oops.
Okay. We'll look at the clams. Which ones?
Great question. Yeah. Maybe the ones that are in the bottom, showing up in the bottom now.
Which ones are you?
Oh.
Why don't you come on? Because I think we're seeing a different monitor, so.
I'd say we can keep going. Those are kind of smaller seeps, but I think we're on a good.
Yeah, this is a good
track.
sign.
Yeah.
Yeah. All right, so you want to keep moving?
Yeah.
Okay.
Maybe we can find some larger fields.
Yeah.
But there's definitely some seepage all along this. Wow.
Yeah.
Okay. I'm wondering if when we climb up this hill, if we can find some more as well.
So there's going to be we're looking at doing some metagenomic sequencing. There's going to be some transcriptomics. Oh yeah, wow. Okay, I see that on the kind of the preview cam over there. Do you see that?
Oh yeah.
Yeah. Oh, that's really nice. That's a really.
Yeah, feel free to pick one.
Yeah. Yeah, there might be some turbid water here. Do you want to set?
Sure.
Just do you want to try and look here a little closer? Oh, and then down here also to the left.
Oh wow.
Yeah. All right.
There's more coming up.
Do you want to settle down anywhere?
I think those clams appear to be dead.
Okay. Yeah. Yeah, we're not seeing. Okay.
But it's a good indication of recent seepage.
Yeah.
So there's And it's definitely all over. Wow.
This is interesting. So we might be actually mapping a recently extinct seep.
Yeah.
So this whole site might have been that. So what we saw before might where we found an active seep, that might have been like the vestiges. So that's a lot of hand wavy, but I work in academia.
Yeah, I'd say let's keep going. Maybe we'll find a big seep.
Yeah. No, that's good.
But I think we should keep the track lines a little bit closer now that we found something,
Okay.
so we don't pass it by.
I'm backing down along the track line. Right
Yeah.
now.
Thank you.
So in addition to gene sequencing, there'll be a lot of, we have experts in lipidomics, so who'll be looking at different lipids, isotope effect concentrations and relations. We're going to be analyzing the water column in a couple of these sites. My lab's going to be looking at culturing some microbes that might have
Let's check these out.
interesting metabolic
Sorry.
properties. Oh no,
The ones down there?
it's over here.
Yeah, because there's a
There's
lot of
that's a
microbial
pretty big
mats.
mat right there.
Yeah. And then let's check if those are alive.
There's a lot of shell ripples as well.
Yeah.
So I can't track it down a little bit here.
Doesn't look like the clams are alive, but it looks like the biomats are
Yeah.
persisting. Heavily associated with the rocks. Yeah. Yeah, I'm looking around some of the cameras. These clams appear to be smaller. Does that mean that they stopped growing due to low seepage volume? That's possible.
Could be.
Yeah.
Getting close a little bit this way.
Do you want to do anything? Do you want to collect any samples?
Can we zoom in on the? I'll come up.
Yeah, please. I'm handing you the pointer.
Right back up.
Yeah, there was something that I wasn't sure whether that was a rocket or not. Somewhere here.
That'll grab.
Do you want to grab?
You know.
No, I'm not sure what I saw. I thought I saw something about Nick.
Nick had mentioned about, Guillermo. They're usually kind of hanging, like almost on the underside of the rock. Is that just more rock, or is that? Biologist. We're both biologists. But this is the wrong kind of biology. Yeah. It's too big for my biology.
No, I don't think that's a rocket bug.
No, I think it's more rock.
Let's look for more seeps.
Yeah.
Thank you.
Do you want to collect any mat samples, or you want to move on?
No.
Okay. All right.
Ideally, I want to find some, because we do have mat samples.
Yeah.
We don't really need more.
That's fair.
We find a bigger seep,
Yeah.
would be totally worth it. If we don't find any, then yeah, let's take some there. It might also be hard here.
Okay.
It's super rocky.
All right. Well, we're moving forward. This is very interesting.
Ouch. We weren't looking at the xenophyre. We were trying to see if there were more brachiopods, and they're really hard to see for the untrained eye, which ours definitely are. Tired and tired.
Let's not say untrained, wrong trained.
Wrongly trained.
No, that doesn't sound right either.
I'm trying to look for microbes. We can see the mats.
Differently trained. We'll go there. We'll go with that. Yeah, you can see some more shells on the right.
Yeah, I mean, this is actually, overall a huge seepage area.
It is. This is interesting. This will be worth really getting into the video data and mapping this.
Yeah. Well, we could, if we have enough coverage, 3D map it.
Yeah.
Unfortunately, we do not have the M3 on the...
See that map? I'm sorry, I didn't hear that last part. Just we don't have the
M3, the multi-beam on the ROV. So
Okay.
we can't map the area. We can map it visually.
Oh, those are bigger, and that biomat's pretty large. Some of those shells are pretty big.
I think they're dead. Oh,
Maybe.
there's one alive.
Yeah, there's at least.
There's one there.
Down and look at them.
Yes, please. Are those up there? Are those what Guillermo was looking for?
The what?
The
Those-
critters. The one, yeah, that just swam away.
Oh no, we've
Okay.
seen plenty of those.
Okay.
No, yeah, I think we can keep going and look for some more.
Okay. All right. We can keep on. So we are noticing a difference in the size. Some of the shells are bigger. So
Yeah.
this really could be kind of the slow decline of a big seep, which is interesting.
I don't know if that means we need to go even lower and
Yeah.
on the bottom of this hill that we were looking at, or if there could be more up on the hill. Oh yeah, the common brachiopods on the rock to the right hanging. Wait, I didn't see them. But luckily we also sampled a couple.
And you can see how much sediment there is here. It
Yeah.
really is a lot.
I'm wondering if Lauren would like to be here for that, or if we should just let her sleep for a couple hours.
I would say that we haven't seen anything that's really new.
Other than like in terms of area.
I mean, area, yeah, right. So we have good area.
But yeah,
But nothing. I
not
think
a-
nothing new to sample. So.
Yeah, very true.
But man, this would be an awesome video. My vote would be to give her at least 3 hours. Give her 3 hours.
Well, in 3 hours, realistically, we'll be recovering. perhaps,
Hmm.
if the plan doesn't change.
Yeah.
There's more on the left.
This is the original one. I just came down.
Oh,
to the
okay.
right because
Yeah.
I couldn't see it, so I didn't want to land on it.
Ah, I see. No worries. I think there's 1 or 2 clams that are alive.
Yeah, 1 or 2 that are alive.
Mm.
Okay.
Are those tubeworms up there
Meaning by the rock?
on the rock? Yeah.
Yeah.
Oh, and I always forget to log.
Huh. Yep. We got tubeworms. So again. Oh, yep. And that's where your clam is.
Yeah. And the tubeworms. Dun, 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 dun. Methane seep.
Did we want to grab any of those clams?
I think from what I heard Lauren say was that we had enough
Okay.
of the clams.
Okay, so they got you. I wasn't here when you guys were grabbed
Yeah.
those from the other seeps. So.
I think they did. So I don't want to.
Mm,
I don't really want to sample the last one.
that's fair.
Standing here.
Earlier, when we found the seep, we did a temperature probe. What was that? Do you remember? Were you up for
Yeah,
that?
we did do temperature and there was no difference and no. And that was a seep with more
Yeah, it was
with more
More-
clams. Yeah.
Yeah.
Well, compared to some of these, there were there were like 10 clams that were alive, maybe
Yeah.
20, but yeah, there was no difference in temperature when we first measured it.
Yeah. Mm,
So I wouldn't expect anything here, but
Yeah.
we don't know.
Okay.
I was just curious.
Yeah. Oh,
So we're discussing one of the signs for a methane seep can be temperature changes.
yeah. No, I think we can keep going
Yeah.
because we have 5 push cores left, so I don't really want to take a sample here unless we find
Yeah.
anything extraordinary. Looks a bit like hydrate there, unless that's quartz. I don't. I
More
didn't see
the-
any hydrates.
It's a bunch of the brachiopods, right?
Where?
Here. And I think these are we collected like 8 or 9 of these I
Oh,
think
okay.
before, so I think I'm just noting that we see them. It looks like they're standing out from the rocks.
Oh, but you did say that they sampled those?
Yeah, these are the ones some of the ones we sampled before. Yeah.
Okay.
There's some more down here too. Well,
Oh.
by more I mean one. There's one more down there. That's more.
That is one more. by different clams. I'm not sure.
Yeah. I see.
It's hard to say based on the one live one that we saw.
Yeah. I mean, the shells
Because-
do look like a slightly different shape, though, so that might be the case.
There's one of those green spoonworms.
Got some more shells here and there.
Yeah.
Hey, RV, do you want me to start
What
moving?
is that on the left?
On the left, like over here?
Yes.
Yeah.
Oh, maybe
That's
it's just a
that looks pretty dense right there.
No yawning. Oh, we did say, what was it? No seeps. No sleeps. We found seeps, so we can go to sleeps. Oh, is that a microbial mat?
I think so, yeah. I think it's a pretty good intact one, actually.
Yeah.
Oh, that looks more like a little bit like the wispier ones we saw before. What is that? I think it's just yeah, that's animal of some kind. Do you want to sit down and look at this?
Not.
We don't have to?
We can keep going.
We can keep going? Okay.
Yeah. Kind of have to be mindful of time a little bit.
Yeah, that's a really good one though. It's interesting. Cool.
Yeah, it's a very pretty one.
Yeah.
I wonder what kind of, if it's a, I can't find the words anymore.
I mean, it definitely looks like there's some kind of biomineralization,
Yeah.
biomining kind of
Yeah.
effect there because it's actually changing the color of the
Yeah.
rocks as
Mhm.
well. So.
I mean, that's the orthogenic carbonate, isn't it?
Sorry.
Hey. Be interesting to see if the seepage increases if we go up the slope and then
As we
when
go down and up and down and up.
up and down, yeah.
We can move over to the north and go back up and slide over.
I didn't hear that.
Where would we go?
We can go further to the north.
That one or that one?
Well, right now we can see our trail over there.
Yeah.
So we started following something right here, and then we'll straight down. We can
Do you want to head like for this waypoint, or do you?
We can either go to the waypoint or we can go out in between. You guys, and if you think we might have better success going up further.
Just want to weigh in here? Talking about the possible routes.
So we can go up and then go over here. We can come over here and come up. Continue down. We can swing over there. Whatever you want.
Maybe up but close so that in case there's something we don't need to know about this.
All right.
What do you think?
Well, there's definitely activity here, but it doesn't look like it's recent. So we can go along here. So back from where we just were, and just continue along this line. Sure. Okay. Sounds good. All right, Steve.
Updated waypoint.
I was gonna thank her, but she's gone.
Yeah. One of the students was here with us. She said she's a night owl, and
Yeah.
Much a whisper.
I was gonna go grab coffee, but she's already on her way.
So you probably all heard that we were discussing the need for coffee. So I think I took about a 2.5 hour nap as it all came out, but I'll sleep when the RV's on the way back up.
Me too.
Yeah. These, then I'll probably help unload, help get my student processing, and then take a nap. And
I think Lauren is gonna help.
yeah, but she needed to sleep. I think she'd been up for 20 plus hours.
Yes.
So.
I know. I was there. No, tomorrow is gonna be a short dive, so we thought we're gonna push through and increase our chances of finding methane seeps, which we did. We found methane seeps. Now we're looking for something even bigger.
Yeah. But we've accomplished a couple of the expedition goals already, so that's good.
Absolutely.
We're looking for even better examples.
Yeah. So we've got the minimum amount of samples, or not the minimum, but we've got the samples that we took has all the groups.
Yeah.
They were covered, so now everything that we're collecting is pretty much just the cherry on top.
Yeah.
So if we find anything really interesting or a region where we definitely want to take a core, something that looks different.
I think you and Lauren, in consultation with the geologists on the team, and Oscar, Antonio, you guys have a plan for later today for the next dive, which will be kind of following the same strategy.
That reminds me of the dive plan. Thank you.
Yeah. That's beautiful.
That is very pretty, and I have to lock this. That is probably some type of anemone. Right?
Yeah.
I think we saw one earlier too. Yeah, so the next dive that was planned, it kind of depends on what we will find here. So the idea was there is a couple, more crinoids. There are a couple of different geological features, and forgive me if I don't have all the terms right. There's where the upper continental shelf ends, there's the continental margin, which is where we wanted to look for methane seeps in the first place.
Huh.
So there's one tiny place that we can. That looks like an interesting feature.
Thank you. Thank you, Amber. Amber's one of the grad students working with us.
Do you want to take over the
You are 100% right. The whispering has no effect. Oh my god, the coffee smells so good. So really, the grad students have been doing such a tremendous amount of the processing work once the samples come off the ROV or from the water collections. I know my student's been filtering 30 to 50 liters of water a day. She did 70 on one day where we did a particularly extensive water profile. So they've been working really, really hard, and they're up as soon as the RV comes on deck. They're ready to start working on it. What is that?
You're already here.
Okay. Is it glowing as an artifact of the light?
No, it's a shell. It's open shell. The inside is
Okay.
filled with dirt. So,
Amber's taking over on data logging for a little bit. Yeah, I used to be a night owl when I was in grad school, and then that's over the years it's kind of changed. So now I really have morphed into a morning person. So.
Yeah, my previous research is based mostly computational, so it doesn't matter when I do it.
Yeah. Yeah. Yeah, kids kind of make a difference. So now I get up at 4 o'clock in the morning because I can have like 2 hours of getting stuff done. Versus my wife would like to sleep in on the weekends, but my kids are like me and they don't sleep in. So at like 6 a.m., all three of us are awake. Actually, when I was in grad school, I needed, I was working on a plant project, and I needed microscopy time in doing live microscopy work on the root of the plant. And so I needed to be able to take measurements every like 30 minutes. And so the only way I could get access to the microscope I was using was to basically schedule to work nights. So I moved to nights for about 4 months. So I'd come in at like 7 o'clock at night and work till 7 or 8 in the morning. The only time I showed up to lab during the daytime was for meetings. So I was not allowed to get out of meetings.
We have a running joke in computational chemistry saying we work 9 to 5 jobs, but 9 p.m. to 5 a.m.
Mm-hmm. There's server time available. Don't see anything super active. Keep going.
Sorry.
That's my phone telling me it's 5 a.m. to wake up. I've set a couple alarms on this ship based on what we're doing. I think I have a 3:30, a 5 a.m., 7 a.m. Since it's 5 a.m., I'll do my top of the hour. So if you're just joining us, we are on hour 23? 20, somewhere between 22 and 23 hours on this dive, looking for methane seeps, active methane seeps. We identified some inactive ones. This area we've been exploring for several hours now. Looks like it's a declining seep. So we're watching sort of what happens either right as the seep is ended or right as it's ending. Oh, yeah, there's spoonworm. There's quite a number of those around here. Amber signing off. Jess is back in the data logging seat.
What was that?
I said you're back in the data logging seat.
Oh, thank you. Yeah, back with the coffee.
Yep.
At 5 a.m., 3 hours of dive time.
Yep.
More. Let's see if we can find some bigger methane seeps. Thank you, Josh.
Thank you. Well, that's a beautiful picture.
It was really weird to see people who are like now getting up.
Oh yeah.
Yeah.
Yeah, I've before we set sail, was doing my normal schedule before we like started processing samples and stuff and doing dives, and so I was pretty regularly kind of up and about at like 3, 4 in the morning. So yeah.
Oh, I will work on the dive plan in the meantime.
Okay.
Gonna need to send that out.
And I can update the schedule for the dive, like for like data logging and.
Oh, thank you.
Yeah,
Yes.
that's no problem. I mean, it's really
Yeah.
just looks like all I'm going to do is take all the shifts and move them over a couple of hours. I'll tweak a
Yeah,
little
I'm
bit
sorry about
around mealtime. I try to follow your example there and make it so that people get 30 minutes for lunch and dinner, but you know, it's social niceties.
Very kind of you. Thank you. We all appreciate you. It's pretty.
I'm following your lead. You're the first one who
Well-
suggested that, and I was like, I can do that.
No, that was because I made a mistake the first day.
Oh.
So I learned from that.
No,
And I
you
realized
gotta
that
own it and
wasn't
be
that-
like, well, my brilliance.
So I'm pretty sure tomorrow's dive will depend on what we find today, but
Today's dive.
Oh, today. Oh, no. Oh,
Today's.
no.
Then I think we should say the next dive.
The next dive.
I think the next 72
dive.
hours we're just going to call it like the day.
Okay. It's been a day. The next dive will depend on what we find today. But so far, I think the plan is to go to a different dive site. We'll see.
So that one's about 7 hours, 8 hours. It's a shorter dive, right?
Well, we had to shorten that one because the last dive is going to be our very deep
The
one.
deep one, yeah. So that's where we're hoping to
Yes.
find.
Well.
Yeah, we're hoping to find some of the macro living fossils. So, which we've already found a few. And the couple of the marine biologists have already potentially identified a couple of new species. There's still more work to be done after that statement, but
Yeah, for them, for most of us, except for me, the work begins after this cruise.
Yeah.
So this is sampling. This is the fun part. And then afterwards, we take the samples from the lab, for the microbiologists, or for the sequencing and microbial community analysis.
Yeah.
We then extract it. We spend months in the lab just extracting sediment samples
Yeah.
from the deep sea.
Here before.
we were here before.
No. So there was seepage here.
Yeah, there's seep here before. Yeah.
So we find a lot of evidence for past seepage or recent seepage.
Yeah. So this is, I mean, it's interesting. It's a fairly large area then.
I don't understand geology well enough, but I'm wondering if we're moving up on the ridge, if that'll be younger, and then there could be potentially more seepage.
Yeah. Or could it be older because the ridge is forming because it's being pushed up? So then those would be the things on the top, which would have been pushed up first.
So it should move down.
Yeah.
But the fault lines can also occur
Yeah.
after it's bulging up. So that's where I always get confused.
Yeah. Did you say wild guesses?
Huh?
Did you say that we were making wild guesses?
We're
they're
making very informed decisions.
that's they're educated guesses.
Educated guesses, yeah, as best as we can. We spent countless hours looking at maps, talking to geologists, at backscatter. So usually what we did was Alcor was mapping overnight during our while we were moving to a different dive site. So we were mapping the dive site ideally the day after that. And then the day after we would look at it and try and decide on a good spot with the geologists on the team and their collaborators. And this is how we picked this dive site as well. It's been really successful.
It really has been, right? So I mean, even if we're not finding tons of bubbles, we see lots of evidence of small amounts of seepage and former seepage. So I mean, we really, y'all definitely hit on, a success.
Oh, definitely. So the reason why I think half of the group came here is because of methane seeps. So finding these is really a substantial part of some of these projects.
Oh, definitely. I mean, from the astrobiology perspective, those were pretty critical. Methane is an important molecule that we think about in astrobiology in terms of what plants might once have been like. So we think there was methane on Mars at some point, which could have been generated by methanogenic bacteria that could have been responsible actually for creating sort of a Martian ice age and helped cool the planet, and actually may have ultimately led to Mars becoming uninhabitable. So they could have entered a bad biological cycle, so to speak. So it's amazing, even with all of the mapping and the analysis that all of y'all that y'all did, we still come down to having to get here into the site and look, and this is the process.
This is ocean exploration.
Yes.
Speaking of the Mars exploration, how would you prevent contamination from something that you bring from the Earth atmosphere into a different atmosphere and ensure it doesn't contaminate the environment that you've got there when you want to take samples?
It's a real challenge. So we were talking to a guy from 3M who specializes in building clean rooms. And, when we were like, okay, so you need to have something that won't let you bring anything in and also have you not take anything out. And he just looked at us and said, clean rooms don't work that way. And he was like, clean rooms either keep the stuff inside clean or keep you clean from the stuff inside. And so it really is an interesting challenge. Planetary protection is something NASA spends a lot of time on. And one idea has been try to irradiate everything that goes up. But they've actually had a couple studies that show that that's not really immensely effective. So you can take the wheels of the rover and roll it across some regolith from simulated for Mars, and the bacteria that get caught in those wheels can survive radiation blasts. So I think the one of the big questions about the biosignature business is exactly that, is that as soon as we get there, we've already contaminated it. There's almost certainly bacteria on the wheels of the rovers we've already put on Mars that I don't know if they're still surviving there, but the DNA signatures are there. So, you know, we may be forced to look for signatures that don't exactly look like Earth biology, but how do we know what those are? So, yeah, this is one of the arguments from why we are slowing the rate at which we're trying to go to Mars to some degree is how do you fix this problem? So that's my roundabout way of saying, you know, we don't really know yet.
So we're already colonizing other planets.
Yeah, in a way, yeah. So that's okay. So one of the ideas that I have for sustainable agriculture on Mars is the Martian atmosphere is heavily CO2. I think it's over 80% CO2. So significantly more carbon than ours in terms of composition, but it's only about 2% Earth's density in terms of atmospheric density. But we can trap that carbon because during the winter on Mars, it actually is cold enough that we get dry ice deposits. And so you could take that dry ice, collect it, and use it to generate CO2 that you could give to plants. So then the plants would fix Martian carbon. And so the first people that eat plant on Mars grown in that atmosphere would essentially be Martians. So then we'll have become Mars, part of Mars. So that feels very science fiction-y sometimes. But it's really one of the only practical ways we're going to be able to grow material there. Driest farmer of Mars.
The other seep that we found that was on the bottom of this slope, is that right? I think so. But we're coming up to not yet, but at some point, to a zone where 3 fault lines meet. So maybe that's where we'll
That might be a spot of activity.
There's something going on here. It looks like carbon deposition.
There's another question. So when you get samples back, be it from the moon or even from Mars in the future or something, how does this work in terms of, again, contamination or biosecurity? Is it like the 1968 Andromeda strain models for microfaunal?
Yeah.
Somewhere in the desert, in a secret facility underground, and then analyze the stuff? Or?
Yeah. So actually, we have asteroid samples have been returned. The lunar samples were kept under lock and key for a really long time, and they already found out that they got contaminated. And I think again, they're going to do their best to try and keep it sterile. So they have. I think the group that's doing the Mars return mission already has like a whole clean room design set up to try and minimize the contamination. But it's going to go to very select groups that have already established that they've got the protocols in place. The lunar stuff is the leftover lunar regolith that we have has started to find usage in some institutions. They're now starting to release a little bit of it. There was a paper earlier, it might be 2023 now. There were some professors at University of Florida that actually got their hands on lunar regolith that was stuck in the wheels. So from the moon lander, and they took that and they grew plants in it, but it already was contaminated with earth microbes and everything. So it's going to have to be very like a very high biosafety level facility to try and make sure that we don't get any contaminants. But again, it's the same problem is, as soon as things get here, they start to get, it's pretty hard to keep them sterile actually. So in some ways, some of the arguments are that the approach that we've had about sterilizing everything means that we're not really equipped on how to manage the contamination, right? So if we did more kind of looking at the mixture of biosignatures with actual biology, we might get better at teasing out contamination. Yeah, like I said, there's a whole NASA division dedicated to planetary protection, and they're both loved and hated because they're the ones who kind of put the brakes on a lot of things where they're like, and how do you keep that from contaminating Mars? And everyone's like, oh man.
So they wouldn't allow you to run your experiments on Mars?
Not yet. We're not there. So I mean, there's quite a lot. One of the reasons why the Mars mission is 30 years out is really the planning. Right now, just the logistics of getting there is actually, we could build something to get us there, but the 6 to 9 month travel time is a real issue. And there's radiation exposure issues that are with long-term space flight like that. To get far enough away from Earth, you start getting basically cosmic radiation. So, it's bad. It's just the technical term. It's bad. I actually was on the dissertation committee for an engineering PhD student who was building essentially a force field that based on generating a magnetic field that could deflect high energy particles. And so he was testing with an electron gun in a vacuum, and he showed he could generate a field around a cylinder. So the idea was, if you got a large enough scale, you could build that and have that magnetic field running around the ship when you were approaching like a particularly high radiation area, and it would deflect the particles. And the preliminary data looked pretty decent, but you'd have to the idea would wrap it around the passenger part of your vessel so you could protect your crew, your plants that you were growing. So he and I are actually in discussions now, because now we have a really big chamber that we inherited from NASA that we can pump down to below Mars atmospheric pressure, and we want to muck around and see if we can build his device to scale in there.
cool stuff.
Yeah.
So would you like?
Is that that's a rock?
Yeah, you want it?
No.
Come on.
No, we're good. Yeah. We actually, I think Lauren already got a rock exactly like that.
Can I have it?
What's that white thing there in the back? Was that? Is that a
This
rock?
one on the.
On the right hand side, yeah.
Yeah.
Where we're at?
Yeah, I think it's just a sponge.
Oh, I see.
You guys want a rock? You can take a rock.
Oh yeah, go
Yeah,
for it.
yeah.
Please.
Get it right.
Alright, driver, can we pause the move, please?
What was it, baby?
Picking up some rocks.
It's beautiful and intriguing how space exploration and deep sea are linked. It really is, especially as we start to think about some of the ocean worlds. We were having a conversation earlier in the dive in the morning about some of the ocean worlds, and somebody brought up Titan, and in order to get to the ocean on Titan, you have to get I think it's 50 miles of ice that you got to core through before you get there. And we were, I teach a class. Teach is a stretch. It's a paper discussion class. We get together and read papers 3 days a week on space biology. And one of the projects we were looking at was a plan to build a device that could go through the 50 miles and.
What? That was yours?
No. That we were reading that paper,
Oh,
not mine.
I
No.
see.
So we were looking at a couple of designs for it, and the biggest problem really is after you get through 50 miles of ice, how do you control it?
No, I remember a presentation where that was included.
Yeah.
Oh, I see.
Yeah.
Yeah.
Hmm?
Found the alien map.
What is it, Amber?
Yeah, there's some symbols and stuff.
Oh yeah. There's like
I
a
think that's
organization. It's just trash. Yeah.
But you're all right. I will love this. Thank you for the reminder.
Yeah, it's just a map.
It's a napkin. That's weird.
Oh yeah, it looks like a picnic.
You get one of those?
Yeah, if you want one. Yeah.
Yeah. The biggest one you can get?
That white on the rock, it's just a discoloration. It's not an animal, is it? That's not really a sponge, is it? It is?
Yeah.
Wow. It really does just look like a white spot.
I don't know if you can get that one to try. I like your ambition.
Be bold.
Look at that one, that's fine. Alright. Let's go back a little left. What's that?
May I request a rock after you guys are done?
Sure. I mean, it's my call, but I don't care.
I didn't.
It's a rock.
Yeah.
I'll request a rock.
Okay. Since we're stopped, yeah, Amber would like a rock as well.
My hair caught.
one would you like?
Geology is coming.
Yeah.
Yes, can I have that one?
The
Yeah.
one in the back?
Yeah.
Yeah.
Alright, if we can reach out, my dude. It's good.
Is it big? It's
It might be more in there than you think, but we'll see.
It's just 2.5 hours. It's just 2.5 hours. I need to finish this one.
Cuba.
It's like 10:30.
Yours might be cooler than mine.
Oh wow, beautiful.
Might be swapping rocks.
I mean, it's nice to have.
I thought I wanted this one. You were wrong. You picked the wrong rock.
You picked the big one, which is not as cool as the small one that I obviously wanted from the beginning.
Well, yeah, but they only put one off, so I don't have to do it.
Pretty good view to sit on it. There you go. Oh, yeah. Moving on to sit. Good job.
Yeah, it's
Hmm?
definitely a sponge on there.
Yeah.
It's supposed to be
Cool. Into the milk crate. I mean, the rock bottom.
And then, yeah, there's the. Cool.
Oh yeah, yeah. Now it's very clear to me.
Now that we're done playing.
Yeah.
Back to work. Thank you for that.
What do you think? Should we go further up the slope, or should we try kind of further down?
Up or down?
That's the question. So the other seeps were down. The
Yeah.
question is, will we expect them further up, or should we try?
Which way are we
have
headed
a-
right now?
We have a geologist present. We can ask.
Yeah.
Oscar.
Geologist. We're doing Ask a Geologist. Should we go up or down?
I need to do the shit right now.
Yeah.
Whoa. I thought up.
Okay.
This is good.
Okay. You're thinking up?
Yeah,
Okay.
you go down
Right
first
there.
and make sure. Okay, go up. I don't know.
Oscar says go up, and then said I don't know.
But up, you mean north?
Yeah.
Do we mean north?
I think that would be east. Right? Up would be.
Northeast?
Oh, that up. The different up. Okay, we could try that. So to go to skip the transect because we're we have 2.5 hours more of bottom time. So cutting this short a little bit and going up. I think that's where the triple, the fault
triple
lines kind of
faults.
converge.
So which of the waypoints is that? Or is it?
Could try that.
Okay. You just want to cut over to those then? Yeah. They popped up in some other places. That's former seep activity. Kind of, you get some microbes that alter the mineralogy. Discussions.
I'm not around. I just decided.
What do you think?
What are we doing?
Okay.
Should we move in that direction?
Great, ROV updated waypoint, please.
Updated waypoint.
Excuse me.
This is what I was expecting. There's a large,
Yeah, it's super. Yeah, it's a really heavy sediment area, or marine snow area. So it's just sort of suspended, and they're a little bit above. That's interesting. Mhm.
Thank you for the audience that have been sharing with us these super excited moments. And well, I hope like some are leaving now. So thank you very much for being there with us.
You should introduce yourself.
Well, I am Victoria, and I am, maybe you have already listened to me these previous days. I am a student and I'm helping in this expedition. And yes, I'm starting sea logging tour now for the following couple of hours maybe. So, Andrew, as I'm just arrived here, could you tell me what happened in this time?
Sure. So I took over around 3:30, and over the last couple of hours, we've just been sort of following this area where we found the original methane seep around 6 p.m. last night. It looks like, based on what we're finding a lot of clamshells, but mostly dead. So it looks like what we've found is probably a recently expired or a extinct or dying seep in this area. And so we're trying to find some other hotspots like the ones we found earlier last evening, so that we can find a few more. Along the way, we've seen some interesting rocks, some more of the sort of blue-tinged biofilms that we saw on the loose rocks. And we found one or two spots where there was a live clam, but not quite the same amount of life that we saw in that one spot.
Nice.
But we've still got about 2.5 hours left.
2 hours.
2 hours. So.
You want me to spin around and fly this backwards so we can see where it's going and then just go?
I can just go.
Okay.
Yeah.
Hello. I think he wants to come with us. No.
Looks like it's another spoonworm.
Yeah.
Yep. Trying to charge up the phone. We hope we find a large active seep too. But there's also the dive later today, so. So we have about 2 days worth of dives left. So we're going to be doing one more dive later today for about 8 hours, where we're going to be, looking for more methane seeps. And then the last day of our dives will be going to the deepest of our sites, where we're hoping to find some larger macro scale. As a microbiologist, if I can see it with my eye, it's too big. Living fossils. So when we use that term, though, when we say something like when we're talking about living fossils, we're actually looking at, for a lot of us, we're actually interested in living fossils within the microbial mats, within the cores that we're looking at. So bacteria that species that reflect, this place in a period of time from, tens of thousands or even millions of years ago. So thinking more about that idea about contaminating Mars, we don't completely even agree how we're going to set up settlements there. So there's like one strategy is you're going to live out on the surface, and then to protect yourself from radiation, you need to dump about 3 feet worth of regolith of the surface material on top of everything. So everything will be covered in like mounds of rock. The other is actually NASA had a whole Caves on Mars project. So their idea was to basically inflate structures inside of some of the caverns in Mars. So basically you'd have kind of a protected environment. The caves are really nice because just like caves on Earth, they're kind of more temperature regulated. So you'd have kind of year-round conditions that you were kind of stabilized with. But that would also affect how you manage contamination. Kind of like there are dust storms on Mars that can last for like 6 months. So I think caves make a lot of sense. So. Well, yeah. Oh, I don't know. Yeah. So this was sort of a area that we just picked a random paper and read. So yeah, it's an interesting consideration. So the amount of ice is a lot more than what we find here for anything to get access to water, because nothing just freezes over that far down.
Big rock. Big rock.
Well, I mean, it looks like there should be some traces of stuff all around here, but it's not. Oh, wow. Well, look at all that.
It used to be dead, though.
Yeah, I think it's all. I think, but again, it's.
We were just above this, so there was nothing up there.
Okay. Oh, this is the area we were just above this area. And we're coming down. So.
It looks like it's stuck in ice. It looks like it's smaller seeps.
That's where we were.
There's a lot of shells below us now.
Okay.
No, I think so. Because I don't think that fell because it's you see here. Those are active microbial mats. So the further we come down, the more they still seem to be active.
Yeah, there's some below this as well.
Yeah.
Oh yeah.
That's fence up there, so lots of shells below us. Okay, we're going down, looking at shells, looking at seeps, looking at mats, backwards towards 6 right now. That area, and then kind of just stay low and come off.
Yeah, I thought you could do both then.
I turned it off. Well, you do cruise. So else.
So cruise and bunker.
Yeah, because the log does. That's why I see. So it's cruise starts.
So we went to 6 now.
Yeah, it's kind of backing up.
Some cool bubbles.
Yeah.
Thanks.
Thank you so much.
Like 2 hours.
Yeah, a lot of oyster shell's still here.
Morning, everybody.
Hello.
Good morning.
Hi, everybody.
Morning.
Been up all night?
Oh, what?
Everybody's been up all night?
I know.
No? Y'all got some sleep?
I got almost 3 hours of sleep.
I figured it'd be a long one once we found that seep, huh?
Yeah. We traded off.
That was a pretty good spot, huh?
Yeah. Yeah, Katie, my grad student stayed down here for a while, and then I went to bed and swapped up. So.
That was pretty much exactly what we were hoping for, huh?
Yeah. So I think anything else is gravy at this point. I think we're gonna
Like these sites that are dead or extinct, they're interesting for some people, like
Yeah.
for some of the young people. So it's great we have found this. We can see
Yeah.
this state.
No, and we collected a lot of cores and samples there, and collected a little bit of others. We saw some previous, like some extinct methane seeps earlier, and then I mean, really, this whole area does seem like it's sort of on the decline, or recently extinct seep.
Yeah, this area for geologists, this area. Hi, here Oscar Urquilla from Centro de Astrobiology in Madrid, geologist. Yeah, one of the we're looking now is very different sediments that we saw in previous dive. Even yesterday at the morning, when we start this dive, the sediments are quite different than what we saw right now here.
This is about the closest we're going to get to the red circle.
Here, there are more rocks over the surface, more fragments of rocks, and even the outcrop that we passed 1 minute ago, a couple of minutes ago, is a different rock that we saw days before. It's very interesting to take samples of this area for the interest of methane seep and the discovery of this site that is very exciting for many people in this seep right now. I think that we have 3 hours of sleep, but we can compass that with all the discoveries that the sample that right now we are sample. The information that we can recover for that sample will be incredible for us.
Yeah.
Morning, Claude. Morning. How you doing today?
No, so Oscar gave you his introduction. We're at the top of the hour, so I'll reintroduce myself. So if you just joined us, I'm Drew Palmer. I'm a professor at the Florida Institute of Technology. We're about 20 minutes south of that number. That distance keeps changing. We're about 30 minutes south of Kennedy Space Center. I guess it depends on how fast you drive. And I am on this expedition as part of the Astrobiology group. Did you want me? Okay. No, I was asking if you wanted me. I thought you said my name. No? You did not, clearly. Did Jess finally go to sleep? She's not going to sleep? Jess went to sleep?
Yes.
Okay, good. She outlasted everybody. So. Yes. Thank you. So here we are, and I am going to sign off and pass this over. Pass this over to Oscar, who introduced himself just a few minutes ago. All right. Thanks for hanging out with us. So you want somebody to be down here at 7:30 so you guys can make some good breakfast? Actually get phone up to charge.
Hi again. Right now in the lead seat here in control room. Some people need to rest a little bit to the certain moment when the ROV return to the surface. And we keep going, exploring this area, very interesting area in the coast in front of Chile. Looking for methane seep. For the people that don't know this streaming before the hours, I don't know how many hours passed from the moment that we discovered the methane seeps, active methane seeps. That is one of the main objectives of this exploration, this ocean exploration with the Smith Ocean Institute. For all the people, all the party people, all the science party and the crew of the ship is a exciting moment for found these methane ships, and we keep following other to try to find another methane ships to recollect samples, to explore, to study that for geologists, biologists, microbiologists, chemists, physics. A lot of people of different sciences, and well, there are no enough words to explain how we feeling about this discovery. With me is Victoria Vaca.
Hi. Hello, everyone.
Number, I don't know if you want to say something.
Hello. Yes, I'm still here.
The three are students, PhD candidates. Well, I'm not yet, but in the future will be a doctor. But we are very excited to be part of this scientist expedition, and we work very hard to try to do the best for everyone here.
So, yes, how are you feeling, Oscar and Ander? Like in this moment we have just discovered this place. How did you live it?
Super excited about the discovery of our first methane seep earlier yesterday.
Yeah, yesterday in time, but maybe not in our head of the hours we asleep. But yeah, yesterday. Oh, here we have another one. We can show the cells that is the main that we're looking, and all that gray, blue, white lines are microbiology mats.
Yes.
That's another clue to find these methane ships.
Do you want to put a marker here?
Yes, please. Wow. So methane ships, for a curious explication of what is that, is an emanation of methane from the underground in the oceans. This methane is used for different organisms, as the same that we have right now in the screen, to provide the food and the element to grow up. It's very important for the biology of this area, because they provide a good site to grow, to extend in a few areas and
You guys want to keep going or stop?
yeah, keep going, and maybe we can return later to one sample, and maybe the people that need to recover some sample.
Yes, the reason we are so excited about these bivalves we are seeing is that they are unique, or they only thrive in these methane-rich areas because they are living, or what I was told, they had this, they lived in some symbiosis with methanotrophic bacteria, so that's the reason they are good indicators from methane seeps, and that's the reason that we are so happy to see those little living beings. And also we are seeing a lot of groups that seem to be dead, so there are indications that they were like active methane seeps in maybe in a recent past, and maybe they are already extinct.
Una actualización quizá para los que nos estén escuchando en español. Ayer por la tarde, en hora chilena, ahora mismo son las 6 de la mañana, descubrimos las emanaciones de metano, methane seep en inglés, 2 emanaciones en las que vimos bivalvos vivos alimentándose de ese metano. El momento vivido aquí en la habitación de control o el control room es difícil de explicar. Quizás si podéis volver atrás en este vídeo nos escucharéis gritar, sonreír, abrazar, exclamar por lo súper interesante que ha sido este descubrimiento.
Y como aclaración, si volvéis para atrás en el vídeo, es cierto que no se ven burbujas, o sea, el metano es un gas y hay veces, corrígeme Óscar si me equivoco, hay veces que estos methane seeps forman columnas de burbujas, pero otras veces no, y por eso era tan complicado localizarlos, porque realmente no se pueden detectar de lejos, no hay cambios de temperatura, o no tienen por qué estar asociados a cambios de temperatura o a cambios físicos que se puedan detectar con sonar o con los elementos que tenemos en el barco.
Sí,
por eso tuvimos, bueno,
sí, es así, como comenta Victoria, no es una tarea, no ha sido una tarea para nada sencilla el poder detectar estos lugares aquí en el fondo oceánico.
Hemos tenido que ir barriendo el área y buscando una aguja en un pajar, ir barriendo el área de forma visual con SuBastian y buscando signos o indicadores de estos lugares tan especiales. Y de hecho estas bivalvos que hemos visto, que a lo mejor no llaman mucho la atención, son muy importantes porque es una especie que ya se ha descrito que vive solo en estos lugares, en estos methane seeps. No sé cómo se llama methane seep en español.
Serían como fuentes de metano o algo parecido.
Vale.
Sí.
Entonces, estos bivalvos, lo que ocurre es que viven en simbiosis con bacterias metanotróficas, por tanto se alimentan. Estas bacterias son capaces de aprovechar el metano para producir compuestos y obtener energía, etcétera, y viven en colaboración con estas criaturas que hemos visto, estas parecido a mejillones blancos, y por eso nos emociona tanto encontrar estas especies que indican que lo más probable es que nos encontremos en una fuente de metano. Además también vemos estas manchas blancas filamentosas que son biofilms de bacterias, que también son indicadoras la información que tenemos de otras fuentes de metano, sabemos que también nos están señalando que seguramente nos encontremos en un lugar rico en metano. Entonces, todas estas, además de datos geológicos que teníamos previamente, no ha sido casualidad que estemos investigando este lugar. Sabíamos, o sea, todo parece indicar que efectivamente estamos en este fuente de metano.
Sí, en methane seeps en esta zona frente a las costas de Antofagasta en Chile. Bueno, como ha comentado Victoria, no ha sido sencillo. Hemos tenido los participantes en esta expedición, hemos tenido que decidir qué áreas priorizar, basándonos en informaciones previas, también en bathymetría que se ha ido recogiendo por parte del Falcor, el barco en el que nos encontramos ahora mismo del Smithsonian Institute, y hemos intentado encontrar aquellas fracturas o lineamientos o estructuras que están asociadas a estos methane seeps en otros lugares del mundo donde ellas se han encontrado. Y bueno, esta inmersión comenzó hace casi 24 horas y durante unas 12 a 13 horas estuvimos recorriendo la superficie siguiendo estas estructuras hasta que finalmente ayer por la tarde los descubrimos y seguimos viendo bastantes de estas methane seeps en el mismo área de trabajo en la que llevamos trabajando las últimas 11 a 12 horas.
Como ya hemos ido explicando en exploraciones anteriores, en inmersiones anteriores, somos un equipo multidisciplinar y este momento ha sido posible gracias a la colaboración de geólogos, de microbiólogos que nos han indicado que efectivamente estas alfombra de bacterias son propias de estos methane seeps, de biólogos marinos que también nos han ayudado a identificar especies, así que eso ha sido un trabajo, sí, esto ha sido posible gracias al trabajo conjunto de todos los miembros de la expedición.
Los trapetes microbianos que habéis visto, de esos colores blancos, azulados, grises, también son un indicativo claro de la presencia de estos methane seeps y casi nos indican que están activos todavía. Sí que hemos localizado algunos ya extintos o que parece que ya no emiten metano, pero si podéis volver para atrás, el primero podíamos ver perfectamente como los bivalvos, los mejillones, como ha comentado Victoria, estaban alimentándose, sacando sus elementos, sus órganos filtradores para alimentarse y bueno, ha sido un momento bastante excitante de ver en pantalla cómo esos organismos estaban alimentando del metano. La dificultad, como ha comentado Victoria, es que a veces este metano no se ve como una serie de burbujas saliendo de un lugar, sino que es como una parte transparente del agua, sin poder distinguirlo, y si no hay una emanación suficiente es casi imposible de detectar por radar, que es otra de las técnicas que se pueden utilizar para intentar detectar estos methane seeps.
Y otro de los indicadores que hemos visto es la consistencia de los sedimentos, ¿no, Óscar?
Sí, hemos visto también como cambios en los sedimentos y estamos esperando a recuperar las muestras que ya hemos recogido en el SuBastian para comprobar la presencia de carbonatos. Ayer noche aquí en Chile vimos algunos afloramientos de rocas que podrían corresponderse con carbonatos que suelen estar asociados a estos methane seeps. En las inmersiones anteriores que hemos tenido en esta campaña oceanográfica hemos recuperado muestras de diferentes afloramientos, también de sedimentos, pero por desgracia ninguno de ellos hemos visto la presencia de carbonatos o al menos no los hemos podido detectar hasta el momento. Pero como mi compañera Ana de Dios, que creo que estará escuchándonos, que ahora no está aquí en esta expedición, pero ha trabajado mucho para que nosotros también podamos detectar esto, y bueno, desde aquí quería darle las gracias de forma pública por su labor y por su dedicación para que este descubrimiento también se produjera. Ana nos comentó también que otra de las posibilidades era encontrar los carbonatos asociados a estos methane seeps. Vemos también, bueno, la presencia de más tapetes, más de estas conchas de bivalvos. Y bueno, la roca parece un... It's like a sedimentary rock.
Sedimentary?
Yeah, that's a sedimentary rock. It's a conglomerate that is a formation of big rocks with a small cement, but that's interesting.
And
collecting that one clam, I think that's a different species than the one that we saw before.
Sure, which one?
The one in the back that is alive. If you can grab it with the...
It's under here.
Under here, yeah. There's one.
So what we have in front of us right now is for the look that have is maybe another kind of sedimentary rock. This named conglomerate. A conglomerate is a mass of small-sized grains with big ones, and indicate the movement of great mass of water with sediment that is deposited after a flow, for example. I thought that this is, for me, is the first time we saw in this dive. In previous dives we saw sands and clays, but the presence of this kind of rock indicate that there are big movements, or also there was a big movements of flow of water to come here with this material. The red color could be the presence of iron. Okay, you can. Okay, you can. Okay, you can see in the screen the presence of the big rocks with the wall structure with that red color, and there are the different cells. Aunque la expedición estamos buscando estos methane seeps, el poder recorrer estas áreas implica también intentar obtener toda la información posible sobre todo lo que encontramos delante de nosotros. Limitarnos únicamente a los objetivos que nos habíamos planteado sería pecar un poquito de ser poco ambiciosos en cuanto a nuestro trabajo. Por eso queremos que las muestras que recojamos obtener la máxima información posible que podamos obtener con diferentes análisis, visiones y compartir datos para hacer que la ciencia avance y que los descubrimientos que podemos obtener de estas misiones sean mucho mejores, no solo para el conocimiento, sino para el avance general de todos nosotros.
We're now trying to grab one of the clams, as it seems like it's a different species than what we've seen before.
I could but for the biologist it is going to the bio box 1A, can we?
To
One chamber A.
Chamber A, yeah. Also, this expedition, we are looking for living fossils, not only the methane seeps. The living fossils are organisms that are still looking and genetically identical at fossil that we can find right now in rocks in the continental part of Chile. And the idea of this exploration is to find these organisms because the environment is inalterated for the last 100 to hundreds of million years. So it's possible that the biota and the ecology of these areas is still the same than millions of years ago. También, como veis, estamos recolectando algunas muestras de organismos para que sean estudiados por investigadores de diferentes localizaciones en el mundo. Wow, esa muestra para que sean estudiados en la búsqueda de los que se llaman fósiles vivientes, que son todos los objetivos de esta expedición. Yeah.
Did you want to go back to the seep side, to the other one? Or?
I think we need to explore this area a little bit more and maybe later return to that area.
Sure.
Vamos a explorar un poquito más este área. Hemos visto hace unos minutos otra zona bastante interesante con tapetes microbianos y algunas conchas más de mejillones y estos organismos.
Bivalvos.
Bivalvos, perdón. Gracias, Victoria. Y vamos a seguir explorando un poquito más este área. Vemos cómo al levantarse el SuBastian del terreno sube el material del fondo, que suele ser partículas extremadamente finas, lo que a veces puede ser un poquito problemático para recopilar las muestras y ver lo que tenemos enfrente de nosotros. Cabe recordar que estas zonas están en una oscuridad perpetua, que los animales y organismos que vemos están adaptados a estas condiciones tan extremas. Ahora mismo estamos a 2.850 metros de profundidad exactamente y a una temperatura de 1,8 grados centígrados. Esta temperatura, a pesar de que hemos pasado durante casi 24 horas aquí abajo ha permanecido casi inalterada. Si tenéis oportunidad de regresar y habláis en español, durante parte de la inmersión en la que también estuvimos Victoria y yo, pero nos acompañaron varios de los investigadores chilenos que están en esta expedición, explicándonos un poco las condiciones de vida de estos animales y datos súper interesantes de lo que supone vivir en estas áreas y también de lo emocionados que estaban sobre explorar estas zonas y encontrar nuevas especies y organismos para catalogar. Vamos a recoger.
I'm
not sure it'll fit in there, but I'll leave it go. If it won't, can we put it in 3A or 3B? If it won't fit, can we put it in 3A or 3B?
Alright,
Whoa.
Rick, so you happy with the ship's position, or do you want me to back off?
Maybe you can put in
I think you're all right. Thank you, though, Jace. We're just taking a sample here real quick.
Yes.
Alright.
Or maybe Biobox A. That's the gun.
With the plug, yeah.
Yes.
3A? Okay.
Well, no, 1A.
Okay, but if it won't fit, then we can do 3, right? If it won't fit, we can do 3, right? I think it's pretty big.
Okay, well, okay, or 3.
We'll try one first.
Oh, or maybe 3B. That I don't know how big were the coordinates.
Lo que estáis oyendo es tomando la decisión de dónde vamos a meter la muestra que vamos a recoger ahora, que creo que es una esponja, esa estructura en forma de flor que hemos visto ahí, como si fuera un tulipán. Se trata de una especie de esponja. Cabe recordar que el SuBastian tiene la capacidad de recoger muestras de agua, de sedimentos, de organismos, pero también testigos de roca o de sedimentos para poder recogerlos y traerlos hasta el barco, el Falcor 2, en el que nos encontramos ahora mismo haciendo esta transmisión y explorando. Creo que el mapa por donde hemos estado recorriendo el área os sale de vez en cuando en la pantalla, pero podéis ver todo el recorrido que hemos seguido para encontrar estos Methane seeps y es increíble la cantidad de lugares que hemos encontrado hasta el momento. Lo que vamos a recoger es un tipo de esponja, ¿verdad, Victoria?
Sí. Bueno, por lo menos los expertos comentaron ayer que era un tipo de esponja. La verdad es que no puedo aportar mucho más.
Sí, pero bueno.
Sí.
Ahora mismo despiertos en este barco de la comunidad de los científicos no estamos unos pocos. Es muy importante el descansar después de todas estas largas horas para cuando vuelva el SuBastian a la superficie estar activos para hacer el mejor de los trabajos en la recogida de las muestras. Los participantes en esta misión científica procedemos de Chile, de España, en mi caso del Centro de Astrobiología en Madrid, también investigadores de Estados Unidos. Amber is from Canadian, or you are American or Canadian?
Canadian.
Canadian, okay. Another one from another country, Germany. So we have a World War party, scientist party. So it's very exciting that we can share our ideas or experience it to try to do the best for everyone here. Lo que vemos es todo el sedimento que se ha levantado, que parece que en este área es mucho más fino que en áreas que hemos explorado anteriormente, porque tarda muchísimo más en precipitar y volver al terreno, lo que hace que nos tengamos que retrasar un poco para poder trabajar mucho mejor con el SuBastian. 2 pilotos están continuamente con el submarino, controlando tanto su desplazamiento como los 2 brazos robóticos que veis ahora mismo. Bueno, ahora mismo veis solamente el derecho, pero el SuBastian tiene un segundo brazo a la izquierda con el que recogen muestras o trabajan con las diferentes herramientas de las que consta este submarino. Y ahí tenemos la esponja recogida. Increíble. Uno de los aspectos que hemos comentado ya anteriormente es que los organismos más adaptados a estas condiciones tienden a tener una coloración blanca, porque los pigmentos son caros de producir a nivel biológico y por lo tanto el reducir cualquier tipo de pérdida de energía en producir algo pues es beneficiosa para los organismos. Aunque aún así hemos visto multitud de peces, de moluscos, de diferentes especies con colores bastante sorprendentes.
Okay, it's bigger than I expected, so.
Okay.
Yes. Thank you. Yes.
Yeah. Yeah, Victor.
Yes. Well, I am reading the live chat now. I don't know if you had already commented that we are, yes, we are a team of people from different countries. We are from Spain. There are people from, well, yes, you commented it.
Yeah, I comment
Okay, well.
a few minutes ago.
Okay.
But yes, we are a multinational team.
Yes. Like right in this room, there's German, Canadian, and Spanish. But yes, we are from different countries, from different fields of knowledge. So it's great. It's and for all for us students, it's like a great opportunity to meet like a really nice
Science
bunch
and
of people.
yeah.
From the science and personal point of view. And also, we in this room we have also the incredible team from the Smith Ocean Institute, the pilots and the communication. I don't know. Is like. Well, so they are great and they make this possible.
All the crew that is in this ship is incredible with us. The technician, the chef. maybe all the people here in the ship we were very thankful with the chef. And the Smithsonian Institute provides the best that they can to do our life better and easier to our job. And wow, it's incredible to be here. Amber, what are your thoughts about this? What do you think about the be here?
On this expedition?
Yeah, in this expedition.
Definitely probably once in a lifetime experience for me, because I'm not really doing any ocean research.
Yeah.
I've been on desert expeditions before in Utah, Arizona in the US. And this time coming to Atacama Desert, definitely like a good comparison between the two sites. Yeah.
Yes. Well, as an explanation, before embarking, we spent some days in the Atacama Desert. I think it was great to see, like to be able to compare both systems.
I think we went about 3,000 meters above sea level,
yep.
And now we're 3,000 meters down.
Yes, and the air was quite desertic, but in a different way. So, and both are extreme environments where the organisms that are able to be alive and thrive in there are quite special.
And many of the team and many of the groups on this team are interested in extremophiles, so life living in extreme conditions, either super dry desert or deep ocean. So
yes, and well, we have been talking about this in the previous immersions and even in this immersion, but yesterday, the importance of studying these forms of life. These extremophiles, and they're not only for just the doing science and knowing our planet better, also for other planet explorations, for practical applications in biotechnology, in medicine, in such different fields. So yes, this is a great opportunity for us, and we hope we can share like our results with society and I'm sure we will.
And also space exploration.
Yes, yes, yes.
Where the Spanish people or this scientific party are part of the Centro de Astrobiología, where we try to do the best for the space exploration and also to find the clues for the life.
Yes. Yes, yes, it's really cool. And both environments were quite mind-blowing. Like the Atacama Desert was so arid. It's, for me, at least for me, has been the first time I have seen meters and meters of desert, like desert in the most literal way. Like there was nothing growing in there. So and now seeing this, the deep sea floor, yes, it's amazing.
Yeah, can do if you want.
Okay, we got this.
Alright, awesome.
Can we stop here for a minute and recovery as a sample of the sediments, please?
¿Cómo? ¿Oscar?
No.
Ah,
Yeah,
Oscar.
okay,
Just while you're working stuff, here, take a stand.
¿Y a dónde va Oscar?
Okay, I'll keep heading towards that waypoint and just not get too close.
Four.
Yeah, that's the coral box one.
Okay, so how do you want to do this?
Yeah, in coral box.
Yes, coral quiver it's called.
Coral
Four.
Four, please.
And using a scoop or something?
Yes, that's good.
Okay.
Oh. This is for you, Oscar?
Yes.
Yeah, so both the desert in Arizona, Utah area and Atacama Desert are used for as like planetary analogs to test technologies for other planetary explorations, and the environment are really I would say similar to Mars visually and geologically in some sense. And the Arizona-Utah area are NASA's Mars analog area that trains like astronauts on how to sample, how to operate technologies in these terrains.
Yes, that area in Arizona, I don't be there but I would like to go there to try what you did in that area, because as you explained, astronauts and technologies are proving in that area to later go to the space and explore Mars, for example, right?
In order to not trick default position. And sometimes they get off over time, especially if you hit something when it gets redone. And so it'll be working really hard to stay in the same spot. And this helps it just be smoother and faster if you do that every once in a while.
Some of the astrobiology scientists and scientists that are working in the space exploration is to looking for in Earth of the analogs of other places in the solar system. For example, Antonio, that is part of this expedition, is working in the exploration of Mars with different missions that are actually in the red planet, as Curiosity and Perseverance, and also with in other that will be launched in future years. And as Amber explained, Arizona is a good place to prove the instruments and the rovers that we launched to Mars. And also, for example, in Spain, in the island of Lanzarote, that is in the Canary Island, is another Mars analog. And other places like here in Atacama is very similar to Mars. There are some pictures that we talked in previous days. If you put the blue sky, so you transfer the blue sky to a red sky, it's probably that we can confuse with the red planet. And also as Victoria told us, the presence of no one presence of vegetable in the or any kind of plant in the desert is sometimes very amazing to show, because you can go for kilometers, we don't see any kind of life, only rocks and sand.
Ah, no, cuatro. Ah, no. So
That's the number 5, right? One. Ah, okay. Sorry.
So?
But the number 4 is closer. That's the number 5, right? Yeah.
Okay, number 5 then.
Number 5
was already open.
Okay, sorry.
Okay.
Oh. Entonces en la 5, ¿no?
Sí.
As we are waiting here for the operations of recovering this sediment, do you want to explain either Amber or Oscar?
Great, thank you.
What are you going to do with the samples? Because now we are watching just how we are taking them, but which is the work behind, or what are you expecting to do or to get with these samples?
Yeah, the goal of recovering these samples is to do description of the mineralogy and the different textures that the rocks or the grains that have, and also the different researchers that could analyze this sample for, I don't know, for example, organic molecule like Daniel Carrizo that is working with us in CAP, or another researcher that try to find fossil like Nick, that is a good guy, is to recover all the information at geology and also biology research. The main idea I have for recover this sample is to know what the kind of composition have this sediment, because as I talked before, the main elements of this area are sediments with clay and sand composition, but also we try to looking for carbonates that has a good indicative of the presence of methane sheets, but also the presence of organics and the activity of organics. Okay, thank you. We'll return to the point I marked before. Please. Yeah.
Neutral. Yeah, we got a lot of samples.
Ahora vamos a volver a uno de los puntos anteriores antes de aproximadamente regresar a la superficie dentro de una hora más o menos. El SuBastian, en cuanto empieza a volver a la superficie, cuando se aleje del fondo oceánico, necesitará aproximadamente unas 3 horas para regresar hasta nosotros aquí en la superficie. Ahora mismo estamos a 2.843 metros de profundidad.
So we are expecting to finish this long dive in an hour. So we will probably be collecting some samples before getting back the SuBastian. That will take another 3 hours after we start here, or well, it starts going up. It is right now 2.8 kilometers away from us into the deep. So yes, it takes a long while until we have it back on deck. Well, it has been like our longest dive, and it has been quite exciting. We have lived like really exciting moments. So yes, I think we all want to have our samples back.
Yeah.
Oh, sorry. We're going back here?
To the point that we marked before.
Okay, sorry.
Yeah, that point, please. I don't remember what it is. Is it?
Yeah, ROV, go ahead.
Okay.
Perfect. Go ahead.
A lo largo de esta inmersión y en las anteriores se va recopilando no solo las muestras, sino también datos de observaciones que se realizan. En este caso ahora mismo lo está realizando Victoria como aquí dentro de la sala de control, pero cualquier investigador dentro del barco, mientras está observando la inmersión, lo puede realizar desde sus propios ordenadores. No es necesario que venga a la sala de control para realizarlo. es muy interesante porque a veces se nos pueden pasar cosas, podemos revisar la grabación de estas inmersiones y poder recopilar la máxima información posible de las observaciones que tomamos.
Sí, y bueno, lo hemos comentado ya varias veces, pero el barco está lleno de pantallas. Aquí estamos, desde que nos levantamos, desayunamos con lo que estáis viendo, con las imágenes que estáis viendo. Venimos aquí a la sala de control, en el laboratorio también estamos viendo continuamente las imágenes que nos manda el ROV. Así que sí, de hecho, hasta algunos estoy seguro que hasta sueñan con
Sí,
estas imágenes.
Te lo puedo asegurar.
Así que está siendo intenso, pero muy emocionante, y eso, sentir que somos los primeros en explorar este área, en poder describir e incluso recoger muestras es impresionante y merece la pena el esfuerzo, y la verdad es que y poder compartirlo también con vosotros, con la audiencia, y poder enseñar estas imágenes al mundo, la verdad es que hace esta experiencia súper especial.
Como ha comentado Amber, es una experiencia única en la vida, prácticamente. Supongo que si lo haces muchas veces a lo mejor lo tomas como una rutina, pero para nosotros, para la mayoría de nosotros, es nuestra primera vez en un barco oceanográfico, y yo creo que pocos vamos a tener la oportunidad de volver a utilizar un submarino como el SuBastian, una máquina impresionante para realizar ciencia bajo la superficie del mar.
Y bueno, eso, si seguimos según lo planeado.
Sí, seguimos con la misión planeada de regresar a la superficie dentro de aproximadamente una hora, un poquito menos de una hora. Ahora mismo lo que estamos haciendo es regresar a una de las zonas que hemos...
Afloramiento.
Sí. Que hemos visto con anterioridad, para tal vez recoger las últimas muestras del día, y bueno, del día, más bien de esta inmersión, porque del día llevamos prácticamente casi 24 horas de inmersión, y vamos a ir a ese punto para recoger las últimas muestras y regresar al barco. Thank you.
¿Eso es tapete?
Parece una
roca, ¿no? ¿Podrían ser carbonatos?
Me ha resultado sorprendente ver ese color así blanco sobre el negro de la roca.
¿Y lo vas a coger muestra? O...
No, vamos a continuar al punto, simplemente era una pequeña observación.
Vale.
Just, what we doing here is like explore another planet, a very strange planet, to find extraterrestrial organisms. Many of the organisms that we have here are like something that is for the mind of Lovecraft, for example. Very strange animals with forms, morphologies, properties, and organisms that for my geology brain is growing up to solve these animals and the diversity of morphologies. Bueno, lo que veis, esta zona ya la hemos visto hace aproximadamente una hora, hemos pasado por ella, y lo que vemos son las diferentes conchas y parte del tapete microbiano que hemos localizado en otros Methane seeps.
duda, Oscar. Esa roca que tenemos ahí en la parte superior derecha, que tiene como una mancha verde, me imagino que será deterioración.
Sí,
¿Es de la propia roca o es de...?
esa morfología, bueno, esa
Color
coloración,
verde.
sí, esa coloración la hemos
Ajá.
visto ayer, también la vimos.
Vale. Ujú.
En parte podría ser el color original de la roca, y lo que vemos, la parte rojiza y negruzca, parece ser más la alteración. Pero a veces es complicado el ver con exactitud las características de las rocas así desde un vídeo. Siempre los geólogos nos gusta más tener la roca presente para verla con detalle. Victoria se está riendo porque creo que ya sé lo que está pensando. Lauren?
Yes.
Lauren, do you want to take something here in this area?
Yeah, I would. Can we take a tour here, capturing this white microbial mat on the top? Yeah. Thank you. Sorry if I sound really groggy. I slept for about 2 hours in the last 24.
So what are you going to take?
Wow. The whole thing?
Well, not the whole thing. I'm gonna break this center of it.
Yeah.
Thank you.
Bueno, lo que veis ahí son esos filamentos que se tratan de relaciones microbianas de organismos, junto, bueno, todas esas conchas de bivalvos.
Estamos hablando de core.
Correct.
13.
Number 13. Core 13.
Not 13?
13. Yes, that one. 13. 1-3.
A rock or something?
Okay.
I think they are interested in the top
It's only
layer,
the top, so it's okay.
it's okay. I'm gonna be careful because the sediment is loose, so yeah. It's not. No, well. Oh.
Bueno, veis que no es fácil tomar muestras de estos tapetes, porque el material que tiene por debajo está muy particulado y tiene una porosidad increíble, lo que hace que...
This is also like very
Yeah.
flat, so maybe you can try in there. This part is not quite.
Maybe, well, okay, we try there.
Or maybe if that doesn't work, this one is also like
Yeah,
very.
yeah.
What? Yeah.
El material que hay por debajo está como muy particulado y tiene mucho, está como muy levantado, seguramente por haber estado pasando por esta zona todo ese gas metano, lo que complica bastante la toma de los materiales de la superficie, tomar un testigo como el que estamos intentando tomar ahora. Lo que habéis visto nos ha pasado ya en otros de los puntos.
Oh. Yes, we are
Maybe with the
seeing.
scope. Yeah.
Yes, we are seeing it's very difficult to take samples from here. The sediment is loose, so whenever we try to, maybe we can ask Lauren. She's not here. Lauren, if you are listening to us, please come to mission room.
Meanwhile, I can try here one more time.
Maybe. Okay.
What's going to work here, though? Is that? Yeah, we just tried using cords, but it's like rock immediately under this. So I think we're going to end up either doing suction sample or a scoop.
Such sample is probably no-go.
You would come back and do a 40 after one for me? I don't mind. Yeah, sure, go.
You want to try to scoop it? You want to try to scoop it?
No, well, she said, if it's possible to do it over like angle, maybe it's easier.
So we want to make sure we get some of the mat. We want to make sure we get some of the mat. And just as long as we get it in the core with mat, however I do it, right?
Yes. So, yes, you can see we are having some troubles to take these kind of samples. The sediments are very loose, so whenever we try to take them with the core, we are like as we try to push out the core of from the floor, the sediment just fell out. And we want to still trying to take them out with the core because it's the best way. We have to maintain that mat so we can recover those bacteria after in the lab. But yes, we have to. We are going to try and see if we are lucky and we can get those lovely bacterial mats. And yes, it's like exploring another planet, and it's incredible. Thank you to the audience for the support. We really appreciate that.
It's all rock. It's just all rock.
Oh, yeah.
We got some of them not just coming out.
No.
Yeah, so there's no sediment here.
Yeah.
It's just rock with a film on it.
Yes.
Yeah.
I'm going to get Lauren to see which.
Lo que vemos aquí y lo que nos está tomando tantas dificultades para tomar un testigo es que además de que el material está muy suelto, la roca está muy pocos centímetros bajo la superficie.
Yeah, there's no sediment here. It's just rock. It's rock with a film of mat on top.
Can we try to get it in a sediment bag?
Yeah.
Okay, let's do that then. Thank you. Is what it is. You guys are awesome.
Allow me to get rid of this so that we can do that. Look up for me. Okay, so the bag's already been used. So the bag has already been used. So I can scoop it and put it into either a quiver or into a bio box.
Okay, which one will be the best to preserve? Like
Well, we can get more into a bio box, but the quiver is going to be sealed.
okay, I think the quiver maybe.
Quiver?
It's-
Okay.
Yes. So because if it's sealed, it won't be
It won't be
moving
floating
as.
around. Yep.
Yes. Okay, the quiver.
Alright.
Thank you.
Lo que vamos a hacer es intentar recoger la muestra con una de las herramientas y meterlo en uno de los...
Yes.
Vamos a intentar succionarlo para intentar recuperarlo todo el material que se pueda.
For the coral quiver 4,
Yes.
7, 6, 8. So which one is?
We're going with 4.
Okay, number 4. Okay.
Como podéis comprobar, no es siempre es fácil poder obtener las muestras que queremos y hay que tomar alternativas para poder tener una muestra representativa de lo que queremos.
guess I'll get another one as well.
Okay, yes. Thank you. Well, we are trying to take the bacterial map. It's not been an easy task, I hope, and I'm sure we will get some. Thank you for the team. The SOI team. I'm sure we will get material to work with.
Ahí tenemos el tapete microbiano que queremos recopilar. Esperemos que caiga dentro y ahí está. It's possible to take another sample with the same mold, but another coral river?
Another or the same?
It's got plenty of space. It's only like that full.
Yes. Well, I guess.
Because it's for Armando.
Armando quiere otro.
Yeah, no, for Aparna.
Okay, well, we can take, because we have enough corals.
For the Aparna.
Vale, okay. Well, we can take more material in the same river and then more material in another one.
Yeah.
Yes. So, yes,
Try
I think
to
we can do
recover
that.
the more quantity that we.
Yes. Yes, I think with two coral.
Yeah, 2 coral.
So another scoop in the same one, and then more in the second one.
Yes, please. Thank you.
Okay.
Vamos a intentar de esta área recuperar todo el material que sea posible, porque os recuerdo que dentro de aproximadamente 30 minutos vamos a iniciar nuestro ascenso de nuevo a la superficie. We'll try to recover all the material that we can, because in the next 30 minutes at 8 o'clock here in Chile, we will go to return to the surface. That white gray material is organic mats that is very interesting and related with the methane sheet that is the main objective of this dive. We found it in approximately 12 hours ago, and we are very excited to this discovery. The researchers that know most of these methane sheets say that this is probably the deepest methane sheet that is discovered in the coast of Chile.
no more from the same place, or do you want to try to find another spot?
Other sample similar to this in the number.
Yes, maybe in another place. Maybe this has been.
From here as well, or from a different?
Yes,
Okay.
from this
In this place.
place.
But maybe we have to find like
In
I don't know how it's like the state of this.
maybe
Oh
here.
yes. Yes, I think.
Okay.
Vamos a estar un tiempito más aquí para recoger otra muestra muy similar a la que ya hemos recogido ahora.
What's that? So, 6. All right. All right, so from where? Do you want to get at the top? All right.
Yeah. Thank you.
Yes, the so I think this one is oh,
I already have 2 cores.
Okay,
I can, if
we
you
can save that and forward if any
Sí, exacto. Yeah. Should have did it like push it like wherever.
El equipo aquí está haciendo un trabajo magnífico para recoger todas estas muestras de organic mats, de tapetes microbianos. Ahora mismo la gente en un grupo que tenemos de trabajo está bastante excitada, pidiendo muestras para recoger antes del ascenso, que están más o menos planificado dentro de 20 minutos. Thank you very much. You're amazing, guys.
The small white crabs and spot lobsters.
Yes, we haven't seen before.
There's no way.
We can try for a while.
So I can't use the suction zapper because there's a squat lobster and a hermit crab already stuck in there, so it won't work. And I can try.
Yes. Well,
I'll try it.
Oh,
I'll try it.
oh, got him. Got them both.
Oh,
suction
oh,
zapper.
there it is.
Oh.
Okay, we can use the suction zapper.
Yes.
Oh, yeah.
We can, yes, they want some more of those, so if we can try, of course it will count. But yes.
Bueno, para que os hagáis una idea, aunque en la retransmisión estéis viendo las imágenes, en realidad tenemos un montón de pantallas a nuestro alrededor con datos técnicos, de análisis, datos del propio SuBastian, pero también cámaras de donde están las muestras recopiladas hasta el momento, incluidos los bioboxes, unas cámaras donde los organismos que se han recogido vivos, o por lo menos lo mejor posible, podemos comprobar dónde se encuentran para ver si hay espacio suficiente para recogerlo. En uno de estos bioboxes hemos visto uno de los animalitos que todavía sigue vivo, por fortuna para nosotros, quizás no tanto para él cuando llega aquí a la superficie. Lo que vamos a intentar ahora es buscar algunos organismos más para su recogida y posiblemente un testigo de sondeo más para el grupo de geología al que pertenezco. ¿Crees que encontraremos a los cangrejillos, Victoria?
A ver, espero que sí.
Los animales aquí, aunque no tienen visión, son escurridizos a veces.
Sí, pero bueno, contamos con un equipazo, la verdad. Aquí la gente de la institución. Y la verdad es que da gusto trabajar con ellos. Como podéis ver, es increíble. Les pedimos algo, señalamos y nos lo dan, así que confío en pescar algún. Bueno, cangrejo está más emparentado a las langostas. Bueno, ahora estamos buscando. No sé cómo se dice exactamente.
¿Qué? ¿Langosta?
El cangrejillo cronaps. Bueno, creo que no.
Puedes decirlo en inglés.
Sí, creo que no lo voy a poder decir en español.
El método, como se trabaja aquí en esta sala de control, bueno, hay 2 pilotos que controlan en todo momento el SuBastian y un personal adicional del instituto. Y luego, aunque me escuchéis a mí que como líder, entre comillas, de este proceso, también está Victoria, que la estáis escuchando, recopilando información de todo lo que se ve en este descenso. También hay espacio para que otros investigadores estén aquí o incluso puedan participar con nosotros en este stream con opiniones o respondiendo a alguna de las preguntas que les queréis hacer. Ahora mismo la actividad está un poquito baja, dadas las horas que son aquí en Chile.
¿Cuándo iniciamos esta inmersión?
Esta inmersión se inició hace 24 horas. Llevamos 24 horas de inmersión. Creo que llegó aproximadamente a esta misma hora llegó el SuBastian al fondo oceánico y
Tarda unas 3 horas en recorrer la distancia entre superficie y fondo oceánico, para que os hagáis una idea. No es algo inmediato.
Sí, así es. Y bueno, nos quedan otras 3 horas hasta que podamos, no vamos a decir tocar con nuestras manitas, porque si no la contaminación sería bastante alta, pero sí para poder tener con nosotros las muestras tan interesantes que hemos recogido a lo largo de todas estas horas.
No veo
No.
ninguno, ¿no? So can we look around? I don't think.
Yeah.
I think we saw some before, but I'm not seeing any. Oh, there's one there in the left.
Big guy?
Yes, yes, there's.
The rock?
Yes, maybe.
Yeah.
We can try to catch that one.
Oh, you had there one? There's one small one right there.
Yes. Mm-hmm. Well, the one you think it's easier to catch?
Neither one would be easy.
Yes.
Yeah.
Oh.
Wow. Here the sediment are more fluffy, more green. As you see, when we try to recover the samples, the core samples in the material, the mud, we saw that it's too hard for us to recover it because the rock is so up in the to the surface. And as you can see right now, the small movement of the SuBastian make that the sediment is take off and cover all our vision right now. Como habréis visto, si nos lleváis unos minutos viéndonos, hemos intentado recuperar unos testigos de sondeo en el área para recoger parte del tapete microbiano, pero hemos visto que ha sido imposible hacerlo con los testigos de sondeo, y lo hemos recogido con una herramienta que tiene el SuBastian. El material, como veis, es sumamente particulado, muy fino, y leves movimientos del SuBastian hacen que se levante muchísimo material. Es algo que en las inversiones anteriores no hemos visto con tanta claridad. Sí que con movimientos del SuBastian el material se levantaba, pero se depositaba muy rápidamente. Ahora podemos ver cómo este queda durante mucho más tiempo suspendido enfrente de nosotros, y que nos impide tener una visión súper clara de lo que tenemos enfrente. Vamos a intentar recoger esos cangrejillos que veis ahí.
It's a difficult task. They move really quickly, but it would be interesting. We want to check if those what lobsters are characterized, like if that species is like a methane seep related. And yes, we have to take them in order to determine which species are they, and also there are some Chilean colleagues that work in the museum, so they want to collect samples so they can show people how rich is their deep sea oceans. Well, right now we are having problems with sediments. We have to wait until they are a bit...
A sediment fog?
Yes, they are a bit set. Yes, well, we can see So, yes, as Oscar said, probably this sediment consistence that is very like fluffy. It's a characteristic of these methane-rich places. So, oh.
Victoria?
Yes.
Are you happy with all of them in the same jar?
Yes, I think.
Yeah.
Okay.
So the
So-
squat lobster. Say they came through squat lobster.
So did we? So we got the.
We got one of them.
Yeah. Right. Okay. One moment. I didn't. So it's number 4. Wait. Sure, I didn't. Okay. Section 4. Okay.
We didn't put much reflection. We danced around the block. Yeah, he was to the right. He was to the right underneath him.
Yeah,
But- Yeah, I'd say, but he was looking at it. No wonder he's still using the
One question in the chat asking if this is a ROV or ROV or managed submersible. This is a ROV. We manage this submarine from the surface right now in the ship called Falcon 2. And this dive that is taken the last 24 hours.
forward a little bit. Oh, really rough. Can't get that anymore.
Whoa. Bye bye, little crab. Now, Lisa, this will be our last samples of the day of the dive, and we try to go to the surface in a few minutes. Yeah. And I can see, I can hear you, Victoria. I don't know why.
Ahora sí, ¿no?
Yes, ahora sí, ahora sí.
Yes, the idea is to come back later in the day to the same site. Oh, look at that.
Oh.
Yeah.
A little fighter.
Oh, come on. Oh.
Come with us.
This is... Maybe it's...
Just stop.
I think we got it.
Yes?
Yes.
Pero ¿ha caído en la jarra ya?
Creo que sí.
Okay. Great.
Fantastic. We have a couple of traps with us. 2 new friends.
I don't think that is it.
Oscar, falta el core de... ¿Y si vemos algún raquiopo? Thank you.
Now, if we can go to a lower part to the canal core, and we found that there are more sediment.
Stand by.
Yeah. I know.
No.
Oh,
Yeah.
there are little friend there.
Oh.
Finally escaped of us. Oh, a real fighter.
So we got one of them?
Yeah, we got one.
Oh,
Well,
how escape?
He escaped. He's too strong. He's too big.
Still, like you were able to grab it?
He's that big.
Yeah. Well, he gained another day of life. Ahora vamos a una zona un poquito menos alta donde podamos encontrar una mayor cantidad de sedimento para tomar una muestra final para el grupo de geólogos que trabajamos aquí en el SuBastian, en esta expedición, y esa posiblemente sea ya nuestra última muestra de esta inmersión, y para regresar de nuevo a el SuBastian regrese de nuevo con nosotros aquí en el Falcor 2 del Smithsonian Institute. Lo que veis ahí, esas partes blancas, son las conchas de los bivalvos que hemos visto relacionadas con los methane seeps, que ha sido el objetivo principal de esta inmersión. Aún así hemos recogido también algunas muestras de organismos, sedimentos, muestras del suelo y del sustrato rocoso.
La idea es volver después a este mismo sitio. Otro.
We can try. I'll finally check a score.
Okay. I heard the suction samplers, the claws, no? No?
So with number 13.
Yeah, but first we will try to pause and do that.
Ah, sí.
Sí, sí.
Vale.
It says on right now, right? Yep. Just be a little bit of a zoom, not too much.
Okay. Little guy. Just
The scan with a...
yes. Yellow. Oh, yes. Oh, come on. No. Let's see. We can see. I don't know if you have that on screen. We can see the jar, so we are able to see if the samples are entering. Oh, there.
Oh, it's there. It's
Yes,
there.
it's
It's
there.
there.
So yes, we can see
We
if the
we check it.
sample
We check it.
into the jar. So. Perfect. Thank you.
Thank you very much, guys.
Yes. And thank you to the audience for the support.
Yeah, we try to an area that is with more sediment, maybe a lower altitude. To final core.
Is it inactive score?
Yeah.
What's
For a stone that is nothing there.
okay.
To compare with metabolism.
Os entendéis entre vosotros.
Yeah. Victoria is asking why we take this sample. It's only for have material and we can compare with the cores that we take in the methane ships. The geology group in this dive take 3 samples, three cores, sorry. One for fossil methane ships and 2 more inactive methane ship, or also how it's looking like an active methane ship. Right now we descend to another zone where there are more sediments to have a sample to compare with these cores.
So, Oscar, ¿podéis usar 14? Porque como parece lo han usado antes.
Bueno, Guillermo ha aparecido aquí por la sala de control muy contento porque hemos recogido un par de cangrejillos para ellos. Así que... This song look great.
Entonces el pushcore 14, ¿vale?
We use the number 14, please, because 13 was used, but it's no...
Yes.
14.
Yeah.
Yes, please.
Yeah.
Sí, entonces está marcado. Sí. Este es para Oscar and Dan.
Geología y Dan. Vamos a intentar tomar aquí un puscor, uno de estos testigos que veis ahí ahora mismo. la idea de este material es compararlo con los otros 3 testigos que el grupo de geología ha recogido en esta inmersión. Los otros 3 testigos han sido recogidos en lo que era un methane seeps que parecía ya fósil, que ya estaba inactivo por la cantidad de conchas y no ver presencia de ninguno de estos bivalvos vivos. Y los otros 2 han sido recogidos en 2 zonas con methane seeps activas, o por lo menos donde parecía que había mucha más actividad. Principalmente el primero de los methane seeps que localizamos, donde se veía perfectamente como los bivalvos estaban abiertos, con sus orgánulos fuera para realizar sus funciones de filtrado del agua. Veis aquí que la zona superficial es bastante material bastante suelto, como hemos comprobado en otras zonas de esta área. Oh. Okay. So number 15 for me. And a final core, the number 15 in the same area.
Oh, what?
Number 15 in the same area, another one, please. Thank you.
15?
15. 1 5.